Chapter 5, Section 4. The surface of the protoplast, its properties and its architecture. There is no problem in cell physiology more challenging to investigators than the conditions allowing dissolved substances to enter the protoplasm or to leave it. This problem has occupied the general interest of physiologists since Noaitzelai's discovery, 1855, of the plasmolysis and deplasmolysis of plant cells afforded a number of botanists, Sefer D.C. Vries, Plebs, and others an easy way to measure the osmotic forces of the living cell and the permeability of the protoplasmic surface to solutes. After enough data regarding the osmotic properties of plant cells had been collected, Overton in 1895 he arrived at the result that, for innumerable organic compounds, there are certain general rules correlating their physical and chemical property to penetrate protoplasm. Pound 31. The picture, as drawn originally by Overton, has lost much of its plainness, as refined procedures have increasingly revealed the fact that genus and species specificity is characterized not only by morphological features and by the elaboration of specific chemical products, but also by physicochemical properties, such as differential permeability. Though many irregularities and deviations from Overton's general statements have been encountered in the course of years, while research on permeability was being extended by the invention of new methods. Over the entire field of plant and animal life, the principles referring to organic non-electrolytes are still fairly sound. Overton concluded from his results that the rate of penetration of organic compounds is determined by their solubility in the lipoids assumed to be present in the protoplasmic surface. However, the existence of lipoid insoluble, though readily ionotrating organic compounds on the one hand, and on the other hand, the intricate behavior of electrolytes caused by their dissociation into ions and by the electric forces of the latter, have called for complementing Overton's lipoid theory by a pore theory. Both of these concepts have suggested schemes to represent the architecture of the protoplasmic surface, and experiments on models as well as on natural objects. To test their adequacy, one plant cells, the lipoid pore theory. The basic results regarding permeability to non-electrolytes have been obtained by studying plasmolysis of plant cells. When the cells are placed in solutions of an indifferent substance, plasmolysis appears if the concentration is increased to a limiting value. Impermeability to the substance is indicated if the plasmolysis can be persistent over a long period of time, and if it can be succeeded by deplasmolysis when the concentration is diminished below the threshold value, as, for instance, with hexaces or hexites, the limiting concentration C(XF rest) in mol dl is a measure of the osmotic pressure of the plant cell, which resembles an osmometer provided with a semipermeable membrane. For a substance to which the cell is permeable, a higher molar concentration, Cl, is necessary in order to start plasmolysis, which, however, disappears after a shorter or a longer period of time, depending upon the rate of penetration. Some substances enter so fast, for instance, many monohydric alcohols, that plasmolysis cannot be observed even with high concentrations. Thus, CIAC, the best procedure for an exact study of permeability is certainly the chemical determination inside the cells of the compounds in question. This can be done with sufficiently large plant cells where a voluminous cell sap, vacuole, is surrounded by the mantle of living protoplasm. Collender and Barlerm used single cells of Characeratophylla, 1 to 4 cm, long with 10 to 20 millimeters, cell sap E, in solutions of a great number of different organic compounds. They followed the entrance into the sap by analyzing samples at intervals with Bang's method of chromic acid titration, and by microchildol until equilibrium was reached, as indicated by a final concentration in the sap 90 to 100% of the outside concentration. If the compounds enter by diffusion, their movement should fulfill Fick's law. See what? Q the surface area of the regularly shaped cylindrical cell, C the inside concentration, at the time see the equilibrium concentration, and P the amount in mol of the substance, entering in unit time across unit area along unit concentration gradient. P was found to be constant. In other words, the penetration actually appeared to be a passive diffusion across the protoplasmic layer into the sap. By determining the time t for half saturation of the sap, C equals convenient relative values of the J Hermitian, this series of the determinations of cell permeability to 45 substances, the most exact so far made, is in good agreement with earlier observations of Barlunda upon plasmolysis of cells of Roa ducality, despite the two plants being very different in size, in their physiological anatomy and in their living conditions. Furthermore, the results correspond to the data regarding the permeability of OX erythrocytes, which were obtained by Heading, applying a freezing point determination, see later B. 238, and to some extent they are comparable to permeability measurements of Stewart and Jacobs upon sea urchin eggs, CP 241. Finally, all of them give an illustration of the old general conclusions drawn by Overton from his numerous experiments on plant and animal cells, Table 12, rapidly. Monohydric alcohols, aldehydes, ketones, hydrocarbons, and their mono-dithiontrihalogen derivatives, esters, many weak organic acids and bases. Less rapidly, dihydric alcohols and amides of monobasic acids, slowly. The trihydric alcohol glycerol, urea, thiourea, more slowly. The tetrahedric alcohol, erythritol, most slowly. The hexahedric alcohols, sugars, amino acids, and many neutral sales of organic acids. It appears that introduction of polar groups, hydroxyl, amino, carboxyl, decreases. That of nonpolar groups, halogens, alkyls, increases the penetrating capacity. Further, in homologous series, for example, of the aliphatic monohydric alcohols or the aliphatic monocarboxylic acids, the permeation rate rises as the length of the C-atom chain rises. It is well known that the experimental results suggested to Overton the idea of referring the differences of permeability to a differential constitution of the surface layer, or the plasma membrane. It was obvious that an interpretation cannot be given on the basis of a chemical correlation between the permanent and the membrane component. Hence, the physicochemical hypothesis of a membrane component with selective dissolving power was proposed. An organic fat-like lipoid solvent, for example, olive oil, seemed to be an adequate model, and it was ascertained by numerous studies that the distribution coefficients oil, water, or even ether water correspond remarkably well to penetrating power, as is obvious from the aforementioned studies of Exlander and Barlund. Illustrated by Figure 23. In objection to the lipoid theory, it has been pointed out that a state of equilibrium as represented by a distribution coefficient can hardly be conceived of as the crucial factor governing rate of penetration. However, see Collender and Barlund, Locke, Sitio. It must be kept in mind that, according to the lipoid theory, the process of penetration consists in, first, the distribution of the penetrating substance at the external phase boundary of the lipoid layer, second, the migration across this layer, and third, another distribution in the inverse direction at the internal phase boundary bordering the protoplasm, the two distribution coefficients supposedly being identical. Then, the second step of the process, the diffusion through the lipoid layer, can be assumed to be the slowest, but in its speed parallel to the distribution coefficient of the substance. Urea, for instance, can enter the cells, though it is practically lipoid insolubly. Below the lines are compounds of an especially high lipoid solubility, for instance, the strongly alkylated ester triethylceirate. These pass into Cora cells more slowly than could be expected theoretically, possibly due to their high MV. A search in the field of giardiate cells revealed sulfur agabegetone, in which plasmolytical study showed that the predominant ahylicol as indicated by higher plasmolytic threshold concentration. See method of Lepeshkin and Trebendel. 229 corresponds fairly well with lower values of MV. In other words, the plasma membrane resembles a molecular sieve with pores allowed in the dissolved substances to penetrate according to their diameter. Plasmolytic threshold concentrations, standing in the table in the wrong order, however, often display by their partition coefficient ether water a relatively great lipoid solubility, just as inadequacies in the lipoid theory gave rise to the pore theory. Faults in the latter point back to the former. Pore theory has been based upon various model experiments with sieve-like membranes, such as the copper ferrocyanide membrane or collodion membranes, varying in average pore size according to their preparation, or membranes of hydrophilic colloids apt to swell or to shrink, Rulin and Hoffman, distribution coefficient. Ether, water. It is of physiological interest to note that the pore size of natural and of most artificial membranes is not uniform, as it is with several porous minerals, for example. Chabacite, which, due to irregular lattice-like crystalline structure, can absorb the molecules of a homologous series of gases only up to a limited 3F, for example, ethylene, but above this limit are entirely in excessi L-butylene benzene in ordinary membranes. An assortment of graded pore diameters seems to be present. The widest pores open only to the largest molecules, which therefore traverse only a restricted part of the total sieve area, while many more pores can be utilized by the smaller molecules. This makes it clear why the rates of diffusion for substances of different NVIE, the amounts in moles, passing along a certain gradient, can differ more widely than coefficients of ordinary free diffusion, or of diffusion through the water-filled channels of a sieve membrane with wide meshes. Callender and Barlund have proposed to fuse the two theories into one, the lipoid sieve theory. They picture the structure of the plasma membrane as a mosaic composed of lipoid areas and sieve-like areas, either one or the other exerting the greater influence. For instance, in Cara, due to the prevailing effect of lipoid, methylura enters faster than urea, whereas in Vegetoa, due to the dominance of porosity, or Ka-fact than methylura. This is no more than a very crude picture that needs a giant deal of modification based on a more intimate study of the spatial relations between the molecules and submicroscopical structures. On the one hand, see chap. 8. And of the chemical differentiation of the membrane components underlying the species-specific differences in permeability, on the other hand. A. Special qualities of the lipoids as factors in permeability. The lipoid theory postulates an organic solvent, the dissolving power of which, compared to that of water, parallels the permeating power of the solute. Overton and others have done a great deal of work in the attempt to find a generally appropriate solvent. The most careful investigation of Callender and Barlund showed that in this respect the classical lipoid model, Ulf oil, gives a surprisingly good result, but is not an ideal solution of the problem. It became more and more clear that there is no one lipoid which fits all conditions. Rather, the oil-like solvent must be assumed to undergo slight variations, either of a chemical or of the physico-chemical nature, to account for the exceptional permeability of certain cells. It seems to LE of particular importance that the LEUoid used for a model, whether it be oline or another oil or natural lipoid like lecithin or another phosphatide, easily hydrolyzes slightly to form a weak acid. This behavior affects their dissolve-in power toward those so-called non-electrolytes, which in the narrowest sense are actually electrolytes having the character of weak acids or weak bases. This idea is suggested by observations of vital staining in Paravecium by Nierenstein, who showed that many dyestuffs enter the living cell, though insoluble in neutral oil, and that a congruence with the lipoid theory can be achieved by referring the staining power of the dyestuffs to their solubility in a mixture of oil plus oleic acid plus diamylamine, the acid component of the mixture providing solubility of the basic stains, the basic component of the acidic vital stains. This suggests the interpretation that a similar mixture of several components may be present in the cell surface. A stronger argument for such an assumption follows from the discrepancies frequently observed in the order of the permeation rates of the organic non-electrolytes. When one compares the behavior of different species, Wilbrandt, Collier, and Barlund, Locke, Sitt, these authors have discovered that there are certain species of plants which give preference to the entrance of non-electrolytes containing NH2 glucose, and other species will, in contrast, prefer compounds that lack this basic character. For instance, char A is more permeable to melanamide than to erythritol, more to urea than to glycerol, more to propionamide than to ethylene glycol. But with RUO, the succession goes in the inverse direction. Correspondingly, the distribution, olive oil plus oleic acid, water compared to olive oil water, is remarkably greater with melanamide, urea, and propionamide than with erythrotoglycerol and glycol. Following the aforementioned interpretation, this would mean that Cara has a relatively acidic surface solvent, ROAO a relatively basic. Furthermore, Jacob studying erythrocytes, CP 240, from a large number of species of mammals, reptiles, and fishes with regard to their permeability to urea and to ethylene glycol and glycerol, has found that, in general, with mammals and reptiles, the rate of entrance of urea far exceeds that of glycol, while with fishes the contrary is true. Correspondingly, H.D. Fiedler, on the basis of numerous observations with plant cells, found urea to penetrate faster than glycerol in almost any of them, which he accordingly designated the urea type, while a few, such as ROO, were more readily permeable for glycerol and were designated the glycerol type. The complexity is still more enhanced by observations that, within the same species, either one or the other type shows up, showing, depending upon the season or upon the age or upon the site of the plant body, supplying the experimental objects. It seems reasonable to refer these variations to a more acid or a more alkaline reaction of the e.g. rhodoplasm according to the metabolic state of the plant. But whether all these phenomena can be summarized alike as due to chemical or physicochemical variation in the lipoid components of the cell membrane is by no means proved. One point at least should be stressed in this connection, viz. The majority of these experiments, for reasons which will be explained later, p. 287, deal with substances whose low lipoid solubility and rather poor correlation between lipoid solubility and permeability favor the concept of their passing across the pores. Then one would have to account more for a chemical affinity proper, instead of a solution affinity, between the membrane material and the penetrating substance, both of them reacting with each other as acid and base. Special kinds of porosity in relation to permeability, the Cara experiments of Callander and Barlun have led to the hypothesis that, beside the prominent influence of lipoid solubility, a diffusion through pores KHB flare DC shot. 62. 355, 1934. 60, 53, 1932, 66, 133, 1937, plays a distinct role, which is evidenced by the fact that compounds with a relatively small volume penetrate the L's faster than could be anticipated from their lipoid solubility. In other words, while the TUK route is accessible to all the Lioid-soluble substances, irrespective of their MVA, the pores between the lipoid areas afford a second means of entry to substances with a low MV. The next question is whether, with other plants than Cara, the role of the elasma membrane as a mediator of the intake of non-electrolytes is divided into two parts in a similar manner. It has been shown before that Vegetoa is, so to speak, the reverse of Vihara, since its permeability is almost exclusively a pore permeability, though evidently including some lipoid effect, CP 283. An analogous behavior is displayed by epidermis cells of Gentiana aspermiana, and by the diatoms Melacera and Licmaphora. However, there are numerous connecting links, the analysis of which is worthwhile in order to throw light on the many possibilities for satisfying cellular requirements. Porosity can be expected to influence permeability in several ways. First, a variety of widths of pores has been observed by looking for the limiting MV of the lipoid in soluble substances able to titimetrate. For example, the threshold substance for Vegetoa is for ROO saccharose 70.35, for the mushroom Saliote melanome 22.9. Second, the G-ORS areas in one membrane may contain a great number of narrow J, whereas in another a small number of wide pores. though the sum of the single pore areas available for diffusion may be the same. This does not mean that the ratio of the penetration constants of two substances traversing these membranes will be the same. The ratio will be smaller with the membrane containing the wider pores, larger with the other. It seems to be indicated by the unusually small differences among the penetration constants with the diatom molecira, which for other reasons as well is believed to have wide pores, Markler locks it. Third, the relative extent of lipoid and porous surface in a membrane may contribute to the nature of its permeability. Into the root cells of lemna, the voluminous molecules of methylurea penetrate more slowly than those of urea, as though porous areas were the larger and more influential portion of the membrane. In Cara, the lipoid effect is predominant. See also p. 235 fourth in a study of the electrical properties of the surface membrane of Nutella. Curtis and Cole came to the surprising result that only 13% of the surface area is involved in the considerable increase of permeability to ions, decrease of resistance, concomitant with excitation, see Sec 5. In other words, 87% of the area should be considered a rigid skeleton-like substance. Comparative physiology of permeability, the quantitative analysis of cell permeability as a passive diffusion-like process has resulted. So far, A2K Hefler Bergis 5, 1934. As non-electrolytes are concerned, in the isolation of only two chief factors functioning jointly to separate solutes from solvent in the fluid surrounding the cell, first, selective dissolving, second, selective mechanical separation. As a third factor, evidently, a chemical separation can be added, referring to basic or acidic properties of the solutes. More factors may be segregated, as will become evident, for example, in the following section regarding electrolytes. So far, so many variations of each of the factors have already been surveyed as to give a fairly embarrassing picture. However, at least under certain restricting conditions, a rather Cernulae pattern appears, as shown, for instance, in Figure 24, CP 238. Calandrin has compared the cell permeability of 16 utterly diverse types of plants: flowering plants, mosses, green alga percent diatoms, brown algae, red algae, blue-green alga bacteria, towards 7 non-electrolytes. The plants are arranged according to increasing permeability to erythritol. The chemicals are chosen under the viewpoint that a moderate lipoid solubility and a fairly moderate MV make it possible to migrate along either the lipoid or the porous path, according to the prevalence of one or the other disposition. A graphic presentation of his results looks embarrassing enough, but a certain common trend of the curves cannot be denied. Erythritol, for instance, without exception, is the slowest. Propionamide, without exception, the fastest of the compounds. Between their lines, the curves are often overlapping, due to the highly varying distances between the corresponding points of the curves, with the result that, instead of the order I to 7 of the compounds, as it obtains with the majority of the objects, the cells of row 9, for instance, follow the order. 2 IV, the sub, is it sub? However, this great irregularity is obviously the effect of the third of the aforementioned factors of permeability, since, according to P. 235, the penetration rates of the compounds I, RV, and vice, owing to their NH2 groups, can be foreseen to be slow, for example, in relation to Spirogyra 7 or Viaceaura 11, figure 24. One may conclude from this analysis, as well as from other points, that an understanding of species specificity, which often is believed to rely upon morphological or biochemical features, can also be approached by physicochemical study of membrane permeability. This will become even more conspicuous with erythrocytes, where species-specific permeability is manifested to an amazing degree. To stat, permeability of erythrocytes to non-electrolytes. No other cells have been used more as the object of permeability studies than erythrocytes. They are single cells available in large numbers, and their behavior, though different from species to species, is on the whole constant if taken from the same sample of blood. In contrast to MLSA of the plant cells mentioned in the last section, They are immersed normally in a well-balanced sexy-5 solution. But in an isotonic solution of an indifferent non-electrolyte solution, they undergo severe alterations of their normal permeability, which, however, can be largely reduced by adding small amounts of electrolyte, for example. 0.1% NaCl, Cl Sec 5, Chap 16. The first systematic information about permeability of erythrocytes was contributed almost contemporaneously with the fundamental work of Overton by Grimes and Hedden. Grimes has observed that the blood cells of chicken or of horse undergo hemolysis if added to an isotonic solution of 1. GCAF cells of Plagiothisura dendliculature. Idogororus P. S. root cells of Lanina minor, 4. Pilaella litoralis. 5. Zignana. Yaio osporoine, subai dernial cells of Curcuma rubricolis. 7. Spiragira sp. 8. Leaf cells of Clodia densa. 9. Epidermis cells of Roeo discolor. 10. Epidermis illas of Taraxacum pectinatiform. Leaf cells of Hara ceratophylla. 12. Intranodial cells of Ceramium diapenum. 18. Bacterium periole. 14. Oscillatoria princeps. 15. Melicera sp. Pb. Limophora sp. Aliphatic monohydric alcohols, ether, esters, glycerol, or urea, but remain intact in saccharose, lactose, mannitol, inositol, dextrose, and asparagine. Hedden determined the freezing point depression after equimolar amounts of the organic compounds had been added to a certain volume of blood, and to the same volume of the serum, according to whether the freezing points of the fluids are different or not. And whether they become more different with time, one is enabled to distinguish between impermeability and permeability, and, eventually, to determine even rates of penetration. Hedden with ox cells obtained the following series of increasing penetration. Saccharose, hexoses, 2 hexetes, pentates, jantixi, erythritol, glycerol. Ethylene, glycolol, Gruyere, aldehydes, ketones, esters. The results of Grimes and Hedden obviously are similar to those of Overton regarding plant cells, P230. They show that lipoid-soluble substances, for instance amyl and benzyl alcohol, ethyl-urethane, ethylacetic ester, independent of their molecular size, can readily enter the cells. Hedden, referring to the lipoid theory of Overton, already paid attention to the fact that urea and glycol, though lipoid insoluble, have a high penetration rate. After these early investigations, many technical improvements toward more quantitative results have been developed, all gauging permeability by osmotic swelling and its final stage, hemolysis, either by direct measure of the cell volume with the hematocrit or by the increasing transparency of the cell souces ention to light, at best applying a high hydrocell or by taking the distribution of light at the cell surface as an indicator. These methods have been used particularly to compare the species-specific permeability of erythrocytes in a singular way to that plant cells, CP237, comparing the penetration rates of substances characterized by a moderate lipoid solubility and a moderate molecular size. Thus, according to Höber and Zoroskov, with blood corpuscles of rat, man, pig, and ox, the permeability appeared to decrease in the order acetamide ulpropianamide, lactamide, malonide, also to decrease in the order urea methylurea diurea, and even irrespective of the increase of lipoid solubility. To decrease in the order methyl alcohol, ethyl alcohol, propyl alcohol, butyl alcohol. In other words, the influence of porosity outweighs the influence of the dissolving capacity of the lead. It appears that two groups of mammals can be sharply distinguished. They differ in other respects also. The red cells with a high permeability to glycerol are distinguished by a low temperature coefficient of penetration, and their permeability is diminished by CO2 and other acids and by traces of Cu. A similar correlation has been found with glycol. A hindering effect resembling that of Cu has been found with EII, ZenCo, and Hg, and is possibly based upon an acidification of the cell surface due to the formation of undissociated complex salts of the heavy metals. A series of experiments concerning erythrocytes has been mentioned before with reference to the permeability of plant cells to organic substances containing in their molecule 1 or 2 and H2, groups P235. Jacobs had found that the renal cells of mammals and reptiles are more permeable to urea than to glycol, while those of fishes and birds show the reverse behavior. Höber and Zyrskov, including in their study some amides and glycerol, arrived at approximately the same result. More experiments are needed, however, to secure this correlation between certain chemical properties of non-electrolytes and their penetration power. It seems not unlikely as yet that the hindering effect of acidification on the permeability of the erythrocytes of mammals is a counterpart of the opposite effect of the NH2 groups. Especially fascinating problems are presented in this field by observations dealing with compounds, which, in spite of their lipoid insolubility and their large MV, enter the erythrocytes of certain species and thus fit into neither of the main theories of permeability. These problems have already been touched upon in findings of Jacobs, that erythritol, which, due to its somewhat smaller MV, is enabled to enter very many cells, passes into mammalian erythrocytes with exceedingly diverse speeds, fastest in less than 5 minutes, into those of mouse slowest in more than 24 hours, into those of ox and sheep. Similar studies sprang originally from an interest in the blood sugar in human physiology and pathology, and were stimulated by the fact that glucose can enter the erythrocytes of man, but of no other mammal except monkey. Besides glucose, the other hexoses and pentoses likewise enter the human cells, but neither glucoheptose nor methylglucoside. Further, not disaccharides, hexites, pentites. More recent studies have amplified the knowledge concerning the human cells for dis and pentoses enter somewhat faster than hexoses. Second, the aldohexoses faster than the ketohexoses fructose. Both facts are interesting in contrast with the earlier observations of Wilbrand on the passage of sugars across the intestinal membrane of rats, where pentoses have been found to be slower than hexoses, though having the smaller MF, and where poisoning with iodoacetate abolishes the characteristic succession of the hexoses, while here it is indifferent, see Sec 8, Chap 34 to 5. In extending these studies toward the erythrocytes of other mammals, more species-specific variations have been encountered. According to Höber and Ulrich, mouse erythrocytes not only fail to admit the hexoses, but allow the hexates mannitol, sorbitol, dulcitol to enter freely. Rat cells do not display this characteristic feature. The red cells of the guinea pig show another differential permeability. They admit amino acids glycine, alanine, but exclude hexoses and hexites, Höber and Ulrich. Still another pattern appears with dog cells. Pentoses are here the fastest. The ketohexoses, fructose, and sorbose enter more slowly, and the aldohexoses are practically non-permanent. Wilbrand. Finally, the cells of the ox, pig, rabbit, and rat show the same impermeability as may be expected with lipoid-insoluble substances of a moderate NVI such as sugars, hexites, and amino acids Kazawa. Certainly, exploration in more species with more substances under more varied conditions with an additional study of other cells will disclose more specificities. One would like to know whether such peculiarities of permeability correspond to special functional requirements of an animal or its individual cells. For example, the behavior of certain erythrocytes towards sugars and amino acids is perhaps associated with some role they play as regulators of the normal unbalance between intracellular and extracellular fluids. In the present state of our knowledge, it is impossible to distinguish a true specialization from an incidental feature, a mere lusus naturae. See further, Sec 34 and 35. Permeability of some other objects to non-electrolytes. There is so far very scanty knowledge about the permeability to non-electrolytes of single cells. Other than erythrocytes and plant cells studied by plasmolysis or osmotic swelling and shrink, the only equivalent CIS thematic investigation is that of Overton concerning muscle tissue, a study more qualitative than quantitative. Marine eggs such as those of the sea urchin are single cells which appear appropriate for study by osmotic swelling and shrinking, but scarcely more is known than that the permeability rises in the series aceamide, apropiamide, butyrylamide, and glycerol ethylene glycol. That methyl and ethyl alcohol enter very rapidly, and that glucose does not delp. These successions are like those found with Cara, C. Collender, and Berlund-Locke. In other words, the succession of the amida parallels their lipoid solubility, while the order of the lipoid insoluble glycerol and ethylene glycol suggests their passage through the porous areas of the membrane. P235. With a membrane of a more outstandingly porous type, the succession of the amides probably would run in the opposite direction. CP237. Further, the observations of Höber and Zoroskov, P239. Overton's J procedure for studying the permeability of muscle is analogous to his plasmolytic studies of plant cells, P230. Frog sartorius muscles placed in isotonic Ringer's solution keep their weight constant over a long period. When a non-electrolyte is added in a certain molar concentration to the Ringer's solution, impermeability is indicated by a permanent loss of weight. Addition of a substance with a relatively small permeating capacity is followed by a loss of weight, which disappears after some time, differing according to the rate of penetration. With a quickly entering substance, the results of these experiments are about the same as those outlined in the table. P. 230 for CFERA, for instance, urea, which is distinguished mention by Overton as entering especially slowly. In analyzing the data concerning the permeability of the muscle tissue, one may refer the effects to the muscle fibers themselves, which occupy between Fi and of the total volume in the frog sartorius muscle, depending upon whether they have been soaked in Ringer's solution or in plasma. In other words, the interfibral space is J5 to this space readily communicates with the surrounding fluid by free diffusion. And calculation shows that if, for instance, the Cl percentage in this fluid is very, the Cl content of the muscle changes in such a way as to indicate a distribution equilibrium whereby CLL is located practically to completion outside the fibers. That is, the chloride space is equal to the interfibral or tissue space. This justifies the assumption that the aforementioned results concerning permeability or impermeability of the non-electrolytes are bearing upon the see more about the interfibral space later, P253-1, plant cells. Since Overton's fundamental study, plant cells have often been considered impermeable to inorganic neutral salts, or, at most, only slightly permeable. This conclusion was drawn from observations on cells, which from water were transferred into NaLLCLs of such a strength that plasmolysis apparel, which seemed to be persistent for some time. Later, with a more refined procedure, it has been shown that the plasmolysis, sooner or later, is succeeded by deplasmolysis, which, after a while, may be followed by another plasmolysis, often leading to DAT. This sequence of events has been interpreted as due to an impairment of the cell surface by the solutions, which brings about an abnormal permeability to the salt indicated by deplasmolysis. The consequent SKEA of solutes from the cell interior ends in collapse of the protoplast. The concept that in its normal state the membrane is impermeable is supported by the following observation. Brooks, by measuring the conductivity of the surrounding fluid, has stated that taraxacum cells placed in distilled water suffer a slight loss of electrolytes, but that this loss is markedly increased in dilute natal solution. Second, cells have been shown extremely impermeable to salts or their ions presented in small concentrations, provided care is taken to keep them under physiological conditions in other respects. For instance, several ions, the and others added to seawater as the normal milieu of the algae do not appear in the cell sap even after hours or days. Very little CS was found even after more than a year. RB, somewhat more a lantern analyzed by spectrography samples of the cell sap of two species of carisui after salts were added in small plasmolysing concentrations to the brackish water in which they live. The result was that, even after 8 to 14 lays, Lee salt did not reach a concentration within the sap equal to that in the surrounding medium. Light accelerated and anoxia retarded the move. Returned to the normal lye-free medium, the cells did not lose the lye previously taken up. Third, the strongest argument for impermeability, or at least dispersibility, sap of hal-isophis mol-1C1 water. Sap of Cara ceratophyllate alternative, a dynamic equilibrium, is more probable. This would explain the aforementioned observation of Colander P243 and similar results obtained with Nutella dye presumably. Some active factor is superimposed on a passive distribution, permeability being not so much a matter of simple diffusion as of active transfer, which needs an energy supply. This will be discussed in detail later, see Sec 8, also Sec 5. Fourth, returning to the problem of plasmolysis and deplasmolysis of plant cells under the influence of neutral salt solutions, and to its interpretive shown as the consequence of an impairment of the normal surface condition 5. It can be shown that a pure Nye salt or K salt in contact with a cell creates an abnormal condition of the surface, and that the addition of small amounts of certain other salts restores normalcy. For instance, Nutella cells can be shown by the diphenylamine test to take up nose from a non-OA solution. But nose does not appear in the presence of Nate dal LPA in the balancing effect of Chi in concentration small compared to N or K has often been demonstrated by the intensified plasmolysis obtained with the mixture, and degrees with different cells. It is more evident with Ilhoa or lupinosis than with lamium when permeability is measured by rate of plasmolysis. Alkali ions are found to increase permeability according to the series. An icing effect of Ca is similar to that of Esser and Ba. Mg resembles more closely the alkali ions. The result of these and other studies is the concept that applying solutions of appropriate mixtures of salts, or more precisely of alkali and alkaline earth salts, for investigating the permeability of plant cells instead of the unnatural solutions of single salts, the cells appear to be impermeable or fairly impermeable. It will be shown later that this is equally true with numerous animal cells. Since this antagonism in plant, as well as in animal, cells is an antagonism between mono and polyvalent cations, which was first interpreted by J. Loeb to be an influence upon the colloidal constituents of the cells. See Sec 5, Chap. 16. The conclusion seems indicated that a normal molecular architecture of the cell surface is a prerequisite for normal impermeability or dispermeability, see Chap. This concept does not conflict with the aforementioned postulate that energy, the properties of the cell surface, can be changed toward a greater to erythrocytes, a permeability to anions, on the background provided by the studies of Hamburger, 1891, Gerber, 1895, and Koch, 1897. It has been generally accepted that the erythrocytes differ from other H. Caho universe, Dorpat, Bot, Opera, Emmer, 1924, Osterhout, Trondle, Caho, Locke, EA. About contradictory results, CP through 2024. For an interesting study of the permeability of Vegeatoa to organic anions and the physicochemical viewpoints concerned, C. W. Ruland, H. Ulrich, and G. Yamada planta cells and their selleclive permeability to inorganic anions and their impermeability to cations. The well-known physiological significance of these facts is their influence upon the acid-base equilibrium in blood, evinced by the alternating exchange of Chi-LL fluoride shift, see also Sec. 8 Chap 37. In view of this situation, an isotonic solution of an inorganic salt may appear to be osmotically indifferent to the red cells suspended in it, as was found 50 years ago by Grinds and ammonium salts display an exceptional behavior. In their solutions, the RCAD cells hemolyze with chloride and bromide faster than with nitrate, sulfate, and phosphate. This behavior occurs solely with erythrocytes and is affected not by permeability to the chocation, NH4, but by anion permeability. Slightly hydrolyzed and due to its small f, the free 23 enters, whereas nothing happens after that with common cells, except a minute increase of alkalinity with erythrocytes, their full lows in exchange of internal O and external continues until hemolysis appears. A picture of this progress is given by figure 25. It was mentioned before that the isotonic solutions of inorganic salts are osmotically indifferent to the red cells. This is true only when monovalent anions are concerned in the exchange. In an isosmotic solution of polyvalent anions, blood corpuscles shrink. The explanation is that in order to maintain electroneutrality, a SO4 ion, for instance, exacts 2 Cl or 2 HCO3 ions in exchange. At the same time, the osmotic resistance rises. This presumably is due to several circumstances, among others to the dehydrating effect of the polyvalent anions, sulfate, phosphate, tartrate, citrate, and others, which results in a closer packing of the structural components of the surface membrane. This effect, coupled with the greater hydration of these ions, see Sec 5, Chapter 16, accounts for the polyvalent anions penetrating the membrane at a distinctly lower rate than the monovalent do, comparing, for instance, the exchange of Cl against HCO3 and of Cl against SO4, as has been shown by chemical analysis, or by applying an AgL electrode in order to follow the exit of Cl-G25. Jacobs. Our understanding of erythrocyte permeability to organic anions is so far highly unsatisfactory, or the reason that a great number of factors must be taken into account in order to explain the effects, which are chiefly osmotic changes of the cell volume and hemolysis. For the effects depend upon whether the organic acids forming the salts are strong or weak, whether they are lipoid-soluble and surface-active, the ions are strongly hydrated and have hydrotropic or solubilization properties, Chapter 20, and whether they are poisonous, acting either on the surface or on the entire protoplast, after having entered freely. It will be helpful to begin with comparing the Na and the NH4 salts, the Na salts of the aliphatic fatty coids with a chain of C atoms not longer than C5 are fairly indifferent, provided the PL of the solutions has been buffered to about 7 point. Beginning with valerate, CH or capronate, CE, and proceeding to the higher members of the series, hemolysis occurs, decreasing the pH favors the lytic influence. Increasing the PAI weakens it. JAG for an explanation of these findings. Several factors must be taken into consideration. The fatty acids are weak acids, all those from Co to C9, liavi pK of about 4.8. The lower the PL, the higher is the concentration of the free acid in the solution. Their undissociated molecules are lipoid-soluble, see Chapter 12. So far, the break at CS in the graph of their effectiveness seems unexpected, but the same break appears if one compares the surface activity. Either water, air, or double water hydrocarbon of the aqueous solutions of the NA salts from C2 to C5, it rises slowly, then from CE abruptly. The lower members of this rise can be classified fairly near to CLL in physicochemical, Chapter 15, and in physiological respects, Chapter 34. Whereas the hemolytic effect of the higher members must be attributed not only to an exchange of anions, but also to hydrotropic effect, probably exhibited at the exterior as well as at the interior, turning now to the NH4 salts of the fatty acids. Not much remains to be added. The main point is that but increasing from C2 to CS diom, this increase corresponds closely with the increase of lipoid solubility. Hence, these compounds can be supposed to pass through the erythrocyte membrane by the lipoid route as well as the pore route. The Na and NHADH salts of aliphatic di and tricarboxylic arid monocarboxylic acids. The Na salts of the C4 compounds, succinic, tartaric, fumaric, and citric acid, have been known for a long time to be fairly indifferent to various cells and tissues. See the potential measurements. The same is true with the NH4 salts too, which rather resemble NH4-2S4, CP240. This can be ascribed to several circumstances. We are dealing with rather strong acids, which as such are lipoid insoluble, but also because they are strongly hydrated due to the polar carboxylic and eventually also hydroxyl groups. Hence, they diffuse slowly and traverse pores with difficulty. In addition, they are strongly hydrophilic toward colloids. In other words, they compete with these colloids for hydration water. They are surface inactive and lack hydrotropic properties, chap 20. Among the NH4 salts, the rate of hemolysis is greater in solutions of the K-series, lactic, and pyruvic acid. That is, monoxymonocarboxylic acids than in solutions of C4. It is still more increased with the corresponding Ca compound, ammonium glycolate, which approximately equals ammonium acetate. This seems contradictory to the response of muscles, which display an injury current with antibenzoate and salicylate, Höber, Locke, Sit. However, it is questionable whether an alteration indicated by the electric behavior of the surface membrane of muscle is equivalent to an alteration which releases hemoglobin. The latter is believed to be a rupture of the membrane, whilst the former can be brought about even by a slight diminution of the lateral adhesion forces in the molecular array of the surface structure. The hemolytic action of NH4 salicylate is comparable to that of NH4-C, in contrast to the salts of the aliphatic monocarboxylic acids P247. NH4 salicylate is the salt of a rather strong acid, 7 DKK equals 3, and as such not subject to much hydrolysis. On the other hand, the free salicylic acid is highly lipoid soluble. Thus, the rate of hemolysis appears to be the effect of rival factors. Furthermore, denaturation of proteins, including hemoglobin, can be produced by salicylate as well as by the salts of other aromatic acids, probably due to their polar-nonpolar molecular configuration, subchap 16.2 and chap 20, and is followed by the escape of solutes from the interior of the cells. This seems to increase the osmotic resistance and so partly offsets the lytic tendency. Obviously, further study is needed to settle the relative importance of these conflicting influences. Behavior in solutions of non-ionating non-electrolytes. It has been known for a long time that in non-electrolyte solutions, saccharose, a leakage of electrolytes, as indicated by chemical analysis and by a rise of osmotic resistance. Jacobs and Parpart have found that this process occurs in two stages. One, which is reversible and rapid and finished after about 1 minute, and one which is irreversible, slow and extending over a long time. The nature of the second process will be discussed later, see subchap 16.3. The first one is due to the steep gradient of anions from the cell interior to the non-electrolyte solution, causing an exchange of Cl and HCO3 ions from inside for O ions from outside, and hence resulting in an increase of acidity outside of alkalinity inside. The latter brings about an increase of ionization of hemoglobin, that is, the formation of polyvalent hemoglobin anions, HBDE, replacing the monovalent HTO7 anions according to the equation. Consequently, the internal osmotic pressure diminishes, as indicated by the increase of osmotic resistance. B. Permeability of erythrocytes to cations in tables of T that accepts T, P244. It has been shown that various species of plant cells preserve in their cell sap characteristic mixtures of ions with steep gradients, some uphill, some down toward the outside solution. This is equally true whether the normal medium of the plants is fresh, brackish, or sea water. This looks like convincing evidence of impermeability to these inorganic ions, unless one takes into consideration that this ionic distribution could also be the result of a dynamic equilibrium, which the living cell maintains with metabolic energy and which gives way only in death to a stable state. Similarly, the species-specific distribution of inorganic ions in erythrocytes, which is apparent from the following table, can be thought of as being essentially the result of activity during an early stage of the life cycle. When the red cells were being generated in the blood-forming organs, however, Although erythrocytes have been supposed to be exceedingly inactive, biochemically as well as structurally, it has been found recently that a slow shift of cations against the concentration gradient compensates a preceding loss, CP 251, as appears in Table 17. K and MA are distributed very characteristically in the red cells of different mammals. First, there are two groups of animals, one with a preponderance of K in the cells, the other with a preponderance of AI party. Second, this selectivity is independent of the cation concentration in the blood serum, as the ionic composition of the serum is practically identical in all mammals, with A far in excess of K. Now, this characteristic unbalance, Ka, is easily upset under many circumstances. Almost any kind of alteration of the normal surrounding medium is followed, referring to a leakage of Kk can be released, for example. By slight hyper or hypotonicity, accompanied by a deformation of the cells, by a small change in the balance of cations in the suspending salt solution, by a rise in temperature, even by mere standing in serum or in Ringer's solution, or by centrifug ENG. Furthermore, the addition of certain cytolytics or NAF leads to an escape of NAO, disregarding for the moment the effect of cytolytics, and a fluoride selective ease permeability is likely to be lost, as indicated by the SKC of K, as the result of relatively slight changes of the physical properties of the surface, milder changes than those that cause visible leakage of hemoglobin. Presumably, the changes consist in loosening of the linkage of colloidal aggregates. The same assumption has already been proposed, p. 245, in interpreting rates of plasmolysis and deplasmolysis as a measure of permeability to agents as gentle as neutral alkali salts are, where the effects of ions increased gradually in the order. The not K, and so for CLL, no 3BR, an order well known from colloid chemistry as the Hofmeisters or lyotropic scries. Numerous similar observations will be referred to later, in muscles particularly studied by measuring, for example. The membrane potentials of the fiber surface, a reversible loss of selective ion permeability, follows exposure to various cations and anions of alkali salts, and has been correlated with the state of colloidal components of the surface membranes. The same agents, viz., isotonic or slightly hypotonic solutions of the alkali salts, when acting upon the red blood corpuscles through hours or days instead of minutes, increase leakiness so much as to enable hemoglobin molecules to escape. In this phenomenon, also, the strength of the cationic and still more of the anionic effect is found to vary according to the lyotropic series, though numerous irregularities have been observed. Depending upon species-specific properties of the erythrocytes and upon additional factors connected with the surrounding solution, Chap 16, to see further the studies of Davson regarding cat and dog erythrocytes, loci citi. Among the various conditions abolishing the normal impermeability to cations, fluoride deserves special discussion, because it suggests an influence for many details concerning these alterations of the normal ionic distributions. With respect to hyper and hypotonic solutions, E. Ponder and Saslov, J. Physial, 70, 169, 1930, 73, 267, 1931, E. Ponder and Robinson, Biochan, J. 28, 1940, 1934, M. H. Jacobs, H. N. Classman, and A. K. Parpar J. Cell and Confusiol 8, 403, 1936, Better 2. Davson, E. Hid Dial, 10, 247, 1937, 1940, Biochem J. 8338, 1939. Regarding centrifugation in serum, Ringers and SCF. Danielelli and H. Davson, IIDD 32, 981, 1938. Regarding lytic agents, F. Danielelli and H. Davson, Ibid 32 to 991, 1938. Regarding photodynamic dyestuffs, H. Davson and E. Ponder, J. Cell, and Confusiol, 1667, 1940. Regarding fluoride, W. Wilbrant, Faraday Sock 33, 956, 1937, Pfluger's Archer F DGS Physical 243, 519, 1940. Regarding temperature, M.H. Jacobs, H.N. Classman, and A.K. Parpart, J. Cell, and Comp Physical 8, 403, 1936, regarding average cat and dog corpuscles, 2 Davson, Ibid, 16, 317, 1940. J. Physical 101, based upon breaking the normal concatenation of reactions participating in glycolysis. According to Wilbrant, after addition of 0.25 molten NAF to a suspension of human erythrocytes in NaCl solution, an enormous increase of osmotic resistance occurs. This is not due to precipitation of CI, since iodoacetate, another agent which interrupts glycolysis, also raises the osmotic resistance by releasing K. The poisoning effect in both cases must be inferred to the breakup of the glycolytic sequence, as the poisoning effect runs parallel to the glycolytic activity of red cells of different species, and is nil with non-leucolyzing pig cells, though they are rich in K. See Table 17. Another argument for normal glycolysis being indispensable to the normal impermeability to K is the fact that the increase of osmotic resistance after addition of NAF is abolished by a proper amount of pyruvic acid, see Sec 6. This raises the question as to the the causality of the erythrocyte membrane to retain cations. On the one hand, normal glycolysis may be effective by warranting a structural situation which retains the cations. On the other hand, glycolysis may maintain ionic equilibrium by releasing energy for the back transport of K, which is on the way to escape from the interior of the cells. Examination of this alternative so far has led to contradictory findings. According to Wilbrant, no change of the normal cation distribution occurs if the temperature is markedly lowered. Harris, however, also working with human erythrocytes, has found that when they were stored at refrigerator temperatures 2 degrees to 5 degrees under sterile conditions for 5 days, K-ion leaked out into the plasma along the diffusion gradient and re-entered when the cells were returned to temperatures of 25 and 87. A deceleration of K was more complete after glucose had been added. These latter facts recall the experiments of Hoagland and Davis with Nutella cells, which at a low temperature or in the dark become leaky to the inside. 8. Recapture it at room temperature or their exposure to light. See Sec. 8. Chap 88. 3 other animal cells. The selective anion permeability of blood corpuscles is unique. Its existence has been proved directly by chemical means and indirectly by observation of osmosis, and can be accepted as indicative of a purely physical-chemical property of the cell surface. In addition, there are signs of cation permeability, which is probably due to an active transfer. Now, before turning to studies concerning other animal cells, the situation in plant cells may be recalled. The conclusion was drawn, p. 245, in discussing them, that the quiescent cells are generally impermeable to anions and to cations, but that the quiescent state, according to manifold convincing evidence, can give way to a state of activity varying in degree from ample permeability to both cations and anions to almost perfect impermeability. This will be shown later, especially in Sec 8, been shown by the observations of Ostrauch-Collender, and others already in Otlier Way is known, and will be discussed lat T to dinoserication and perhaps anion permeability in plant cells, the measurement of electrical potentials across the cell surface. This permeability cannot be proved to exist by chemical analysis because of the high electrical resistance of the membrane, which prevents a sufficient number of ions being transported to ion, for instance, with cells of Gelaliscystis, where the protoplasmic wall is the seat of an electromotive force of 70 to 80 MV, which is maintained for weeks, and which has been observed to drive a current of 1 to 5 microamperes for several days through a capillary electrode, fixed inside the sap and connected through the external circuit with the second electrode attached to the outside of the eggs of murine invertebrates or animal culls, which are single like erythrocytes and readily available for experimental purposes, but so far have scarcely been utilized for studying permeability to the common ions. As was mentioned before, Pete, 246 other cells, in contrast to erythrocytes, do not cytilize in NH4CIA unless appreciable amounts of bicarbonate are present. Thus, the volume of sea urchin eggs remains unchanged for a long time in isotonic NH4CSE, or NH403, as well as in the corresponding Na and K salts, but they swell and die in solutions of the ammonium salts of the lower fatty acids. This is due to the strong hydrolysis of these salts compared to that of salts of strong acids. This has been proved by the observation that increasing the acidity, for example, from 7.8 to 6.2 increases the rate of swelling. At this PW, the rates rise in the order acetate propionate, butyrate, valerate. This is the order of increasing lipoid solubility and is an example of the rather general phenomenon that the weak acids enter cells and are disposed to do so, because the undissociated molecules of organic acids and bases are often lipoid soluble, sec pages 259f, though to different degrees. Thus, at pH 7.8, swelling of the sea urchin egg is almost imperceptible in a solution of ammonium acetate, whereas in ammonium valerate the cells are cytolylized within a few minutes. The reason is the low lipoid solubility of acetic acid, BB's single muscle and nerve fibers. These objects can be compared with single cells since their fibers are held together by rather loose connective tissue, the interspaces as a rule being open to the surrounding fluids, CP242. Information about the distribution of the most common inorganic ions, K, NaH, GLL, and oil, is desirable. Studies on plant cells immersed in water or aqueous solutions, such as seawater, led to the concept that a practically static equilibrium between cell and surrounding is the result of impermeability to the ions, see especially Table 15p 244, though in addition, with certain cells and under certain conditions, the ionic distribution must be conceived as due to a dynamic state. Muficles in order to reach a corresponding statement for muscle. The entire muscle, ordinarily the frog sartorius, is soaked in isotonic solutions, and chemical analyses are made of the ionic contents of the muscle substance, the interfibral fluid being assumed to communicate freely with the outside solution. Urano and FAR have soaked muscles at a low temperature for several hours in isotonic saccharose, with the result that K, which is the predominant cation, here as in so many other cells, escapes only to about 6% into the surroundings, whereas ELI contained in muscle at a considerably smaller molar concentration appears during the same period of time up to 90%. The escape of Na is similar to that of TL, although somewhat slower and more irregular. By these facts have been interpreted as indicating that K is retained inside the fiber, while Cl is extrafibral. This conclusion is confirmed by the fact that K escapes as completely as Cl does after injury or death of the muscle. Accepting this idea that Cl is entirely extrafibral, one can calculate, by comparing the Cl content of the total muscle with that of the surrounding fluid, the volume of the so-called chloride space, that is the interfibral or extracellular space. It has been found to be about 14.5% of the total volume. This is in satisfactory agreement with the histological tissue space, as determined microscopically in frozen sections of frog sartorius muscles, regarding the K found in muscle as in diffusion equilibrium with plasma. The concentration of intrafibral K proves to be from 30 to 40 times higher than the extrafibral. This K must be assumed to be largely free to account for osmotic equilibrium, see Sec 7. However, this ratio, 30 or 41, is not constant, but varies in both directions in response to experimental changes of pH or of the outside concentration of K. This has been shown by the following observations: Mond, Amson, and Netter, by perfusing frog muscle with ringers, Fenn and Cobb, by placing the isolated muscles in ringers containing various amounts of K, have found that, at a certain maintenance concentration, K neither enters nor heaves the muscle. but that this equilibrium can be shifted by changing the outside DDO. For instance, at 7.2, the equilibrium concentration was found to be 19 rng% at PCl 6.323 milligram percent and at PHI 6 46 milligrams percent. This means that increasing the 2-ion concentration raises the tendency of K to escape and must be compensated by a higher K concentration outside. In other words, the equilibrium state is defined by OH equals CoA K or I, therefore, at a certain ratio OH, if K is added to the outside solution, must rise. Since normally Ka is considerably greater than XO, K will be shifted against the concentration gradient. See further PA9. This situation has been illustrated by an interesting Nipswell experiment of Netter-Dictois, has shown Ly measuring the M Umbrae potentials, which are displayed by a dried collodion membrane intermediate between different electrolyte concentrations, that this decombrane is permeable to H and K, but not to NOR, to CLL and other anions. CPA 17. In this, it resembles the surface membrane of muscle fibers, which were considered selective ion permeability. Among other experiments, Netter separated a solution A consisting of mol 14, 0K2, so 4 plus inol, low il2 for 2, from a solution 6 consisting of mol 1400 X-dose-4, which was rendered isotonic by addition of a proper amount of glucose in order to avoid a shift of water. Obviously, H in this cis-CEMI tends to pass the membrane, following this DCP concentration gradient 10-10. But for electrostatic RC such a passage is not possible except by exchange for an equivalent amount of K. Notwithstanding the fact that the concentration of K is initially the same on both sides, such an exchange does proceed, and K is transported against an increasing concentration gradient. Netter has interpreted this half-eating, among others, by the following consideration. Hot and can meet in the pores of the sieve-like membrane, moving in opposite directions, and the chance of their meeting at the same moment is proportionate to IOX. The same is true to the correspondications HEHA, their chance of meeting being proportionate to 1/6K, the exchange in T. E first direction is thus cancelled partially by that in the second, and goes on until equilibrium is attained when the experimental results are consistent with this theory. The following numerical example illustrates one of the experiments. Because of the thickness and very low permeability of the membrane, the equilibrium state was not reached even after 13 days, but it is plain that under the conditions of this XJ experiment, while the concentration gradient of 11 decreased by diffusion, a strong concentration gradient of K was established. This model elucidates how the high accumulation of K inside muscle fibers may arise, and how after reaching an equilibrium, each upset of the latter must be succeeded by a redistribution. Another conceita about ion permeability of muscle has been suggested radical interpretations. Usually start from the premise that the muscle membrane is permeable for Cl as well as for K, but not for Na, and that the muscle fiber normally contains large amounts of K and small amounts of Na and Cl, all of which are osmotically active under such conditions after immersion into an isotonic natal solution. CLA could not enter for electrostatic reasons. Also, Cl hardly could be exchanged for equivalent amounts of organic anions from within the fiber, for example, the large anions of the various phosphorylation products, or colloid ampholates. On the other hand, after adding small amounts of KFL to the natal solution, the entrance of Cl together with K is hampered by the steep gradient of K from inside to outside. Only after the K concentration outside had been raised in the experiments of GFI Voyle and Conway to a milliequivalent to 0.09% Kertia. A distinct entrance of K and Cl in nearly equivalent amounts was observed and was continued up to 300 milliequiv del, corresponding to has been found by Hegnauer Fenn and Cobb HED to create unphysiological conditions for muscle. The excitability is strongly diminished, the oxygen consumption is raised, phosphocreatine is split, and contraction begins. Also, swelling of the muscle is often provoked by KTL doubt. Therefore, possibly permeability to anions is caused in these experiments only by alterations of the normal permeability instead of pre-existing. No mention is made the processes they studied. The next problem is to investigate whether the behavior of KNA and CLL is specific or may be correlated with that of other inorganic salts. This problem has been attacked mainly by perfusing frog muscles with various alkali ions and by measuring muscle potentials. Mond and Amson degrees have observed by chemical analysis that not only K above a certain concentration level passes over from the capillaries into the muscle, but also CS and prob, ably RB, whereas anali and do not. This is in agreement with the potentiometric measurements, pages, and can be interpreted to signify permeability, probably dependent upon an exchange of these cations with hydrogen ion, P254. In other words, this entrance should not be accompanied by an osmotic effect. As a matter of fact, muscles in isotonic solutions of indifferent inorganic salts, as well as of indifferent lipoid insoluble non-electrolytes, behave like an osmotic. Raising the tonicity diminishes, decreasing the tonicity enhances the volume or the weight, as known already from Overton studies. Even in isotonic solutions, after a longer period of time, however, minor changes of volume may appear, which are dependent upon both cation and anion, and are probably due primarily to swelling of hydrophilic colloids, rather than to osmotic swelling. This influence is especially evident with K salts. It has been described by Overton, but needs further investigation, that in isotonic solutions of K-sulfate, tartrate, phosphate, and acetate, muscle shows a perfectly normal appearance over several days. Though it is entirely inexcitable, depolarizing effect of the K ions, it readily returns to a normal physiological behavior after being transferred to NAN salts. Swelling and death, however, occur in the corresponding solutions of K chloride, bromide, iodide, and nitrate. Possibly the stronger hydration by the second group of anions alters the colloidal compounds of the surface structures so radically as to lead, after some time, to disorganization and disintegration, see Sec 5. Further information about the permeability of muscles to ions is obtained in intact muscle, which are in contact with two different electrolyte solutions. The results of such measurements will be presented later, pages 313 from. They allow the conclusion and fairly satisfactory agreement with the results of the perfusion experiments that the surface membrane is permeable to KRB, less so to CS, not to Na and Li, and scarcely or not at all to anions. The order of the anionic and of the cationic influences again suggests alterations of the colloidal structure of the surface. So far, the passage of ions in one or the other direction has been described as a passive process. Even the migration of K against the concentration gradient into muscle, as in Netter's model, seems to depend upon the distinctly smaller penetration power of one ionic component, as in a Donan equilibrium, and does not demand energy from a metabolic process, see also Introduction to Key. 8, however, as in plant cells and in erythrocytes, an active factor, also, must be dealt with in muscle. HepMap has raised rats on a diet deficient in K, with the result that nearly half of the K in the muscle was replaced by Na. There was more intrafibril than extrafibril. After restoration of K to the diet, the rats rebuilt the normal content of K in their muscles up to a gradient of 31. Steinbach degrees has observed similar supplementary exchanges of K and Na in the isolated frog muscle after soaking it in K-free CK ringers. Fenn and Cobb have found TFI indirect stimulation causes rat muscle to lose about 15% of K, which is replaced by Na, and that during recovery, K is regained and N lost. That is, during recovery, K returns to the fiber, not to the plasma, both against the gradient. Facts like these suggest that work is required, but the situation is very much unsettled as yet, especially from lack of metabolic study of the problem. Nerfiptat. J and nerve, as in muscle, the essential units, the fibers, are held together by connective tissue including fluid-filled spaces, which are more or less open to the surrounding fluid and allow free exchange of their solutes with those in the latter. It has been shown previously that only indirect information about the ionic contents of the muscle fibers can be gained by soaking the muscles in various isotonic solutions and calculating the contents of the fibers from analytical data, referring to the entire muscle, P253, and more reliable data concerning the fibers of the nerves have been afforded during recent years by utilizing the giant nerve fibers of the squid loligo, which can be isolated from the smaller sized fibers in the stellate nerves of these marine cephalopods. Just as the characteristic distribution of ions between the cell sap and the aqueous surroundings of giant plant cells, Valonia, Halicystus cham, and others, has been elucidated by direct chemical determination, P244, so analyses of the fluid contents or axoplasm extruded from the axis cylinder of the giant fibers and uncontaminated by such extraneous matter as fiber sheets, connective tissue, or interfibral fluid have contributed data which are particularly valuable for a better insight into the electrical processes as the most significant signs of nerve activity. The composition of the squid axoplasm in regard to inorganic ions resembles in a general way that of muscle, CP253. The most important difference between inside and outside solutions is in their content of K and TL. Ki is about 18 times higher than Ko, as found in the blood fluid, results with muscle, C, however, P255, an appreciable, though low, concentration of Cl inside the nerve fiber, so that Cl here cannot be considered entirely extracellular. Chemical analyses, as well as conductivity measurements, show that there is a remarkable excess of inorganic cations and that the deficit of inorganic anions probably indicates the presence of organic anions of a rather small ionic weight, about 130, and corresponding ionic mobility with chemical properties which suggest amino acids. Earlier observations concerning the properties of commoner specimens of nerves, medullated nerves of frog, unmedullated of lobster and crab, are in general agreement with those regarding the squid nerve following the procedure of uranoenphar. P253, Fenn and coworkers have found that from frog nerve soaked in isotonic sugar solution, CLL and most of the Na readily escape, while K is relatively indiffusible. The amount of LCL present in the nerve varies in proportion to the outside concentration and can be readily exchanged for other anions. Assuming, on this basis, as in the earlier discussions of ionic distribution in muscle, that K is mainly intrafibral, Cl and Na extrafibral. The extrafibral space in frog nerve light has been calculated to be about 64%. The data indicate that K many times higher than KQ. It has been stated that here, as in muscle P254, at a certain limiting KO concentration, K neither enters nor escapes from the nerve. That above this threshold value, K enters and does so against the gradient, and that this behavior is probably due to an exchange of K and H. Nerve differs strikingly from muscle in that changing the outside DDL-AI is not to be followed by a K shift in either direction. For instance, K does eat enter the nerve when the outside H concentration falls. CP254, concerning permeability of the nerve fiber to other inorganic ions than K, very little is known. Frog muscle, as the perfusion experiments of Mond and Amson, P256, have shown, is probably permeable to CS but impermeable to the cations N, Eli, and Ka, and to the anion Cl, Brarierci, and so forth. Conclusions regarding the behavior of nerves can be drawn only from determinations of the resting potentials, which may be interpreted according to the theory of Bernstein, as due to selective or preferential IOA permeability of the surface membrane, see Chap 1765. By this, MTD Tuhad Netter has found that the permeability of frog nerve to cations decreases in the order KWRB2 lolly, whereas at least during a limited period of time, the nerve is not permeable to any anions. For the permeability of nerves during their electric activity, see P321. The group of weak bases and weak acids includes innumerable compounds important to physiology, pharmacology, toxicology, and clinics. The bases are distinguished by the presence of excess ions in their aqueous solution, the acids by excess sucin, but their characters are often more latent than manifest, since these substances with decreasing strength approach the behavior of non-electrolytes, for example, urea, acetamide, phenol, glucose. In the wide range between strong and weak electrolytes, the ratio of ions to molecules present in aqueous solution conforms to the mass law and is represented numerically by the ionization dissociation constant, pK. Among the manifold aspects of the biological activity of these compounds, only the question of permeability will be discussed in this section. Previous sections concerning the permeability of plant and animal cells to ion electrolytes and to ions contribute a groundwork which may be summarized briefly for the coming discussion. Strong bases and strong acids resemble the strong neutral salts in being ionized to completeness, and in largely lacking, at least primarily, the power to pass into cells. Since the pathway formed by the lipoid components of the surface membrane has very little, if any, dissolving power for ions, what little penetration by strong neutral salts does occur depends upon whether the membrane affords an adequate pore, WDDT, or pore affinity for the migration of ions, chap 17, solutions of weak bases and weak acids may penetrate the lipoid layer at a rate correlated with their solubility therein, provided their constitution is suitable, especially provided that their array of polar groups is sufficient to prevent a marked hydrophinity, p. 231 in Sec G5. Chap 20. The early observations of A. Bethe, O. Warburg, and E. N. Harvey upon the umbrella of Medusae, upon the leaves of Elodia, upon paramecium and sea urchin eggs. By applying neutral red as indicator of the intracellular reaction, have shown that strong bases, like now, KO2, and NC2H640H below a certain concentration, paradoxically enter much more slowly than the weaker bases NHS, NH2C, 2HB, and HT2SC3 and others. The strong bases actually penetrate fairly fast also, but this is due to the strongly injurious effect of free O-ion. Corresponding results have been obtained with oxalic, citric, tartaric, and the strong mineral acids on one hand, the weaker acetic, uric, and benzoic acids on the other hand. Again, the penetrating power of undissociated molecules in the solution of a weak electrolyte has been proved in quantitative experiments of Osterhout, with 1x28 Darage ditch between C, the distribution of 2,2S between C, water, and the sap of Valonia cells was investigated at different UTL values. The normal pH of the sap is 5.8, independent of the outside reaction. The pH of the seawater, about 8, was adjusted to a range of values from 5 to 10 by suitable addition of HELL or now. HOL was introduced into the seawater and its distribution at equilibrium observed. That at pH 5 in the seawater, H2S is present almost completely as free acid. At pH 10 as ions H's and S, can be calculated from pKgs further, has been determined directly by vapor pressure measurements. The analyses of H2S in samples of the cell sap, whose pH remained constant at 5.8, gave the result that at equilibrium throughout the pH range from 5 to 10, the inside concentration of molecular H2S was practically equal to the outside concentration. In other words, at pH 10, the inside concentration was near 0. At pH 5 was close to the outside concentration, as shown in the following table that chips out sap wine. Of hydrogen sulfide expressed as percentage of sulfide, it follows that the Valonia protoplast is permeable only to the unionized H2S, impermeable to its ions. In order to determine whether this entrance of molecular H2S is simply a diffusion, cells of Vallonians were again exposed to various external concentrations of undissociated H2S. While the rates of entrance of H2S were followed at intervals of time from 1 to 5 minutes, were proportional to the concentration of the molecular H2S in the external solution. So far, only a few weak acids and weak bases have been mentioned as being obviously enabled to enter living cells by the presence of their undissociated molecules. But physiology and pharmacology have afforded a great amount of evidence that the stimulating or paralyzing or other specific effect of weak aliphatic or aromatic acids or amines, the alkaloids, in particular, can be enhanced by the addition of proper small amounts of stronger acid or base. That is, by shifting the equilibrium toward the undissociated components. This raises the question which physicochemical properties, or whether in all cases the same properties of the molecules, are acting to cause the enhanced effect. The problem has been attacked by Poijärvi in experiments with several kinds of plant cells, which were immersed in mostly equimolar solutions, 0.1 enol, of a great number of N bases of a rather diverse structure. The sap of the experimental cells was stained either by their own pig, not or by adding neutral red. The entrance of the weak bases render alkaline the naturally acid cell sap, and thus their presence Bt conjugac visible by the color change of the indicator-like pigment. This change proceeded with time through a series of intermediary shades of color, each indicating a certain degree of alkalinity, to a final equilibrium notable by a certain definite color, for instance, a yellow color of the cells, initially red stained by neutral red. This procedure was used to measure the rates of permeation and to calculate permeation constants, CP230. Results obtained with cells of LaHoa discolor are pictured in Figure 26, corresponding to the graph for non-electrolytes given on P232. The permeation constants P have been allotted against the partition coefficients ether, water. In addition, the molecular volumes, or rather the molecular refractions, are indicated. Although for several reasons mentioned by Poijärvi, the colorimetric method applied provides no more than rather dubious approximations. The following conclusions may be drawn. 1. Over a wide range of partition coefficients from novocaine cocaine. Sevadenin equals 12 to ethylenediamine equals 0.0085. The permeation rates of these organic bases are correlated with their lipoid solubility, while the strength of the bases, as also noted by Poijärvi in his Table 11, varies over a wide range, independent of the partition coefficients. 2. Again, as with non-electrolytes, the penetration power of bases with low MV, for example, NH3 or NH2, or NH2, GHA is greater that of bases with high MV atropine, Bruce, and Savan, smaller than could be expected from their lipoid solubility. The first fact corresponding similar conclusions previously discussed may be referred to poor permeation. The second, perhaps to the membrane enclosing the protoplast as being inhibitory to diffusion of the large molecules, or to their slow diffusion in the interior of the lipoid layer, CP. 231. In order to explain the entrance and accumulation of K so often observed with plant and animal cells, Osterhout, instead of assuming the exchange of Kappa for AT cross a cation permeable membrane, P. 253 also, Sec 5, Chap 17, has suggested permeation across NOE, like membrane, Sec 5, Chap 19, such as guaiacol by undissociated molecules of the strong base CoH, which would depend upon whether CoX-OHO-excetophyl KX-OLB or not. As a matter of fact, with Vallonia, it has been shown that increasing PLL in the seawater is followed by an increase of the rate of entrance of K, and vice versa. But P-log telon, as Coeffistener experiments with the teleconflict with the hypothesis, in that, for instance, the rate of entrance of K is independent of the external between 6 and 8 di, the higher rate of penetration of K compared to NA in Osterhout's model studies upon organic solvents like guaiacol, is correlated with the greater dissolving power of guaiacol toward CoH than toward NAOE. To end this chapter, a special group of electrolytes will be discussed, distinguished by their staining Jower. which has made them an important tool not only in histology but also in experimental physiology, where they serve to trace the pathway of dissolved substances across cells and tissues. They are suitably classified as basic and as acidic dyestuffs, the first ones bearing color indication, the second in the anion. 1. Basic dyestuffs. Many well-known basic dyestuffs, such as the typical vital stains like neutral red, toluidine blue, brilliant crescel blue, rhodamine, methylviolet, resemble the colorless weak bases, which were mentioned in the last chapter, by the fact that the rate of their entrance into cells depends upon the concentration of the undissociated molecules, either present or liberated by adequate amounts of alkali added to the solution of their salts. That the distribution equilibrium shows equality of the free base inside and outside, independent of the concentration of their free ions. This is evident from the following observations of Irwin.A. A solution of azure B of a certain is divided into two TUO portions, A and 6, by interposing a diaphragm of chloroform, which has come into equilibrium with the dye. If acid is added to portion A, the concentration of free base is decreased, and dyestuff moves from H across tin, chloroform membrane, to A and other wants dyestuff is accumulated. This is analogous to the distribution of H2S in the Valonia experiments of Osterhout, P260. In another experiment, the sap of Nutella cells is stained with brilliant crescel blue. Then the cells are transferred into samples of water with the pH vary in G from 5.4 to 8.2, and the rates of decoloration of the sap are determined. It appears that the rate of exit increases as the external pH is decreased. On the other hand, the basic dye being added outside, the rates of entrance increase when the external Pell leaven is shifted from pH 6.1 to pH 9.3. Other illustrations of this principle of distribution of basic dyestuffs will be mentioned later, Section 8, Chap 37.3, in discussing glandular secretion. One consists in the striking transit of these substances across the gastric mucosa and accumulation in the acid gastric juice, up to concentrations many times higher than in the blood into which they have been injected. In these experiments, accumulation is a mere passive phenomenon. Further, Chambers and Kemptert have compared the passage of two dyestuffs through the tubular epithelium of the frog kidney. 1. The basic dyestuff neutral red in line with other dyestuff bases responded passively to changes brought about inside the tubules. The other, phenol red, a sulfonylane, was actively transferred by the special internal mechanism of the tubular cells, Section 35, which is unaffected by change of permeation by colorless weak bases, was found in the experiments of Pojarvi, P261, to be correlated with their solubility in ether as a lipoid-like solvent. As for dyestuff bases, Nierenstein, doing an extensive study, has compared their staining power for paramecium with their relative solubility in lipoid solvents. He observed, on one hand, that the vital staining parallels distinctly the distribution between water and oil, but on the other hand, that this correlation is intensified by adding some oleic acid to the oil, thus showing that the solvent affinity is to some extent a chemical affinity relying upon the presence of basic radicals in the dye molecule. See pages 234. See also the model experiments of Butyrosek. 5. Another factor has to be considered, acting either at the surface of the cell or after the dye has passed the surface lipoids and has entered the interior. Basic dyestuffs are known to react with cell colloids and especially with the colloid amphilites. These are mainly euolivolent amholite proteins, which are assumed to contain in the large structure a number of cationic and anionic centers. 5, pages 296 seller. The anionic centers are the site of an electrostatic adsorption of basic dyestuffs, which generally exceeds the adsorption of acidic dyestuffs at the cationic centers. Because in cell colloids, the anionic character ordinarily surpasses the cationic, as indicated by the generally greater effectiveness of cations. The electrostatic adsorption of dyestuffs, including the acidic ones, is rather stable due to the colloidal or semicolloidal behavior of the dyestuffs themselves, which prevents dissociation and diffusion. 2. Acidic dyestuffs. These are the neutral salts of organic carboxylic and mostly sulfonic acids. In contrast to the aforementioned basic dyes, most of them are strong electrolytes, especially the sulfonic acid dyestuffs. In this respect, their penetrating power cannot be expected to be much influenced by additional acidification or alkalinization, as was that of the basic dyes. In general, the acidic dyestuffs are lipoid insoluble, except a few methyl orange, tropolone 0 and 0, brilliant orange G. Only a slight increase of lipoid solubility can be attained, even when the alphalecity of these acidic compounds for neutral oil is strengthened by the addition of diamylamine, as that of the basic dyes by the addition of oleacid in the experiments of Nierenstein. This suggests that the large body of nonpolar radicals attached to the chromophoric part of a dianion provides enough organophilic, lipophilic strength to overbalance the great hydroaffinity of the sulfonate group, see Sec 5, Chap 20. In any case, according to Nierenstein, at least a slight capacity to enter paramecium can be observed with those acidic dyes, whose entrance into an oily phase can be brought about by diamylamine, CP235, however. In extending these physiological studies of the properties of the sulfonic acid dyestuffs to many different objects, one encounters the embarrassing fact that, while many cells fail to show any color after a long exposure to strong solutions of readily diffusing, non-toxic lipoid insoluble sulfonic acid dyestuffs, other kinds of cells accumulate them quickly and copiously. This uptake is to be referred to an active transport brought about by the living cells, since it can be inhibited or diminished in a reversi bull manner by narcotics, by an anoxia, by low temperature, and by other means. It is encountered particularly with animals in gland cells, adenoid activity of Overton, in cells with phagocytic or with strong metabolic functions. This will be discussed in detail in Sec 8. Here, it may be mentioned briefly that liver and certain kidney cells, when exposed to the dyestuffs in question, display a homogeneous coloration of their protoplasm distinctly more intense than that of the surrounding solution. Often the color is raised to an especially great concentration in vacuoles inside the cell, or in the secretion expelled by the cells. Chap 35.2 and 38.2. Only when the dye offered to the liver or kidney tissue is highly colloidal suspensoid. In its solution, do the cells fail to incorporate it. Numerous phagocytic cells, fibrocytes, histiocytes, throughout the body appear loaded with the particles. To this class of cells also belong the reticulate epithelial cells of the liver stellate cells of Kupffer. The majority of cells remain colorless in solutions of the readily diffusing lipoid insoluble sulfonic acid dyes, but special cells are encountered near the vascular bundles which take up the dyestuffs, except in the presence of ether in narcotizing concentration, or during anoxia. Each plasmolysis or deplasmolysis in plant CLs, each shrinking or swelling, decrease or increase of volume in animal cells, achieved by placing the object in an anisotonic solution, indicates permeability to water. In general, this is manifested by entrance or exit of the water toward an equilibrium state, which, as previously shown, can be attained at a speed that depends upon the penetrating power of the dissolved substances, their oil, water distribution coefficients, or their MV. True rates of permeation of water, however, can be determined experimentally and directly only by establishing the osmotic gradient across the cell surface with dissolved substances, which are unable to penetrate, for the simultaneous pymulation of solvent and solute necessarily influence each other, since the rate of entrance of water at any given instant depends upon the amount of dissolved substance that has already mittered the cell, and vice versa. Taking this into consideration, the following procedures have been employed, dealing WDD at the change of volume of various single cells. 1. Sea urchin. Due to their spherical form, the volume of sea urchin eggs can be determined by measuring their diameter, either individually under the microscope, or more accurately by a diffraction method, which gives a statistical average of the diameters of many thousand single cells. Simultaneously observed, the rate of volume change can be bold to that describing Pick's law of diffusion, where phi is expressed in cubic micra, the surface area A in square micra, the concentration difference between inside and outside ACI, KCI, in atmospheres of osmotic pressure, and where IC is the permeation constant. Then K is computed from a convenient integrated form of this differential equation. In of time, density across the area, see further R. Callender and H. Berland Actabot. Then, 11. Experiments upon unfertilized Ardbasia eggs, for instance, swelling, observed in 2040, and 60% seawater over a period of 15 minutes, indicated that the permeation constant Kk at 20 degrees Celsius was 0.1 of water per surface per minute for each atmosphere of difluorines in osmotic pressure, with a longer duration of the experiment, or WDH, a greater dilution of the seawater. Slight deviations of the constant appear, which probably are accounted for by incipient disintegration of the surface. With other species of echinoderms, nearly the same values were obtained, Kk 0.1 to 0.4. It does not make much difference whether the determinations of rate are made to erythrocytes. A certain small amount of blood or of a dense suspension of blood corpuscles is rapidly mixed with a certain large volume of distilled water or of several hypotonic solutions of an indifferent substance, sucrose, dextrose, and the increase of transparency of suspension is observed until the osmotic hemolysis has reached a certain arbitrary degree, CG 75%, again, the increase of volume with time is expressed co, which, provided one neglects, for simplicity, the escape of salts, the osmotic resistance as influence to where Ci is the initial concentration inside the cells and co the concentration, at which the arbitrary degree of hemolysis is observed. For certain reasons, only observation times up to about 1.8 seconds are regarded as give incorrect figures. Following this procedure, K has been calculated for ox cells to be about 2.5. For human 3, plant cells. In corresponding studies on plant cells, the volume changes can be measured by plasmolyzing cells, which have a sufficiently regular cylindrical shape, CPM. 235 degrees, the rates of water penetration have been shown to vary from species to species and from site to site of the plant body over a wide range, more than 51. This may be referred to differences in viscosity of the protoplasm, adhesion of the protoplast to the cell wall, and other special conditions. It has been mentioned before that one can obtain an exact value for the rate at which water leaves cells in a hypertonic solution only when the solute applied to the surface is one to which the cell is impermeable. But if a penetrating solute is applied in the presence of an isotonic solution of a non-penetrating one, for example, 0.5 multi ethylene glycol to sea urchin eggs in seawater, the rate at which the solute enters, as well as the rate at which water leaves, can both be calculated from certain measurements. Initial shrinkage appears, which is followed by return to the original volume as the penetrating solute becomes equally distributed between cell and surrounding. This is analogous to plasmolysis and deplasmolysis mentioned earlier in connection with the behavior of plant cells immersed in a hypertonic solution of a penetrating non-electrolyte, P229. In this process, the two permeation rates considered independently of each other would be described. Jacobs has shown that 3 determinations, 1, the initial volume of the cells, 2, the minimum volume, and 3, the time required to attain this volume, are sufficient to yield the separate values of FCOI and I water from a 1. From experiments of Luxiques, Hartline, and Ricca on Ardbasia eggs. 2, from experiments of MH. Jacobs on oxerythrocytes, F quadr 2.5 VMV. Data mentioned earlier in this section make it clear that the rate of water permeation can differ from one object to another and can vary depending upon outside conditions. This is especially evident in studies of plant cells where considerable differences up to more than 51 have been met. Little is known concerning the nature of these differences. Probably the penetration of water is mainly affected by mechanical properties of either the surface membranes, including in plant cells the vacuolar membrane, or the protoplasm. It must be kept in mind that there are reasons to believe that, to a certain extent, the plasma membrane is a sieve-like structure, P238, so that the pathway of water is like the interstices of a filter. This is more or less true also with the protoplasmic cell body. Sexy 5, the building material of this sieve structure is partly constituted of various hydrophilic colloids, see Sec. 2 and 3, so that water traverses not only interstices like pores, but also intermicellar spaces between the colloidal aggregates which build up the substance of the cell. The penetrating aqueous solutions interact with the hydrophilic colloids to bring about a looser or more condensed architecture, which affects the micro and submicro porosity, so that permeability increases or decreases. In any case, it seems a clarifying viewpoint to consider that the permeation of water driven by osmotic pressure across a protoplasmic membrane is like a filtration, and to refer variations of the permeation rate, as brought about by solutes such as inorganic ions, to their well-known interaction with hydrophilic colloids, see Sec 5, Chap E. Such influences have been observed by Lucca and McCutcheon. Sea urchin eggs were placed in a solution of 0.38 mol dextrose, which is approximately isotonic with 40% seawater. The permeation rate was found to be about 0.1. To this sugar solution were added electrolytes in such a small concentration, 10 to 10 mol, that their osmotic effect as such was practically zero. It appeared that various ions had a significant effect upon the migration of water. For instance, monovalent cations, NaK increased the permeation, divalent KMG decreased it. And in appropriate mixtures, they antagonized each other in the typical way, see Chap 16.2a. In this connection, it should be mentioned that narcotics inhibit the osmotic permeation of water, and it is assumed that the hindrance is due to their adsorption at the micellar surfaces, see Sec 5, Chap 23, too, so that the pathway has become narrowed or blocked. In concluding this chapter, attention should be called to some striking physiological phenomena, characterized by the fact that water traverses cell membranes in the absence of a hydrostatic or osmotic drive, or even against such a force. Most of the work on this subject has attempted to explain how the isolated frog skin, as long as it survives, develops a force driving a normal Ringer's solution from outside to inside, even against a hydrostatic pressure. This has been found to depend on the supply of energy by certain metabolic reactions, and to be bound up with electric differences of potential maintained by the skin. The situation resembles anomalous osmosis, in which water, by virtue of an electrokinetic charge, is transported by a membrane potential resulting from the selective ion permeability of the membrane. For other similar effects, and for the theory of anomalous osmosis, see Sec 8.1, the identification of a plasma membrane. It has been concluded from experimental study of cell wire permeability that the surface of protoplast is formed by a special organ, the plasma membrane. Often, this membrane is covered by one or more protecting or supporting extraneous coats, which seem to be readily permeable to crystalloid solutes, except those with very large molecules. These hyaline layers and jelly-like coatings can be removed mechanically, leaving the essential protoplasmic surface layer entirely viable and hence the natural cell permeability intact, although the plasma membrane itself is not clearly distinguishable under the microscope. Its existence, for various reasons, cannot be denied. One of the most convincing evidences of its reality is provided by the following experiment of Chambers. Starfish eggs stained with the intracellular indicator neutral red are placed in isotonic Nessig-4C solution. The color of the egg shows a change to a more alkaline reaction due to the penetration of NIT3, which results from the slight hydrolysis of the salt bauxite instead. Some of the same NE4CI solution is injected with a micropipette into the interior of one of the eggs. Immediately, the indicator assumes a distinctly acid color, and due to the escape of NH3 into the surroundings of the cell, this color spreads in all directions throughout the protoplasm, but not farther than the boundary of the egg has been reached. In a core spawning experiment with an alkaline solution of NaHH3, which contains a considerable amount of KO2, neutral red indicates an acid reaction inside the eggs, resulting from the free entrance of Co. But following the injectee of the solution into one of the eggs, the acid reaction turns to an alkaline one, with the color again spreading as far as to the barrier at the cell surface. Furthermore, the existence of a film-like surface is shown by quantitative studies of swelling and shrinking of the cell body when exposed to heterotonic solutions of various concentrations of a non-penetrating substance. For example, glucose or diluted or concentrated seawater. Provided the cell behaves as a perfect osmometer, the volume changes should follow the Boyle-Van Hoff law, PV equals constant, while if it resembles a membraneless gel, as sometimes has been asserted, the volume changes produced by the various concentrations would not even remotely follow the law. Many experiments have shown that actually the law is obeyed satisfactorily. If one takes into account the fact that each protoplast must contain a dead space filled with osmotically inert substances, such as lipoids and proteins, which are more or less dispersed in the cell body, possibly forming a kind of stroma, whereas the remainder, VFA, holds the osmotically transferable free water, in other words, PFH const. The well-known Donen equilibrium likewise indicates the presence of a membrane. When a membrane is freely permeable to the ions of an electrolyte solution, which bathes both sides of it, but is impermeable to a colloidal ion on one side, for instance, intracellular, an unequal distribution of diffusible ions takes place, expressed by the equation YoE plus S, where X and Y are the diffusible Z, the indiffusible ion, see Sec 1 Chap 5. This distribution has been observed frequently, for example, with red blood corpuscles, see also Sec. 8. Introduction. Even more demonstrative of a special surface barrier is the fact shown been applied to cells only special groups are permitted to penetrate. It is just this property of selectivity that poses the problems of the chemical nature and of the physical structure of the cell surface. It has been found that the two classes of non-electrolytes which are able to enter cells are first, those which are soluble in fat-like solvents, and second, those which have a small molecular size. The problem is to find out whether the chemical and physicochemical behaviors of isolated plasma membranes lend support to the postulates of fat-like and sieve-like constitution. 2. The chemical composition of plasma membranes. Unfortunately, direct information about this composition is exceedingly poor. Practically the only membrane material available in quantities sufficient for chemical analysis consists in the stromata or ghosts of the red cells, and possibly these are only partly pieces of the plasma membrane and partly remnants of a skeleton or meshwork extending through the cell interior. According to Parpar and Zemian, the chief building stones of this structure are lipoids and proteins in the average ratio 1.7. Predominant among the lipoids are the phospholipids, which are hydrophilic colloids. The fairly hydrophobic cholesterol makes up the remainder. The latter and some of the proteins and lipoproteins, which are not more than moderately soluble, seem to confer, at least, some of their characteristic consistency upon the stromata, evinced, for instance, under the influence of fairly strong swelling agents, by the formation of threads, globules, and other products of a gradual disintegration, stromatolysis, resembling the myelin forms of nerves. These results obviously provide an adequate foundation for the theory of permeability. They supplement numerous earlier statements about the dissolving power of cell lipoids, and they allow one to picture protein aggregates as porous structures, chapter 16 and 17, by applying modern concepts of hydration and dehydration of colloids in membranes and fibers. However, no analytical results have so far been brought to bear upon the great variety and relative importance of the solvent factor versus the pore factor in permeability, CP 285, which possibly could be interpreted as due to differences in the relative extent of the lipoid and protein areas in the membrane mosaic, or B, upon the possibility that the chemical composition of the phospholipids may vary from plant to plant and from animal to animal with regard to the degree of acidic or basic character. 234 FF, or of the ratio unsaturated saturatemyl fatty acids, Sec 5, pages 351 and 359, and other factors. It further should be recalled that the present data of quantitative analysis presented refer to red cell stromata only. 3. Physics of the plasma membrane. These latter remarks call attention to the problems of the architectonics of the plasma membrane. Their study has been stimulated and rendered more accessible by the background investigations of Langmuir, Harkins, Adam, and others into the formation and properties of surface films. In view of the dimensions and the chemistry of the plasma membrane, it is important that these recent studies have made it possible to compare natural films with artificial monoion multilayers of fatty acids, sterols, and proteins. Also, with mixed films, the components of which can appear in separate strata or can interpenetrate each other and mutually influence molecular orientation. And with other types of membrane as shown and discussed before Section 2, pages 203 FF. Some of the results of comparing artificial and natural architectures will be referred to in the following Section 5, particularly regarding mixtures of lipids with proteins and the influence of hydration and dehydration upon their microstructure, Section 5, pages 295 FF. Much effort has been spent in trying to construct models which portray the natural surfaces with regard to certain features. In this effort, chiefly, the following properties have been studied. A. The interfacial tension of protoplasts. B. The thickness of the plasma membrane. C. Its electrical conductivity. A. This interfacial tension of protoplasms. This can be measured in invertebrate eggs, which behave like fluid spherical drops when ei distorted, C. betoporus, or of Arbacia. Due to the different specific gravity of the oil globules and the yolk granules included in their protoplasina, are elongated more and more until at a certain force the cylindrical cells break into dough. This can be watched or photographed with a microscope centrifuge. From the force applied and the diameter of the breaking cylinder, the interfacial tension, cell surface seawater, can be calculated. Cole, for the same purpose, pressed a beam upon the top of the sea urchin egg and measured the 2 radii of the flattened cell at various degrees of deformation. The cell surface is formed by a fat-like substance, since fats are known to show a considerably higher tension. As a matter of fact, Harvey and Shapiro's Ching found the tension of fish oils from mackerel and other teleosts at a pH corresponding to that of cells. To clarify this situation, they determined the tension of the oil droplets, which are often included in the eggs of fishes. Centrifuging flattens the droplets by pressing them against the tough outer membrane of the egg. Then, from measurements of the deformation, the interfacial tension, oil egg fluid, can be calculated. It was found to be much lower than the 10 psi or water, about 0.69 centimeter from this. Daniele and Harvey have drawn the conclusion that, among the contents of the egg plasma, there is some substance which has a great surface activity toward the oil, and they offer reasons for assuming that this substance is an egg globulin which forms a film and covers the surface of the oil. CSEC, 2 pages 200 FF. The picture shows 2 monomolecular arrays of fatty molecules, each of them stabilized on one hand by anchorage of their polar groups in the schema of molecular conditions at the cell surface. Aqueous phases and, on the other hand, by lateral adhesion forces between the hydrocarbon chains. Each stratum is covered by an adsorption layer of hydrated protein molecules, which thus provide toward the exterior, as well as toward the interior, the characteristic low interfacial tension. The space between the 2 lipoid layers may be thought of as filled with unoriented Lassu-oroid molecules, so as to allow for the variety of the thickness observed among such films, and discussed in the next section. Some objections may be raised against such a picture representing the aforementioned studies. First, Mudd and Mudd have investigated the wetting properties of cells by observing whether at a saline oil interface originating between a microscopic slide and a cover slip, the cells are hydrophobic enough to enter the oil phase preferentially, or hydrophilic enough to enter the aqueous phase. Erythrocytes were shown to be more hydrophobic, leukocytes hydrophilic, and for this reason the former were assumed. With regard to the erythrocytes, this behavior is in agreement with their electroihortic properties, as the F potential is not affected by adding protein to the rinsed cells, nor by changing the pH, see Sec. 5 Chap. 18. It is doubtful whether this objection is conclusive, as, according to the discussion of Bateman, the Oryx notation of molecules in mixed films containing different lipoid components, oil sterols, and protein is exceedingly intricate, due to adsorption and displacement, to mutual dissolution and esteric factors. Second, the picture in question is probably much too simple. With regard to erythrocytes, this follows not only from chemical analysis of the stromata, P272, further, Sec. 5, P294, but also from optical analysis with the polarization microscope. The latter reveals a many-layered structure of protein lamellae with long axes oriented tangentially. That is, with flat surfaces parallel to the surface and interposed lipoid micelles, whose optical axes are arranged radially, see Sec. 5, pages 296 ff. Nevertheless, the natural architecture of the stromata might be crudely represented by the proposed scheme, at least with respect to stratification. Third, the picture seems to ignore the rather well-established concept of a mosaic of lipoid and protein areas. As a background for the solvent sieve theory of permeability, CP 234. From the viewpoint of surface films, Daniel Isaacson has objected to this theory that surface tension forces acting at the border of the diflurin areas, lipoid and protein, of a mosaic membrane would tend to disrupt the film, since the lateral adhesion of the hydrocarbon chain, which stabilizes the lipoid area, would not extend to the protein area. On the other hand, it has been asserted that a lipoid molecular surface layer alone can suffice to account for permeation of a solute through pores, as well as by being dissolved. In this regard, Michaeliszek was the first to point out that, even in a membrane consisting of a layer of liquid, which functions distinctly as a solvent medium, the penetrating molecules must pass through interstices between the molecules of a solvent, just as they do in diffusing across a molecular sieve like a dried collodion membrane, although there is a marked difference, since the intermolecular spaces in a liquid membrane can be easily deformed and enlarged when penetrating substances squeeze themselves between, whereas the intermolecular spaces in the collodion membrane are preformed in a rigid structure with solid phase. However, the contrast between the classic gyrus membrane of dried collodion and the solvent oil membrane has been overbridged for a long time by Northrop, Michaelis, and Collender, and recently especially by Solnerjit. They showed that rigidity is not an essential character of the collodion membrane. On the contrary, it may stubble exadiol by the uptake of water and change its porosity, and it can display selective permeability toward organic compounds, not unlike that of an ordinary solvent membrane. In other words, permeability depends upon the molecular volume of penetrating compounds as much as upon their affinity toward the membrane as a solvent. See this chapter, pages 235. Returning at last to the picture of a plasma membrane, figure 27, one should inquire after the possible significance of the superficial layer of protein, according to Harvey, Daniel A., and Davson, Zoctatoy. Writ, it may be assumed that adsorbed protein provides the characteristically low interfacial tension of about 0.6 dyne centimeters, found from marine eggs, and so protects the membrane against rupture by surface forces. Without it, rupture could be brought about as easily as in those artificial films, the polar groups of which are in immediate contact with the aqueous phase. 6. The thickness of the piocumtum membrane. Much effort has been spent to solve this important problem by exploring it with chemical, optical, and electrical means. But so far, the yield has been rather poor. Chemical methods. For chemical analysis, cytolized erythrocytes are unique material in being available in sufficiently great amounts, although one has to take into consideration, CP 272, that the ghosts are possibly an aggregate not only of plasma membranes, but also of a meshwork from inside the cells. The study of the ghosts has appeared especially enticing to those in favor of the rather common assumption that lipoids are the backbone of the cell surface structure, after it has been stated that the entire amount of lipoid contained in the erythrocytes is bound up with the ghosts. The following procedure has been chosen to measure the thickness of a supposedly real continuous lipoid layer spread over the surface of the cells. The lipoids of a certain amount of rinsed erythrocytes, the number and the size of which are known, are extracted and dissolved in a small amount of petroleum ether and are SF red on water. A rhinomolar coherent film is formed and its area measured, see Sec 2 Chap 0. This area is found to be twice as large as the total surface area of the erythrocytes used in the experiment. This proves that if all the lipoids are spread on the surface of the erythrocytes, the film must be bimolecular. Taking into account the average percentage of each of the different lipoids mixed in the surface membrane, and the length of their molecules, the thickness of the membrane can be roughly estimated to approximate 48. Obviously, one can make several objections to this interpretation or to the generalization from the experimental data. First, in the structural scheme derived from measurements of the surface tension of cells, figure 27. There likewise appears a bimolecular lipoid layer, but in addition, as components of the plasma membrane, two monolayers of protein, to the thickness of which, according to numerous observations with protein films, C and lipoproteins are regular components of the ghost P272. Third, it remains an open question whether or not the lipoid molecules are arranged as a discontinuous bimolecular layer with protein molecules interspersed in a mosaic pattern, accounting for the permeability proerties of the plasma membrane. Optical methods. These likewise have failed so far to give unequivocal results. From their measurements with polarized light, P275, Schmidt, Baer, and Ponderi came to the conclusion that the envelopes of the red cells are many molecular layers in thickness. A more direct approach to an exact measure has been made by Waugh and Schmidt with their analytical leptoscope. Light is reflected from a step film of barium stearate, that is, from a film which is built up on a glass or metal plate by superimposing one monolayer above the other, and this light is compared with the light reflected from ghosts, which have been dried on another plate. One has to observe which step of the calibrated step film matches the ghosts in intensity of reflected light. In this manner, a thickness of more than 200 A was measured with the dried ghosts of rabbit erythrocytes when hemolysis had been produced at pH 6 of the hemolyzing buffer solution, the thickness diminishing on either side of PL6. After extraction of the ghosts with a lipoid solvent, the maximal thickness dropped from 200 to about 120 A. This possibly indicates that proteins share in the thickness, electrical methods. Assuming that the erythrocytes are surrounded impedance of suspensions of erythrocytes and has interpreted the results as due to the presence of a certain static capacity of the condenser-like membrane. From this value and further, assuming that the non-conducting material is fat-like and therefore may have a dielectric constant as low as 8, the thickness of the membrane was calculated to equal 88 A. We will come back to this kind of measurements in the next subsection, P280. It is obvious that the information concerning the thickness of the plasma membrane is far from being final, because even with the Sanre object, according to the measure applied, the numerical values are scattered over a wide range, from 88 to 200 A, because for technical reasons, the studies mostly were confined to erythrocytes, and because even in the study of the erythrocytes, essential features have been disregarded. Whenever active transfer, P249 and SEC.8, is looked at as a matter of permeability, conduction by the plasma membrane, controlling at least the entrance of substances, then the question must be raised whether the concept of the plasma membrane must not be widened to comprise more constituents than have been pictured in many of the proposed schemes. One would have to take into account certain metabolites and enzymes to explain, for instance, in the case of erythrocytes, why the back transurdative K ions, which have somehow escaped from the interior, P249, appears to be conjugated with the glycolytic breakdown and of carbohydrates, P251. It is hard to believe that such a film as the envelope of the red cells, as currently pictured, is capable of this complex action. More likely, essential components have been extracted or destroyed in preparing the ghosts. Further, one should recall that in plant and animal protoplasts, there has been observed a more solid cortex of varying thickness, lying directly beneath the plasma membrane. See the electrical resistance of the plasma membrane. For about 100 years, by the investigations of Dubois-Lallemande and others, it has been known that living tissue is a poor conductor for the electric current, and that the resistance falls off in death. This process is accompanied by exosmosis of electrolytes. In many plant cells, the loss is chiefly due to the escape of the ions contained in the cell sap vacuoles. In animal tissues, the source is the protoplasm, where the ions could be assumed either to be freely present or to be liberated during its disintegration. Conclusive evidence of one factor or the other being mainly involved would be provided by measuring the internal conductivities of the cells. It means one should determine the electric conduction of a solution which is contained in a closed vessel of non-conducting material. As a matter of fact, this can be cloned by special procedures, with the result that the interior of normal cells is shown to be a good conductor, and that therefore the high electrical resistance of living cells must be referred to their surface. However, ordinary electrical methods also can avail for measuring the resistance of cells, viz. Of plasma membrane plus cytoplasm. The electrical conductivity of a bry of centrifuged erythrocytes suspended in serum or in isotonic salt or sugar solution has been determined with low-frequency alternating current, 100 to 200 cycles per second, following Tilly method of Kohlrausch-Dornbein. The conductivity was found to decrease with increase of the percentage volume of cells, according to Clerk-Maxwell. The specific resistance of suspended particles can be calculated from the formula. Where R is the specific resistance of the suspension, Rho that of the suspending liquid, R2 the specific resistance of the Golgi-ended particles, and P the volume concentration of the suspended material, with red blood corpuscles. By varying the ratio of cells suspending fluid, R2 was found to be practically infinite. In other words, the red cells appear to be practically perfect. The common EG procedure of measuring resistance double on a Wheatstone bridge by leading a direct current across a multicellular tissue cannot contribute much information to our problems, as Tilly intercellular spaces as pathways for a part of the electric flow remain uncertain. This imperfection can be more or less overcome with single giant cells as objects, as for example, Valonias or Niphalod or the axon of the squid. In experiments with the plant cells, the DC has to pass the protoplasm, the plasma membrane, the vacuolar manilrane, and cell sap, and, moreover, a bypath is open to the cell W-all. The squid axon provides more favorable conditions as the actual resistance of the axoplasm, compared to that of the enveloping membrane, is small. When DC is applied, care has to be taken that the current is not allowed to flow for more than a few seconds. Otherwise, polarization of the membranes can be succeeded by altercations of the normal resistance. C. 5, Chap 22. Also, the current strength must be small enough not to release an excitation wave. The DC resistance with the squid with Nutella 100 0 to 200 0. The latter value is possibly due partially to the fact that the outer cell wall of this tap water alga is soaked with toppy water, while the habitat of Valonia is seawater. From these various values, it may be concluded that the plasma membrane, though not a perfect insulator, actually offers an extremely great resistance to ionic penetration, for example. For the squid axon, when we assume the thickness of the nerve membrane to be 100A, the resistance would be about as great as that of glass. The RR salts are rather the same when, instead of DC, low-frequency AC is employed, as is customary. After Kohlrausch J. Rowe owes this form of current for measuring the resistance of electrolyte solutions in order to avoid the polarization effects. But the conclusions from all experiments regarding the resistance of the plasma membrane seem to be entirely upset when one turns over from low frequency to high frequency AI when an electrical potential difference is established across A. Membrane, which is nearly, if not entirely, impermeable to ions at the first moment on one side of the membrane, cations will GNO VC from the electrode toward the membrane. Anions in the opposite direction, the result is a positive charge. The corresponding events appear on the other side of the membrane dorine. This accumulation of ions proceeds until the excess charge prevents further accumulation, so that a certain potential difference is built up, depending upon the charge density and the thickness of the membrane. The ionic flow is stopped. This is like charging a condenser, the static capacity of which is C equals Q vis Q, being the charge and by the potential difference. This static capacity with a variety of cells, erythrocytes, echinoderm eggs, muscle, nerve. Plant cells has Bgen found to be about 1 microfarad centimeters too. However, when instead a high frequency alternating potential difference is applied, ions will have hardly begun to be piled up at the membrane before the direction of the potential is reversed, and they will travel in the other direction. Thus they swing as though no membrane were present in the electrical circuit. But in addition to these electrostatic effects, which refer to ions unable to pass across the substance of the membrane, there can be more or less ionic shift through a resistance, as for example, in a cell membrane, which, due to its architecture, allows a certain amount of real ion permeability. Such a system works like an AC circuit with a static capacity lying parallel to an ohmic resistance. In this system, the maximum current and the maximum potential do not coincide as they do in the absence of a condenser, but follow each other in a certain phase difference varying with the ratio. In such a system, the apparent resistance, impedance, decreases with increasing frequency. Turning now to physiological objects. First, the behavior of a suspension of red blood corpuscles has been studied. The cells are rinsed with isotonic sugar solution and centrifuged to the highest possible volume concentration. Supplied with low-frequency AC current, 100 to 200 cycles sec, their conductivity may be found to correspond to about 0.02% nickel. The high-frequency conductivity is much greater. This has been determined by the way of an induction coupling with the oscillations. This circuit contains, besides a self-induction coil, a variable plate condenser, and a second condenser, which is a glass trough with the metal plates as electrodes, and filled with water. The dimensions of the self-induction and the capacity are such that the frequency of the oscillations amounts to several millions per second. The two circuits, the generator and the receiver, by slight variations of the condenser capacity in the receiving circuit, are brought to maximal resonance, indicating maximal amplitudes of the oscillations. Then the trough is filled with a certain volume of the cell suspension, and, due to the presence of ions in the cells, a certain damping effect of the oscillations appears. This effect can be matched by filling the trough successively with a series of nickel solutions of different strengths. Concentrations between 0.1 and 0.4% were found to be as equally damping as the cells, comparing to the 0.02% nasal matching with the cell suspension at the low-frequency oscillations. The conclusion that the high frequency values are representative of the internal conductivity of the cells is corroborated by the observation that hemolysis saponin, in other words, opening of the envelope of the cell contents, does not, or does only moderately alter the high-frequency conduction, whilst the low-frequency conduction rises considerably, for example, 12 ohm to 165 ohm. The high-frequency current penetrates the condenser-like plasma membrane and passes through the consider ably ionized contents of the cytoplasm. In this manner, one can follow that, starting with about 100 oscillations per second and progressively rising to several millions, the conduction by the interior of the cells increases more and finally reaches a maximum, which in this case, and similarly in others, is about ES to percent of the conduction of the outer milieu, serum, ringer, or seawater. This behavior of the cells is pictured diagrammatically in figure 28, showing the streamlines on their way from seawater through an unfertilized and a fertilized sea urchin egg at different frequencies, indicated in kilocycles. According to Kohlberg, it is made clear that at about 1 kc, the current flows entirely around the unfertilized and the fertilized egg, that in the neighborhood of 50 kc. There is still very little current flow through the plasma membrane, but considerable current is passing through the fertilized T-John membrane, and as the frequency is raised to about 1000 kc and more. The streamlines also penetrate the plasma membrane and spread through the cytoplasm. However, by the extensive and intensive studies during the last decade, with increasingly refined methods, ample evidence has been provided that, following the passage of alternating currents across the various kinds of cells, a multitude of modifications of the time and strength relations of the current are affected, which reflect differences or alterations of the architecture of the cell membranes during rest and activity, during excitation, narcosis, injury, decay, and death. For example, during rest, either the factor of ion conduct ion or the factor of condenser charge and discharge can be predominant, SCG, shown by comparing sea urchin eggs and muscles on one hand and red cells on the other or during activity. The condenser properties of the membrane may display rather no change, while ion permeability does to a great extent, as for example, after stimulation of a squid axon or of a Nutella cell, P3 and 16. It is beyond the limitations of this chapter to outline the methods and discuss the results in their details day bit. It is evident that new ways are opening to describe essential features of CTDL life by the way of the electrical characteristics of the CTDL life, Cells in terms of molecular arrangements and rearrangements. Chapter 6. Section 5. Influence of some extracellular factors on cellular activity. Cells, per natural conditions, are embedded in a fluid medium, the chemical and physical properties of which are rather constant. Deliberate changes of these standard conditions, are to be of essential interest, as usually they bring about functional changes resembling those which are observed under normal circumstances. Such experimental procedures, a resting state can be converted into one of activity. Existent activity can be increased or depressed. For instance, one of the most common signs of activity in living things is motion of some kind, protoplasmic streaming or amoeboid movement, or arythronic shift to and fro in protoplasm, or, most important, cholera, fusion and reduxation can be excited or inhibited. Even development and growth can be deciphered as a kind of motion. Furthermore, changes in electrical behavior are initiated, causing potential differences to appear and disappear. The continuous chemical processes of metabolism, including respiration, can be made to rise or subside. By this way of provoking from the outside artificial changes of activity which resemble normal functional change, one may draw conclusions as to the nature of the processes which underlie both the natural and the experimentally produced events. The extracellular factors which shall be considered here are revealingly chemical ones. Site of the attack might be expected to be either the cell surface or the cell interior as constituents of the cell body which, according to the preceding chapters, can be differentiated fairly well as to their microscopic and their molecular structure. The chemicals which will be mentioned here can be grouped roughly as follows. 1. Chemicals which, at least primarily, are unable to penetrate the cell boundary, and therefore must be assumed to act by changing the properties of the surface layer. Substances of this kind are, first of all, the inorganic salts, but there are also many more substances which often fail to enter directly. For example, organic non-electrolytes, like sugars, polyhydric alcohols, aliphatic acetamides, and amino acids, which seem to be rather inactive toward the cells, to chemicals which enter the cells due chiefly to adsorption or to lipoid solubility or to diffusion, and which for this reason react primarily in the cell interior, but eventually at the surface as well, Among the substances of this nature will be mentioned here, mainly narcotics and cytolytics. It is not the plan to present in the following section a survey of the entire field of extracellular influences. Instead, main results will be selected and analyzed in order to gain theoretical viewpoints which are useful in the interpretation of increasing and decreasing cellular activity in terms of physicochemical phenomena. The influence on the excitability of muscle. No material can be more promising for studying changes of activity due to changes in tri-ionic MTDM than muscle, particularly after, some 60 years ago, certain fundamental, pertinent facts WT discovered. JT was demonstrated by Ehlering that the frog muscle begins to twitch when it is transferred from its natural milieu into physiological salinine solution. Using the heart as Ringer found that the rhythmic action is decidedly interrupted by adding K and A to the normal saline, while Claude Bernard noted that K had a paralyzing effect. Further, W. Biedermani reported that Ka locally increased at the muscle surface produces a reversible electronegativity confined to the treated RA. With this background, the physiological properties of the alkali and the alkaline earth cations and of the inorganic anions have been conjured in numerous experiments. Overton, studying workability to organic and inorganic compounds by elacing frog muscles in isotonic solutions, was amazed to find that in solutires of sucrose, glucose, or mannitol, the muscles, although failing to show any changes in weight or other signs of alteration, lost their electrical excitability, but recovered it after the addition of as little as 0.07% nickel. Continuing the experiments by varying a kind of salt, he discovered that any neutral Na salt, irrespective of the accompanying anion, restored the excitability, but of the other alkalications only was a fairly effective substitute for Na, while S restored slightly and for a short time, and Rb and K not at all. Studying the change of electrical excitability in the isotonic solutions of the various alkyl chlorides. The result was that the excitability was longest maintained in nickel and decreased through the series. Not nil or BYK, however. K has long been known to have a stimulating effect also. For instance, if a small amount of isotonic KTLA is added to ringer bathing a frog sartorius, the muscle makes a number of spontaneous twitches or falls into contracture, which may or may not be reversible. A muscle can be sensitized for this effect to various degrees by placing it for a few minutes in an isotonic mixture of ringer plus one of the alkali chlorides. In this way, it has been found that the stimulating effect of the alkali ions is strongest with K and decreases in the following gen series. K core B gen Shi Li, which is rather the reverse of the first mentioned series. Whether the stimulating or the paralyzing effect appears depends upon the concentration of the salts as well as upon the duration of their action. The stimulating effect is frequently reversible, the paralyzing frequently irreversible. The inorganic anions show a corresponding behavior. Studied by the sensitization method of Lilly, the anions appear to raise the excitability in the direction. So for URR not 1-LSEN. I hit with higher concentrations or after a longer exposure, the stimulating influence of I and SN changes into an inhibitory and irreversible action. The gradual change of the stimulating or the inhibitory effect of the anions is especially well demonstrated in the experiments of H. Schwartz to the frog muscles hanging in isotonic solutions of sucrose plus an salt equivalent to 0.2% nickel are electrically stimulated at intervals of 1 second. In sulfate, contractions stop after a small number of shackles. With acetate, somewhat more shocks are required. With NaCl, still more. And so on, so that the excitability appears to rise in the order S4 and CL I see it. Moreover, a muscle which is paralyzed in AS4 recovers in CL. One paralyzed in NO3 recovers in SN. But one whose contractions have been suppressed in bar does not start again in SOI or in TL. In Movender, regarding CN and I, this is true only with low concentrations. In higher concentration, for example, equivalent to 0.4% nickel instead of 0.2%. The muscle becomes fatigued in Cien, or I after a considerably smaller number of stimuli than in the lower concentralians. It relaxes more slowly. The single contraction ends in more incomplete relaxation. And after being completely fatigued, it may partially recover even in the presence of those anions, which usually are less favorable. The plurivalent cations. The alkaline earth chlorides presented in isotonic concentrations are rather incapable of maintaining the contractility MgCI. But in an infinite number of experiments, it has been shown that either an increased or a decreased functional activity produced by pure natural solutions or by solutions containing, in addition to Na, small amounts of K sufficient to raise or to diminish the activity can be brought back to normal by the addition of small amounts of Ka or SNR, also Mg, and to a lesser degree of Br. This balancing or antagonizing action of the alkaline earth ions appears in many variations in the functioning of plants and animals. The classical example is the composition of Ringer's solution and its favorable action upon a beating frog heart. Extending these investigations beyond the group of the alkaline earth ions, it has been found that the trivalent rare earth ions, for example, LaCe, are very toxic, whereas the DIN trivalent, not triunivalent, cations of the complex cobalt salts act very much like Fa or furthermore. The paralyzing effect of KCl 0.5% in standard saline 0.5% Na can be antagonizable by COMan, and to a smaller extent by Ni, while high FC1J2L high. And Zen were found unable to function in such a balancing action. Also, the spontaneous twitches of frog muscles induced by pure NaCl can be abolished by the same divalent ions. These results are analogous fundamental findings of J. Loeb concerning the capacity of Zen, Mn, Ni, Pb, and Uo to balance, in appropriate small concentrations, the inhibitory effect of large amounts of NaCl, as exhibited in the development FF, the fertilized B-lore proceeding in the narration of the multitude of findings concerning the effects of the neutral salts on the Mushingol activity. The Ferricutian observations will be subjected to an analysis essentially in terms of GEU physical chemistry, since the neutral inorganic strong electrolytes, at least the redominantly present alkali salts, are not involved by straight chemical reactions in the physiological phenomena under consideration. Very often, and for various reasons, the surface of normal cells has been believed impermeable to the neutral salts, mainly because in isotonic solutions, the cells keep their volume or their weight fairly constant, but undergo, in anisotonic solutions, corresponding changes of these manifestations of their osmotic conditions. This behavior complies as well with impermeability to cations plus anions, as for reasons of electroneutrality with impermeability to either cations or anions. Muscles laced in an osmotic solution, likewise, frequently are found to keep their weight constant or better, to show not more than slight changes. The nature of these latter is very complex. They can be the result of changes in the interfibral tissue space, which is OJN, the fertile fusion of both cations and anions in either direction. The ionic composition of this space varies according to the previous history of the muscle preceding work, slight injury, as well as to other factors. Two changes can be due to the colloidosmotic pressure of lip space 3. The fiber surface is permeable to the univalent cations according to their volume, which decreases along the series. This can be inferred from various observations, from a study of the height of the salt potentials produced by various cations, K-salts being found the most effective among the neutral salts. P 313. B, from the fact that, under certain experimental conditions, perfusing a frog muscle with a salt solution containing more K than 13 milligrams percent results in the penetration WNA less than 13 milligrams percent in the escape of, and that the entrance of K causes by ionic exchange, a loss of H, a rise of pH, and vice versa. This pH change is accompanied by a change of muscle weight. Greater increases in weight than those described CD above are observed in isotonic KCl, 0.83%, or in mixtures of this solution with isotonic natriol, about 150 milligrams percent Cl. However, this effect is more or less irreversible and is followed by death. The same is true with RBHA. But this increase in weight is dependent also upon the concomitant anions. Cl resembles BR-DOA N1, whereas with SOI, HPO4, CH3CO, even in isotonic concentrations, the muscle keeps its weight constant for a long time, although its excitability is lost. However, NaCl solutions contain a fate not more than about 0 milligrams PT-cent-KCl, though L-aerolizing the muscle failed to bring about a gain of weight. Therefore, one may conclude that, although permeable to K, Sly muscle is practically able to KCl out at least for some time, and it is still more so and for a longer time to those K salts, the anions of which, according to many other observations, have greater difficulty in penetrating cellular membranes, pages 450. This also refers to SO4, HPO4, CH3 in their behavior toward the intestinal wall, the kidney tubules, the frog skin, and plant roots, section 8. As to the higher concentrations of KCl exhibiting an irreversible increase of weight, this can be perceived on the background of the observation that the presence of abnormal concentrations of KCl releases several metabolic reactions. Starting with about 16 milligrams percent KCl in a ringer solution, the maximum excitability is reached with about 45 milligrams percent. Then the excitability falls off and simultaneously the O2 consumption rises. At about 80 milligrams percent KCl, phosphocreatine is decomposed, and a slight contracture appears, and at about 120 milligrams percent Keel lactic acid is being formed. 5. Lastly, in outlining this intricate picture of the factors which are pertinent in the osmotic balance of muscles as affected by neutral salts, it should be realized that there are reasons for believing that, in addition to passive penetration of ions across the surface membrane, a slow active transfer of ions takes place, as it does with erythrocytes. On the basis of these studies of the various effects of the neutral salts, we arrive at the final conclusion that the very first changes of excitability can be referred to processes going on in the outermost layers of the fibers, which only secondarily are penetrated by the ions. 2. The colloid theory of the change of excitability by ions. The statement of J. Loeb concerning TLI balance effect of multivalent cations toward isotonic natriol in the development of fungulus embryos. P291 suggested correlation of this physiological phenomenon with changes in the colloidal structure, because only the dispersion of inorganic suspensoid colloids at that time was known to be acted upon by ions according to the sign of tellur electrical charge and to their electrogalvanic rules Schulz and Hardy. The fundamentals of such a colloid theory of cellular action have been very much extended, vary on the basis of the experiments on muscles. These results have indicated first that the series of anions, CN, JL, or bar, CLL, so forth, showing the effect on excitability and on the related salt potentiles, 6p313, is paralleled by a series of anions, known as the Ilferneister series EL, or the hydrotropic, or more generally, the lyotropic series, which accounts for the dispersity of many hydrophilic organic colloids like gelatin or egg albumin, with regard to gelation viscosity, colloidosmotic pressure, and other properties. And second, have indicated that the series of cations K lib CS NALI, with its peculiar location of CA, see pages 289, 290, 315, likewise is met under certain conditions pH in the Physico J. E. Harris, J. Bile, Hem 141, 579, 1941, W. Wilbrot, Fliegers, Arch F. D. Physio 243, 519, 1940, See more details in Section 4, p. 251. It is important also to note that under the particular conditions of nutritional deprivation of K, the muscle fibers of rats, which normally are devoid of NAHAV, have been fooled to contain, besides a reduced amount of K, about these experiments on p. 257. Furthermore, K is claimed to be bound to the myosin threads, which hold up the fibrilla of the muscle CC1 to this location of us near to NAMLA instead of a location beyond K, as it should be corresponding to the scale of the cationic mobilities, CP317, also has been observed with nerves, E. Overton, Pfluger's arch, FDGs. Physiol, 106, 176, 1904, H. Netter, IHID R218, 310, 1927, R. H. Bieber and H. Strobe, even 22, 71, 1929, L. Out. Wasiliu and K. Iwano Ibid 236, 184, 1934, Ciliated Epicleia, TTS Lilly, MJ. Physiol, 17, 89, 1906, and 24, 459, 1909. Red blood corpuscles, R. Höber, Biochem, 14, 209, 1908, F. Port Deutsches Arch F. Clit. Matty 9, 259, 1910, C. Kreutz, Biochem, SD Schichter, 137. Physiol, 22, 819, 1939. WJV Osterhout, Ibid, 23, 53, 171, 1939. Potentials of muscle, RATDR, Pfluger's Arch FDGs Physiol. 106, 599, 1904. Chemical reactions of the organic hydrophilic colloids. These interrelationships have suggested that a dispersing effect upon structural colloids as components of the protoplasm facilitates the action, provided that the dispersion does not exceed a certain range of a swelling or loosening effect on the structure. Otherwise, the effect approaches moat and more dissolution as a stage of cytolysis, so that the initial favorable influence, for instance, of skin and eye or of cages 290 and 313, is reversed, provided the effect of the neutral alkali salts in the beginning is mainly restricted to the surface and cannot extend far beyond the superficial envelope of the fibers. The weight of a muscle as a measure of the dispersity of its structural colloids is not to be expected to change considerably in an isotonic solution, and further should show but slight variations on applying, for instance, various anions. As a matter of fact, comparing the percentage increase of weight of sartorius muscles, gold-laced, for 2 hours in a buffered ringer solution, AU in corresponding solutions with ADR, nanos, and NAEG. The ratios for the percentage increases referred to NaCl equals 1 are found for Nabrier equals NaNOA equals 1.5, ANAG equals 1.8. The process of colloid dispersion in general is dual in its nature. It is either a matter of hydration and dehydration of colloidal micelles, or a matter of increase and decrease of their electrical charge, see pages 299 FF. It depends upon quite a number of factors which of these processes is dominant under various conditions. Certainly one of the main factors is the chemical nature of the colloids. But the information about the chemical components of the plasma surface membranes is very poor as yet. The most satisfactory approach to a better knowledge has been found in the analysis of the stromata ghosts of the erythrocytes, which are accessible in great quantity as a relatively uniontaminable material. 2/3 to 3/4 of the lipoids are phospholipoids, the rest cholesterol. Search-prosides are not present in the red cells. 3/4 of the phospholipoids are made up by cephalin, 1/4 by lecithin. Lecithin is an ampholyte due to the presence in the molecule of glycerophosphoric acid on one side, of choline on the other. Cephalin has an acidic character since choline is a weaker base than choline. The protein is more or less insoluble. This seems to be due partially to the formation of lipoproteins, CP297. One such lipoprotein is a reaction product of the acidic cephalin and the basic histone, and globin also has been found to precipitate with cephalin. Products like these, may account for the formation of relatively solid cell structures. Permeability of plant cells, R. Collender, Protoplasma 33214, 1939b, and other biological material. The facts concerning the main components of the protoplasmic surface lead to the suggestion of how the building stones are composed to form its molecular architecture, which has to serve the various demands of cell life. This has been partially discussed in the preceding SHE4, Chap 15. Here, the main problem is how ions are enabled to impose on this structure an increase or decrease in dispicity as it is found with colloidal sols and gels, which are known among proteins, lipoids, and lipoproteins. Dehydration of protein of lipoids and of lipoproteins. In general, fly molecules of proteins or polypeptides are conceived as being shorter or longer chains of amino acids attached to each other by the peptide linkage between the Cu and the NH2 groups. Correspondingly, their molecular dimension ranges from about 5 units up to about 5,000, A equals 0.5 micron. The chain linkage favors a fibrous shape of the molecules, but, as will be seen later. 297. It also does not conflict with the shape of leaflets or with that of more rounded particles due to folding and coiling of the chains. Each moderately long peptide chain resembles a tridimensional rod-like body with a zigzag axis composed of the repeating NCC groups due to the Witterian character of the amino acids. To each of these chains, there is attached at a regular distance of about 3.5 A an amino acid residue as a side chain projecting perpendicularly to the axis and alternately in object excite directions, as indicated in the following formula of a tetrapeptide tyrosylcystenyl volaglian. It is obvious that the length of the side chains is variable, thus determining the width of the rod or fibril. These rods can be identified as such by various optical methods, mainly and by measuring the space of the various planes in an X-ray diffraction pattern, as one does in a crystal lattice. The nature of birefringence is dual. It is either a form birefringence or an intrinsic birefringence. The form birefringence is based upon the presence of rods as symmetric units of an isotropic substance, embedded into an isotropic medium, and upon their more or less regular parallel orientation, whereas the intrinsic birefringence is caused by optically anisotropic crystal-like particles. By either of these methods, changes in the mutual distance and orientation of the submicroscopic units can be observed and measured quantitatively. Even with single ghosts of erythrocytes, fibrous structures have been detected by the birefringence technique, whereas the layers formed by their cellular envelopes are too thin to allow the detection of X-ray diffraction spacings. Fibers, of course, are constructed of several peptide cleanes arranged with their axes parallel to each other and parallel to the main axis of the single fibrils. It is now of particular interest for our problem concerning hydration and dehydration to understand what kind of forces are involved in binding together the individual peptide chains, see Sec 2, Chap 9. The affinity to water of the single polypeptide chain depends mainly upon the water affinity of the individual side chains. For instance, side chains which are composed only of hydrocarbon groups, like the valine residue in the aforementioned formula, have rather no affinity to water, while the NNO-containing radicals, NH2, NH, CUDI, and O are hydrophilic. This is due to the fact that in the NNO atoms of the C radicals, residual lone electrons are available to be shared with residual electrons of adjacent water dipoles, and thus to effectuate a coordination linkage with water. In this way, it is understandable that the hydrophilic or hydrophobic character of the polypeptide chains, among others, is bound up first with the chain length insofar as this decides the ratio of hydrophilic polar and hydrophobic nonpolar groups. The second factor is the acidic or the basic character of the terminating groups, Cui or Nil2, which, depending on the gene, can form electrically charged centers of coordination with water dipoles. With regard to the fiber proteins, which are constructed by linking together several single peptide chains, additional factors contribute to the hydration. First, there is evidence that fibrils with short side chains can approach closer together and thus form fibers with a tighter packing, for example, keratin, than those with longer chains. This certainly is inhibitory to the access of W water molecules. Second, the pH again is an important point, with a pH fairly distant from the isoelectric point. The multivalent ampholyte proteins can be assumed to contain a number of either cationic or anionic centers, which by their electrical charge will repel each other and thus favor the intrusion of watery order. Inversely, at the isoelectric point, parts W.J. Schmidt, Die Hausteine des Tierkörpers im polarisierten Hecht, Bonn, 1924. Die Doppelbrechung von Karyoplasina, Cytoplasina und Metaplasma, Berlin, 1937. F.O. Schmidt, J. Applied Physics, 109, 1937. F.O. Schmidt, Phys. Reverend 19, 270. 1939, F.O. Schmidt and R.S. Baer, Bio. Reverend 14, 27, 1939. Afrey Weisling, Submicroscopische Morphologie des Protoplasmas und seiner Derivate, Berlin, 1939. L.E. Rarpicken, Bio Reverend, 16, 133, 1940. Further, Sear, 2 chap, 7 of the structure opposite to each other may be oppositely charged, and thus, like zwitterions, become linked together by electrostatic forces. Indeed, protein structures ordinarily are found to be more inaccessible to water at the isoelectric point. A third, there have not yet been mentioned the special cross-linkages between individual fibrils, contributing considerably to the solidity of the fibril bundles, as, for instance, the disulfide linkage between 2 cysteine groups, forming a primary valence bond requiring chemical forces for disruption, or salt linkages formed by the reaction between the acidic and basic groups. Taking these various factors together, it is obvious that, in general, an increase of hydration will be indicated by a decrease of the birefringence and by an increase of the spacings in the X-ray diagram, and vice versa. It has Ian mentioned P295 previously that the polypeptide chain is deemed to construct not only the fiber proteins, but also leaflets and other aggregates of manifold configurations. From various kinds of measurements, viscosity, ultramicroscopy, ultracentrifuging, it has been concluded that some colloidal micelles are spherical or ovoid in shape. This is considered to be due to folding and coiling of the polypeptide chains and to holding together such a feltwork by 2 of various aforementioned forces of interlinkage of greater or smaller strength. This affixure accounts for the possibility of different degrees of hydration and dehydration, corresponding to the ratio between polar and non-polar radicals, and suggests thinking of these particles as forming a meshwork filled with more or less loosely attached water, as encountered in gels. Such aggregates often are highly soluble, for example, egg albumin particles, probably because the polar groups are sticking out of the surface toward the water. This concept is corroborated. The surface of water is accompanied by denaturation, probably due to an unrolling of the long chains attended by the rupture of interlinkages in the framework, the exposure of more hydrophobic groups, and the breakdown of SCS cross-linkages, making C groups detectable, and other results. Wide hydration and dehydration of lipoids and lipoproteins. By chemical analysis of the red cell ghosts, it has been shown, pp. 272 and 294, that about 1/3 of this structure is formed by lipoids and lipoproteins, and that the lipoids are partially phospholipids, partially cholesterol. Cholesterol possibly does not participate in functional changes of this and of other cell surface layers, or only to a small degree, because it is a complex ring system of C and H, which, in the case of cholesterol itself, includes but one OH. However, this O may convey to the molecule a weak polarity, as displayed in the CLA-Schmidt chemistry of the amino acids and proteins. C.A. Thomas, Baltimore, 1038, W. Pauli and K. Volk 6, Kolloidschene der Kieweißkörper, Dresden, 1933. Capacity of sterols to form surface films on an air-water interface, and therefore cholesterol might be assumed to play a role as a fairly rigid framework for the more labile hydrophilic colloids, which are included in the cell membranes, CPL. Strues and Sec4. The reactivity of the phospholipoids is much greater, lecithin being an amphiolite, which is ionized to some extent at a pH around 7, and cephalin exhibiting a more definite acidic character. In order to gain more direct information about the structural capacities of the lipoids, it has been found useful to investigate mixed lipoids, for example. Benzene alcohol extracts of the brain instead of studying the pure chemical individuals. If emulsified with water, X-ray diffraction measurements show the mixed lipoids to form molecular layers, each of the two arrays of molecules turning their polar, positive and negative groups outward and being attached to each other by the nonpolar endings of the fatty acid radicals. See figure 27, P274. Between each pair of such bimolecular layers, 65 in thickness, there is interposed a layer of water, the thickness of which is indicated by the X-ray diffraction. SGI acings to vary according to the percentage of total water present in the emulsion, 0% to 75%. Thus, the polar groups of the lipoid phase are anchored in the intercalated water layer, the thickness of this layer varying from 0 to 85 AA. A still better model is provided by a mixture of lipoids and proteins which are bound to each other, as in the aforementioned L-lipoproteins, arising from a reaction between a basic histone and acidic cephalin, P294. The resulting structure resembles the architecture of the emulsified mixed lipoids, but differs by the interposition between the lipoid layers of a monolayer of histone. The polar phosphoric acid graues of cephalin being linked together with the basic groups of the highly hydrated histone, with such strength that the water is expelled by this interaction. The molecular organization of natural lipoid protein surfaces also has been subjected to an analysis, most thoroughly by combining both the measurements of X-ray diffraction and of intrinsic, as well as form double refraction in the study of the sheath of myelinated nerves. Several kinds of investigation have suggested again the interpretation of the findings as due to lipoid protein layers, which are concentrically arranged around the axon, each of them being 170 to 190 A in thickness and composed of two double molecular layers of lipoids, which are separated by a thin protein layer. Between these concentric layers, a great amount of water is interposed. A similar structure has been ascribed to the envelope of the red cells. As mentioned previously, P294, this is marked by a high content of cephalin. 6. The influence of inorganic alkali sulfate upon hydration and dehydration of cell colloids. Returning now to the main problem of this section regarding the nature of the ionic influence upon cell activity, we must determine whether the present knowledge about the architecture of the artificial and natural complexes of the colloidal constituents of cells contributes to an understanding of this influence. The basis of such a study are the aforementioned observations of Hofmeister regarding the influence of neutral alkali and of alkaline earth salts upon the hydration and dehydration of hydrophilic colloids like gelatin and albumin. Two factors are preponderant in the interrelation between ion and colloid. First, the amount of hydration of ion and colloid, or salt and colloid P300. And second, the dehydration of the ions increases in the order CNJO, JO, EBGL, so forth, and RB not li. For the halide anions and for the cations, this is the order of decanasing atomic radii, and thus probably the order of increasing electrostatic interaction of ion and water dipole. For instance, the relative hydration of cations is estimated. 2 equals K equals 544 around the ion as a shell, the strength of fixation diminishing toward the periphery, where the water dipoles can be stripped off with increasing ease. The hydration of the colloids varies over a wide range. This, in general, is DIE to the number and strength of residual valences as hydration centers, as explained before. 296. And thus, the range can reach from the highest grade of hydrophilic behavior to that of the hydrophobic or suspensoid colloids. The interaction between the colloids and the ions may be conceived as follows. In general, the hydrophilic colloids, particularly those which we are mainly interested in, the proteins, are colloid electrolytes, the colloidal particles being either cationic or anionic. As such, they attract the ions bearing the opposite electric charge and repel those with like charge. The strength of the charge of the individual colloid varies with reference to the isoelectric point of the colloid. Another factor is the adsorption affinity of the colloids toward the ions, cations, as well as anions. The adsorption affinity toward the ions differs, increasing with decreasing hydration, and vice versa. In other words, the shell of water dipoles surrounding the atoms interferes with their attachment to the colloid. For this reason, the affinity toward K is greater than that toward Li, toward SDGN, and I greater than toward Cl and so forth. Further, it must be realized that, as another significant feature of adsorption, with lower salt concentrations, a relatively greater fraction is attached to the colloid than with higher concentrations. A third factor is the competition for water between the colloids and the outside ions surrounding the colloid particles according to their respective water affinity, including their osmotic influence. The operation of this factor in the case of non-electrolytes instead of colloids is well known as the salting out effect of electrolytes at Lager concentrations. It is especially strong if the water affinity of both cations and anions is great. The dehydrating influence toward the colloids is attributable to all ions bearing the same charge as the colloid particles, and in addition to that fraction of the oppositely charged ions, which remains unadsorbed. A fourth factor seems to be the ion pair effect, that is the joint action of cation plus anion, which form more or less distinctly either aquacids or aquabases, as indicated by figure 29. Representing the formation of an aquacid dipoles, then the larger I and orients the dipoles in such a way that O is firmly fixed on Li, so that H projects as a free ion. Aquacids are supposed to react more with basic, aquabases with acidic proteins. It is obvious that the combined effect of electric charge, of adsorption, of ionic concentration, and of hydration can bring about most variable and sometimes embarrassing results, and as a matter of fact, the order of cations, or that of anions, or that of both, has been found to run in one or the other direction, or even to turn into a disorderly succession, according to the salt concentration. To the EH further, to the admixture of other colloids or of non-electrolytes like sucrose or alcohol, which themselves have a strong dehydrating influence. For instance, a mere change of salt concentration can cause a reversal of the order of cations or anions in the following manner, Kiel, at low concentrations due to the relatively strong adsorption of K, affects a negative colloid directly by a considerable discharge compared to the less adsorbable Li. But at high concentrations, the effect of Li is more prominent due to its strong dehydrating power, affecting more indirectly from outside the water attached to the colloid, or by changing the sign of the electric charge of a colloid amphilite. By modifying the 7-decidian, a complete inversion of the ionic order is established. With negative particles, the dispersity is diminished by cations essentially due to adsorption. In the order K now Li, and by anions essentially because of dehydration from the outside phase in the order, so for ONCL LBREEI as here. Whereas with positive particles, the opposite order, CN alberkiel so forth, and like Nekea, occurs, but regarding the accomplishment of these inversions of the ionic order, it can be logically expected that by stepwise changing of the ionic atmosphere around the colloidal micelles, one may encounter highly irregular intermediate arrangements, which by no means can be referred to XESE or mental error if carefully checked, especially with regard to the J. As a matter of fact, there are scattered in the literature about the influence of the neutral alkali salts upon the colloid state of lipoids and lipoprotein complexes. Very little is known as yet. Again, there has been observed the ordinary cation and anion series displaying the gradually changing effect upon flocculation or clarification, increase of birefringence, and decrease of X-ray diffraction spacings in mixed brain. Lipoids also were found to be indicative of a rising dehydration after the addition of certain neutral salts in certain concentrations. From the analysis of the architecture of the colloid complex is attempted here, pages 295 to 290. 99. arises a picture of aggregates of many different types of colloids cemented together like stones in a mosaic or disposed in layers, one upon the other. The component colloids in these artificial systems, as well as in natural fragments of protoplasm, are of varying proportions of HEG hydrophilic and hydrophobic types. Therefore, their isoelectric points will be well separated, and the electrical charges will differ from point to point, creating consequently anomalous effects of the neutral salts. Even greater complexities are to be expected in living material than in these models. Such K3 Postamac, Annonst Pasteur, 1685-1901 W. Pauli, Beatrice, Chem, Physio, U. Path, Rugers, Arch, F.D., Theseus. Physio, 186-112-1921. Eschdoken, L. Michaelis, I. Allleitcher, 37, 203, 1925. J.R. Katz, Biochem, Theesti Artiana, 267, 261, 262, 263, 1932. See further H. Ott Freundlich, Alloy and Capillary Chemistry, Dutton, New York, 1922. W. Pauli and D. Velksix, Elektrochemie der Colloide, Wien. Considerations as these are chiefly of importance as stimulus and guide for future studies. C. The interpretation of physiological effects of ions as due to changes of the state of the hydrophilic colloids. It is almost surprising that in the beginning of this chapter, some relatively simple observations concerning the change of muscle excitability by inorganic alkali salts could be made to suggest a colloid theory of stimulation based upon the appearance and muscle excitation of high allotropic order of anions and cations, well known from the physical chemistry of hydrophilic colloids. Such a theory appeared to be highly plausible for several reasons. 1. Each protoplast is like a gel or a sol of hydrophilic colloids. 2. An important part of the protoplast, the plasma membrane, is built up of various colloids to a structure, which often, as in muscle fibers, appears to be fairly impermeable to many solutes, but which can lose this property under the reversible influence of alkali salt solutions. Such solutions are known to have a dispersing and eventually a liquefying power toward organic hydrophilic gels. 3. The dispersing effect of, for example, NaClO can be balanced by the addition of a small amount of alkaline earth, salt in the case of physiological objects as well as colloidal models, however. Many objections have been made to such a colloid theory, as can easily be observed in reviewing the physiological literature. It is true, the order of anions, so for EGL, BREINCN, and of alkalications, LINAK, is met frequently in animal and plant physiology, but it is likewise true that irregularities, either as inversions of the regular arrangement or as complete disorder, have often been encountered, to mention only one old example. Hemolysis has been studied by suspending erythrocytes in the isosmotic solutions of the neutral alkali, not K salts, and adding alcohol, urethane, saponin, vibrio, or applying heat to the suspension, or comparing erythrocytes, the natural cationic contents of which differ considerably, regarding the influence of the anions and the cations upon the order of the hemolytic effect. Very great variations are displayed. The ordinary order of anions and of cations running in either the usual or the inverse direction, or being displaced by a random arrangement. This result can be thought of as being due to the presence of various colloids or colloidal complexes, and to their combinations with the added hemolytic factor, superimposed on the effect of the inorganic salts. But from these and similar findings, the conclusion should not be drawn that the colloid theory is to be rejected as a reasonable explanation of ionic action. On the contrary, all the variations in cellular response to the salt should be analyzed from the viewpoint of colloidal behavior, and are likely to be correlated to colloidal processes, d. the antagonic action of pairs of cations. So far, the effects of single salts only have been taken into consideration. An especially strong superior of the colloid theory of J physiological ion activity comes from the study of EUG airs of salts issuing from the aforementioned physiological antagonisms P291, discovered by J. Lowe. Lai suggested this antagonism to be a physiological illustration of the Schulze-Hardy rule that the flocculate power of an electrolyte depends upon the valence of the ion whose charge is opposite to that of the colloidal particles, and that the influence of valence rises nearly in a geometrical progression. The Schulze-Hardy rule is derived chiefly from observations with the inorganic supersensoid and, in general, hydrophobic colloids. Here, the preponderant unstabilizing factor is the electric charge. Thus, according to their valence, the cations in appropriate concentration can bring about an isoelectric precipitation of an anionic colloid, the anions of a cation. However, hydrohalic colloids, which in general are the prevailing fraction of the colloids in protoplasm, are not subject to the Schulze-Hardy rule, except with certain modifications. By gradually substituting the water in a solution or in a gel of a hydrophilic colloid such as gelatin with polar hydrophilic alcohol or acetone, the hydrophilic colloid is changed into a hydrophilic suspensoid. In the hydrophilic state, high concentrations of salt are required to produce precipitation by dehydration, salting out effect, P300. However, after a certain extraction of water has been accomplished, the colloidal particles attain more and more the character of hydrophobic colloids, which are precipitated by small electrolyte concentrations, following the Schulze-Hardy rule, as indicated by the greater influence of the polyvalent ions. Thus, the precipitating power of both NH4 and Ki-2 toward gelatin appears to be low, although in agreement with the hydration of their ions. But after transformation into a hydrophobic suspensoid, the effect is great with NH4 on the acid side, of the isoelectric point of the colloid with Kachi on the alkaline side. Further, a colloid, which in the hydrophilic state differentiates between the individual monovalent alkali cations and the monovalent halide anions DF ending upon their hydration. As a hydrophobic colloid reacts more or less uniformly to the monovalent or the polyvalent ions, but differentiates among them according to the electric charge. However, even in the hydrophobic state, the colloid can respond somewhat differently toward equally charged ions. For instance, the precipitate ENG power of bivalent cations toward negative suspensoids, like asphosate, increases generally in the order mg/bHe-PB.C8. This is about the order of the standard electropotentials of these metals, and insofar probably the order of their decreasing hydration. Now, as mentioned previously, page 91, phenomena similar to these have been met by J. Loeb in his experiments on the development of fertilized eggs of the marine Teleus funvulus under the influence of various salt mixtures. The development is stopped, and the embryos die in isotonic alkali chloride solution. But highly deleterious effect can be antagonized by the addition of certain small amounts of divalent cations. Most favorable is the effect of the alkaline earth ions. Next are Co and Man. Much more toxic are Ni and Pb in similar experiments concerning the liquefying influence of NaCl upon the cilia of marine invertebrates. The bivalent cations have been found to protect the cilia in the order MGBA, NFA, CD. PBZN, Kiu, Igoto. The last members in this series show the favorable effect only if applied in small concentration and for a short period of time. Otherwise, irreversible impairment occurs, evidenced by a beginning opalescence and flocculation of the protoplasm, NCl ending with death. Obviously, this series strikingly resembles the aforementioned order of the standard electropotentials. The results strongly support the hypothesis that a normal funcluion requires the existence of a certain colloidal state, and this necessitates a certain balance of mono and polyvalent cations. If only monovalent cations are present, the dispersity of the structural colloids is abnormally great. The proper amount of bivalent cations ensures the necessary rigidity. Tillies's concept is strengthened by the fact that trivalent cations, all CRFE, also show an antagonistic effect, but only when applied in concentrations considerably smaller than those most favorable with bivalent ions. These observations are in agreement with the Schulze-Hardy rule, which states that the flocculation power of the ions rises with rising valence, nearly in a geometrical progression. The order of bivalent cations, practically identical with the order of the standard electrode potentials, can possibly be interpreted physiologically as an order of varying hydration capacity. Following the addition of KCl2 to the gel of a hydrophilic colloid, dehydration, in other words, shrinking or stiffening, occurs, indicating the competition for water by the highly hydrated Chia P299, but see further P305. Following the addition of Ni or Cu, flocculation inside the gel occurs due to the adsorption to and the discharge of the colloid aggregates P299. Shrinking is a reversible flocculation, an irreversible process. Thus again, the physiological effects parallel the physicochemical reactions. See also the foot, our main example. The influence of ions on muscle activity lends itself particularly to the correlation of the effect of mixtures of ions on muscle excitability and on hydrophilic colloids. After increasing the colloid dispersity in frog muscles by the addition of small amounts of KTL to Ringer's solution so as to inhibit contractility completely, restoration takes place following the addition of appropriate amounts of bivalent cations in the series KASRE notito, CD2Q, however, are incapable of restoring function. The gelation of a gelatin solution can be accelerated beyond that in the presence of NaCl by the addition of one of the following chlorides in proper amounts, SRDMG. The gelation is slowed by CdO2 and CO3. The complex cobalt and chromium salts furnish an excellent example of the importance of the Schulze-Hardy rule in physiology. Only those complex cations which are bi- or trivalent are capable of antagonizing the paralyzing effect of a small excess of XTL in Ringer, whereas the monovalent complex cations fail to do so. The bi- and trivalent complex cations are comparable in the degree and duration of this beneficial action only to Ta and Senyer, whereas the simple trivalent cations of the rare earths, Ce, Al, Y in much smaller concentrations are strongly toxic. According to Mines, hexamen cobalt salt is rather indifferent toward hydrophilic colloids like hemoglobin, egg albumin, gelatin, and is thus again similar to Siat. All this is probably due to the special configuration of these complex cations, the metallic nucleus of which is surrounded by a SGD of molecules or radicals, resembling the sphere of water dipoles around alkali or alkaline or alkaline earth ions like or Ka, and therefore probably less adsorbable and less capable of discharging the colloidal aggregates than the simple cations 299. It has been emphasized already, Pete 290, that the alkaline earth ions and especially Ka are often much more powerful in balancing the unfavorable dispersing influence of the monovalent cations than are the other polyvalent cations. This seems to be due to the fact that upon their stabilizing, anti-dispersing action, there is superimposed a chemical reaction which links together more firmly the colloidal units than could be done solely by the presence of alkali ions. The conceit here of a solidifying influence of Chi is suggested by innumerable observations, particularly upon tissues, the cellular building stones of which are loosened by a lack of alkaline earth salt. For example, the blastomeres of sea urchin eggs, the cells of root hairs, the epithelium of mucous membranes, and others. The reason for this is that the cells normally are packed together by a cementing substance with the character of a weak acid, forming rather insoluble Ka salts, which, in the absence of Chi in the bathing salt solution, dissociate and are transformed into the softer and less resistant Nal salt. Such compounds are the pectinates, the soaps, the phospholipoids, some proteinats, and others. These reactions taking place in the RDR color space and in the cell surfaces can be pictured by the following equation. assuming that in this manner a chemical reaction is superimposed upon a physical chemical reaction. The gradually differing effects of the alkaline earth and the other bivalent cations can be read as dependent upon weaker or stronger, upon more reversible or more irreversible reactions of these ions with the cementing substrata. This concept is supported by the following observations. According to Locke and Overton, a nerve muscle preparation loses its indirect excitability after being transferred from Ringer into Nakel and regains its activity after the addition of Ti. Cia can be substituted only by Sr but not by Bom-G-Coman the HCCA. However, the direct excitability after being abolished by NaCl 0.05% TL is restored after the addition of any one of these bivalent cations. A similar behavior has been found with the heart and the stomach of the frog. Their contractility, as manifested either by spontaneous pulsation or as the response to an electric shock, is lost in NaCl and repairs only after the addition of Chi. But if paralyzed in Ringer plus 0.75% KCl, the heart recovers also with Kio-Men, the stomach with Men, and hexamen cobalti ion. Locke has accounted for his observations regarding the loss of the indirect excitability in the absence of Chi as indicating an interruption in the pathway of the impulses at the myoneural junction. In other words, normal conduction requires a cement between nerve and muscle tissue which can be loosened or tightened reversi bly, but must possess some intermediate degree of stiffness in order to allow the conduction wave to travel across the synapse. In addition, there is needed a certain consistency of the structural colloids of each separate tissue, which after being abnormally softened by a surplus of K can be brought back to its normal state, not only by Ca or SNR, but in accordance with the Schulz-Hardy rule more or less by the adsorption of any bivalent cation. These different kinds of linkage between the colloidal aggregates, a physicochemical linkage on one hand, a more chemical one on the other, are manifested not only in a concatenation, as that on synapses, but also in many other cases. One particularly illuminating example is the behavior of the Funvulus and Brua in LLB's experiments. The predominant influence of valence is displayed only while the embryos are developing inside the egg membrane. This latter is a dead structure, probably keratin-like, and as such tough and rather impermeable to water. But a pure Nakel solution renders this membrane permeable, and it is this loosening effect which is antagonized rather indiscriminately by bivalent cations, as stated by Loeb. However, the embryo liberated from the cover can be protected against the injuring effect of pure Nakel only by, and the rather closely related SRNG, whereas even if A is useless as a substitute for Ca. Thus, it becomes understandable why the combination of the cations Na, K, and Chia mostly satisfies the wants of active protoplasm, ensuring its physico-3 effects of substituting non-electrolytes for electrolytes. It was mentioned previously, p. 289, that frog muscle soaked in isotonic sucrose solution, after a short time, about 15 min, loses excitability, but regains it after immersion in Ringer or any iodoxic solution containing Na ions. Chemical analyses by Urinol and by Fair showed that during the paralyzing process, the muscles, if carefully prepared and kept at low temperature, lose considerable amounts of Nakel, up to 90%, by SKE from the interfibral space, but of K, though present in large quantities inside the fibers, as little as 6%, P253. Further, it was shown that phosphate and lactate appear in the sugar solution, and that these losses, which indicate a rise of permeability, are accompanied by a strong increase of 2 consumption. In addition, a slight contracture is often observed. According to Overton, similar effects are brought about by the isotonic solutions of other lipoid-insoluble and chemically rather indifferent substances like lactose, maltose, glucose, mannitol, alanine, and asparagine, which obviously by no means are physically as harmless as originally assumed. These striking effects of the isotonic non-electrolyte solutions can be counteracted by adding small amounts of salt. 0.7% NaCl is enough to preserve the normal state. According to phenyloxyether, they're abnormal with respect to the physiological effects of the bivalent metal ions. It should be mentioned that the activating properties of MFCO possibly can be correlated to some extent with their activating or reactivating power towards certain enzymes, particularly toward those concerned with oxidation reduction processes. See Sec 6 chaps 27 and 28. Also LF. Hellerman, Physiol, Reverend 17, 454, 1937. Further, it should be borne in mind that amino acids and proteins are known to form stable complexes W with certain heavy metals. See Carl L.A. Schmidt, The Chemistry of the Amino Acids and Proteins, Thomas, Springfield, 1938. 2. Consumption and phosphate loss are considerably reduced also by the addition of COC or CLL in low concentration, the latter being known to paralyze in higher concentration and to provoke contraction. Even HCl1 and 0.026 mol/pore produces a rapid fall of 2 uptake after sucrose treatment. In order to find an interpretation of these effects upon muscle, it seems to be useful to compare the behavior of erythrocytes under similar conditions. As previously described by Jacobs and Parpart, the osmotic resistance of the red cells of ox, as measured in equally hypotonic solutions, is greater in non-electrolyte than in salt solution. Furthermore, the non-electrolyte solutions, either hypotonic or isotonic, cause an increase of osmotic resistance, which occurs in 2 stages. One rapid, which is readily reversible, and one slower, which succeeds and can extend OVT a long period of time. The rapid process is likely to be due to an anionic exchange through the anion permeable surface membrane, comparable to the Prouvaire-Slay P254 described exchange of K impermeable collagen membrane, by which, beginning with equality of Kj and Co and with high Ho K, is shifted from outside to inside against an ever-increasing concentration gradient, while H moves in the opposite direction toward a diffusion equilibrium in a corresponding manner. The plasma membrane of erythrocytes transferred into a non-electrolyte solution is past the valent amount of O, with the result of an increased ionization of hemoglobin, that is, the formation of polyvalent hemoglobin. In this way, the osmotic pressure within the erythrocytes is decreased, and thus the osmotic resistance increased. The slow stage of the increase in non-electrolyte solution is believed by Jacobs and Parpart to be due to an irreversible loss of electrolytes from the interior of the cells, that is, not only to an escape of anions, because of the normal anion permeability, but also the pathological penetration of cations, mainly from this erythrocyte of muscle. And red cells that are that a common feature of the non-electrolyte effects is a leakage of salts. The nature of this leakage will be discussed immediately, but exchange of ions likewise plays a role in either case. With erythrocytes, due to their anion permeability, the considerable diminution of ion concentration outside is succeeded permeability. An increase of inside acidity occurs. For it previously has been shown that the surface of muscle fibers resembles the collagen membrane in the aforementioned experiment of Netter. And since, according to the theory of membrane equilibria, Ketico equals high Ho, 254, the diminution of Co should be followed by an increase of high, and vice versa. As a matter of fact, numerous experimental results failed to give unequivocal support to this theory. However, by changing Co over Ho artificially or by trying to influence high through metabolic stimuli, anoxia, or metabolic poisons, iodoacetate, so many secondary metabolic reactions are released: production of lactic acid, breakdown of phosphocreatine, dehydrogenations, and others, that even a fairly near approximation to the expected behavior may have a confirmatory value for the theory of exchange of K and H in muscle. Check now. As to the destruction by non-electrolyte solutions of the normal impermeability to salts, The following hypothesis has been advanced by Daniel L. If a long-chain fatty acid is dissolved in an organic solvent, brombenzene, which is immiscible with water and which is in contact with an aqueous buffer solution of alkaline reaction, neophosphate or borate, the fatty acid is ionized and a film of soap is formed at the interface, with the long chain end of the anions adherent to the organic solvent, and with their hydrated carboxyl groups on the other end held in the buffer solution. The interfacial tension is lowered over a pH range of 5 to 10, the tension being lower with the more alkaline reaction as shown in Figure 30. The tension pH curves have the same shape as the dissociation curves for the soap in the bulk solution, only differing in that the range of pH over which the change of the interfacial tension occurs is shifted about 3 units of pH toward the alkaline side. Its shift is thought to be indicative of a Donan equilibrium at the interface, the soap anions being retained in the film, the free buffer anions being predominant in the bulk of the aqueous phase, the cations, including H, in the film phase. In other words, the reaction at the interface can be considerably more E than that in the bulk of the solution. This conclusion is corroborated, among others, by observations upon the influence of ion concentration in the buffer solution. As shown in Figure 30, the displacement of the curves toward higher pAs is greater with 0.001 mol buffer than with 0.4 mol. But adding neutral salt, 0.4. Now, Figure 31 abolishes the shift, which is brought about by dilution of the buffer. This influence conforms to the usual effect of ion concentration upon the Donan distribution, see Sec 1, Chap. According to Daniel L., these observations serve to shed light on the injurious influence upon cells of substituting non-electrolytes for the electrolytes normally present in their environment. Various effects must be dealt with. As far as surface membranes of protoplasts are concerned, which are believed to be built up by organic solvents like lipoids, by proteins and enzymes, the lateral adhesion forces between the structural components of the membranes can be assumed to be affected by the change of 3 and more pH units in these membranes. The hydration of lipoids will be changed. The proteins can be subject to irreversible denaturation, the enzymes to inactivation, and these disintegrating effects will be succeeded by internal alterations of metabolism, by escape of large amounts of K and of possibly sugars, as the chiefly applied non-electrolytes may produce the functional impairment not only as osmotic substituents, but also by independent direct effect. It has been known for a long time that a certain solidification is induced by sucrose in the plasma membrane of plant cells, preventing, for instance, the separate fragments of the protoplast, which can be formed under the influence of plasmolysis by a hypertonic sucrose solution from coalescing again, and causing during deplasmolysis a local rupture of the rigid membrane. This solidification may be compared to enhancement of gel formation and of shrinking of gelatin gels by sucrose and glucose. Owing to their dehydration power, pages 801 and 303, the NCI identically observed smaller effect of mannitol compared to sugar upon the surface of red cells, has been referred to the absence of the aldehyde group, which is thought to react with protein, E. Custer, Bertie, Astue, Boite, Gist, 27, 589, 1909. In the 16th chapter, ions were shown to condition a greater or smaller activity of muscles and nerves, or, better, to raise or to diminish their excitability, a normal degree of excitability being met only in the presence of a certain mixture of ions. Another kind of ionic influence becomes obvious in the ability of muscle and nerve to respond to a local change of the ionic medium by disclosing a potential difference or arresting current comparable to the action current which follows excitation. Muscle and nerve are especially suited for study of these electrical phenomena because of their fibrous structure. There is much less knowledge about i.e. electrical properties of single cells, which, as more or less spherical units of small dimensions, arc, in general, less suitable for attaching electrodes. The resting potentials, which are produced externally by altering the ordinary ionic atmosphere, require particular attention because by such ionic effects, light is thrown on the nature of the action potentials. The likeness of which to the artificial resting potentials is often deniquated or doubted. 1. The salt potentials of resting muscle and nerve. If one end of an uninjured frog sartorius muscle is brought into contact with ringer, and the other end with ringer, the nacle of which is replaced by another alkali or alkaline earth chloride, and if the two solutions are connected by suitable electrodes in a potentiometric setup, an injuring current develops, the strength and the direction of which depend upon the cations replacing anoat. The cations appear to follow the order. K, Q, R, B, I, C, C, N, L, E, K producing the strongest negative pole, via weak positivity, compared to NOT. This series has been met already, p-pound 90, as the order of rising excitability after a short duration of exposure or after exposure in a fairly low concentration. However, after treatment for a longer period or by a greater concentration, the increase of excitability as exhibited with K or RB changes to a decrease, leaving the electronegativity unchanged, CP-pound 80. The order of anions is CNGINO3, BRHCl, with CN producing the greatest positivity, which, however, after exposure for a long time or to a relatively high concentration, reverses to negativity. The same is true to a smaller extent with II. The excitability under corresponding conditions likewise changes from increase to decrease with SCN and IP290. In general, the effect of cations is GRCator than that of anions. Nerves are similar to muscles, with one lead on the intact surface of an excised nerve, the other lead taken from one of the crushed ends, dipping into strong KLL and serving as reference electrode. The potential difference is depressed by isotonic alkali chlorides in the order KK, HBTs, and Ali with frog nerve in the order KBK and Ali with the unmyelinated nerve of the spider crab. Shown the effect of the inorganic anions applied as NA salts has been found to be nil or very small, so that it remains questionable whether these effects are significant of the proteolytics of the nerve membranes or are rather indicative of diffusion potentials. Measurements on single nerve fibers can be expected to make this clear. Another interrelation between excitability and resting potential of muscle is encountered in experience on the antagonistic effect of monoval and polyvalent cations. 304. It appears that the injuring influence of pure isotonic nacle, or NAD plus small amounts of KCl, is balanced by suitable small amounts of KSNR liacum, but fails to be abolished by QeO2 and CD. Furthermore, the trivalent cobalt complex salts, for example, GCO, NN3,6, exhibit a balancing effect, while the monovalent, for example, EK-UN-E2-ETL, do not. Nerves are subject to the same rule. After nerve or muscle has been submitted to isotonic alkaline earth chloride, even KCl, at a strength as high as fully isotonic, is prevented from, or at least delayed in, J-revoking negativity. It is protective not only against KTL, but also against various chemicals, which for one reason or another, sexy later, P338. For example, amylourethane, saponin, ferritrine, salicylate. These latter experiments have been done with the nervidae of the marine spider crab. Taking into account these various observations on the salt potentials, and further referring to the aforementioned effects on excitability of muscle and nerve, it is suggestive to interpret the bioelectric phenomena as surface effects taking place in the plasma membrane. Such an idea was presented for the first time 50 years ago by W. Ostwald in pointing out that precipitation membranes are the seat of considerable electromotive forces that may be thought of as DM to selective ion PQ permeability. This view was subjugated by the just mentioned later observations, according to which the bioelectric potentials are influenced by inorganic ions in such a way as to recall two of the most familiar rules of ionic effects on colloids, the lyotropic series and the Schultz's RD rule. The ionic effects on the potential suggest the presence of a colloidal membrane, which shows both hydrophilic and hydrophobic attributes. The hydrophilic character, on the one hand, for instance, is illustrated by the series of the monovalent cations Li-Na, Cs, Rb, K, which with its characteristic location of else has been met in model experiments only with hydrophilic colloids, 294, and in addition has been encountered frequently in physiology, P293, while, on the other hand, the membrane resembles the hydrophobic colloids in the applicability of the valence rule. Finally, a colloidal character is suggested by the fact that in many of the aforementioned XEEs elements, the physiological activity of the cations has been found to surpass that of the anions, presumably because the colloidal components of the cells generally carry a negative charge. 2. The Mie-Brie theory of ryoepithelials, arguing in this way, the colloid theory of the bioelectric potentials apterars as a natural complement of the rather commonly accepted membrane theory of biopotentialcy, which on the basis of Ostwald's conclusion was developed 40 years ago by Bernstein. Characteristic feature of this concept is the assumption of a selective membrane permeability to cations, particularly to K-ions, which are present in excess inside the muscle fiber and many other cells, and therefore can establish a concentration potential drop from inside to outside across the membrane of such magnitude as is otherwise obtained only with metallic electrodes in a concentration chain. The more selective the permeability, in other words, the greater the difference in the ratios of cationic and anionic mobilities inside the membrane structure and the bordering aqueous solutions respectively, the greater is not only the potentile drop, but also the ohmic resistance in the system. After disintegration of the structure, both the membrane resistance and the potential drop disappear. Therefore, a local damage is followed by the appearance of a potential difference between the intact and the damaged surfaces, and an injury current starts flowing in the outer circuit from the intact surface as a positive pole to the injured portion as a negative pole, with an electromotive force equal to about 70 MV in a frog muscle. The objection has often been raised to the membrane theory that there are other means to account with regard to the location of, in the hydrotropic series near to Ni, and not beyond K. It should be mentioned that Netterfly-Jeffs, Arch FDGCs, Physiol 218, 310, 1928, and others refer this unexpected iocatinithrine to the fact that he resembles the alkaline earth ions in some of its chemical properties. For the persistence of the unequal distribution of ions between inside and outside, then selective ion permeability of the surface membrane. But particularly by recent experiments on giant cells, conclusive evidence has been advanced in favor of the membrane theory. The procedure is, for instance, as follows. A single cylindrical cell of the fresh water plant, Nutella is placed in a groove in such a way that, close to one end, two stimulating electrodes are attached to the cell. Two other electrodes, more toward the center of the cell, touch its surface opposite to each other, thus permitting measurement of the transverse resistance, while another electrode is located at the other end, serving to record the action potential wave which travels along the cell after a stimulus has been applied. This arrangement makes it possible to follow at one point of the surface simultaneously the variations of the resistance and the membrane potential. The results show that, fairly coincident with the action potential wave, that is, the wave of propagated negativity, which has a duration of 2 to 3 s-cons, the transverse resistance drops and then rises again. In other words, the membrane passes through a short phase of structural breakdown as indicated by both the rise and fall of electrical conductance and the concomitant loss and gain of selective ion permeability. The size of the effect of stimulation is such as to bring about, for example, a decrease of resistance from about experiments on the single giant axon of the stellar nerves of the squid, Loliga, as the negative wave of excitation passes between the resistance electrodes. The resistance falls from the resting value. It is suggestive to consider that this closely associated temporary drop of potential and of resistance derive excitation. indicating a loss of selective cation permeability may be accompanied by an escape of electrolyte from the interior of the cell. Although, besides other factors, the short duration of the breakdown, that is, a small fraction of 1 second with the squid nerve, is unfavorable to proof of this assumption. However, in the experiment with the more slowly reacting Nutella, it has been observed that after a single stimulation, the conductivity fails to return to its previous value, but remains at a higher value for some time. And that on repeated stimulation, the conductivity becomes progressively higher. But that there is an immediate return to the initial value when water is circulated along the cell. This may indicate the increase of conductivity to be due to a loss of electrolyte from the cell during excitation. This fits in with many statements concerning the increase of permeability involved in experimental or in normal changes of functional activity. 3. The nature of selective ion permeability of physicochemical and of physiological membranes. The concept of a selective ion permeability of membranes, which at first introduction was considered rather fantastic, but soon was well supported by Kopep's demonstration of the anion permeability of the erythrocytes, became a powerful stimulus to a systematic investigation of various simple model substances with respect to their properties as membranes. Separating different electrolyte solutions, the most important models resulting from such studies are the porous collagen membrane of mycolaces and the oil-like solvent membrane, the dried collagen membrane. See also Sec 1p. 70. Aids in elucidating the behavior of the muscle and the nerve membranes in the following respects. 1. A wet collagen membrane with sufficiently wide pores interposed between two solutions of the same alkali salt in different concentrations yields a low membrane potential, which is equal to a diffusion potential. It is 0 with KCl because with this salt, the cationic mobility U is the same as the anionic mobility V drying. The wet membrane raises the potential. It can approach the theoretically maximal value, which at room temperature and with a concentration ratio 10 one of the two solutions is equal to. This is due to V becoming 0 because by drying the porosity of the membrane is decreased in such a way that it becomes anion impermeable and only the permeability to K persists. The membrane is polarized with the positive pole oriented to the lower concentration. With membranes dried only to a lesser degree, other cations also can enter the membrane according to the order Jibkwandali, whereas the anions have a minor or no effect. 2. Here the order of the cations is not the order of their atomic size, as could be expected with regard to porosity, but it is the order of their ionic mobility, signifying that the atomic size is increased by hydration. In other words, by a shell of water dipolase, largest with Ola and smallest with K3. The preferential cation permeability depends upon the negative charge of the pore walls of the membrane, which is due to the ionizable acidic groups attached to the collagen nitrocellulose molecule. This charge can be converted to positive by impregnating the membrane with adsorbable organic bases, for instance, basic dyestuffs or alkaloids. Or protamines, the result is a selective anion permeable membrane, with the negative part of the electric double layer of the diffusion potential oriented toward the more dilute solution, and with the poxetetial height indicated in contrast to the findings with the cation permeawalk membranes. Not the order of ionic mobility, which would be I-bor-Ichiel-no-ACN. Either is a lyotropic series of anions, P293. Hence, it is evident that the ability of the various ions to penetrate the collagen membranes, both the impregnated and the non-impregnated, does not measure their diameter, including the sphere of water molecules, as at first believed to be the case with the cations, but that it is determined in some other way by hydration. Namely, with cations and with anions, the penetrating ability increases with decreasing hydration, or rather with increasing adsorption. For as has been mentioned earlier, P299, the least hydrated ions have the greatest adsorbability. When a salt approaches by diffusion the surface of the cation permeaulic membrane, the anions act like being repelled. They are restricted in their mobility by the charged negative groups, which are fixed immovably to the collodion. On the other hand, the cations act like attracted with K more effectively than with Li. This is partially due to adsorption, which is greater with the less hydrated K than with Li, and partially, perhaps chiefly, due to the fact that many more pores are available to the small K than to the big Li ion. Correspondingly, with the anion permeable membrane, the less hydrated CN is more effective than SO4, CP299. Thus, it appears that the electrochemical activity of the membranes must be correlated with both the number of ionizable groups per unit of area in the membrane and WDF, the relative size of the movable 4. The same interrelationship between the electrical charge of the membrane substance, the ionic diameter, and the height of the electromotive force obtains with membranes, which are more analogous to the physiological conditions than the colloidal membrane, and the charge of which can easily be shifted, as observed with membranes composed of calc. These membranes are preferentially permeable to the cations on the alkaline side of their isoelectric point, to the anions on the acid side. Correspondingly, their membrane potentials are oriented in regarding the theory of this influence of charge upon permeability. C. K. H. Meyer, with J. F. Sievers and H. Hauptmann, Helvet Chem. Acta 19, 649, 665, 948, 987, 1934, 1937. TRL Procna Acad. Eski EAZ 21 152 1935 Prosiki Soki Expo. Biol Simi 83 282 1935. W. Opa. Physiol 40 204 1938 K. Solner and C. W. Carr J. General Physiol 28 1 1944. Also see Sec 1P 71. Directions opposite to each other on either side of the isoelectric point, and display by the potential height the order of the cations and the anions, respectively. For instance, measuring the influence of the ions on the I-like values, MV, have been observed a similar behavior is shown by natural animal membranes. In particular, the teleotric properties of the frog skin have been investigated for various purposes, see Sec 8. The overall value of this membrane potential is the algebraic sum of at least T free seg R rot E potential. The skin potential CFU Trixori, which is located in the exterior layer of this complex structure, and which is displayed if a freshly excised piece of the skin is bathed on both surfaces by Ringer solution, is somehow connected WDFT life state of the epithelia. A second potential is an ordinary diffusion potential, which is brought to evidence if the skin is placed between salt solutions of different concentrations. It probably is bound up to the DIR layers of the skin, the fibrous connective tissue, including blood vessels, smooth muscles, and nerves. The third potential is a membrane potential, which is left after the skin has been killed, for example, by an acid at about PA4. It is signified as such by the fact that its direction can be changed by changing the pH. Similar to Tly behavior of the aforementioned colloid ampholte membranes. Above 4 or 5, the dead skin is preferentially permeable to cations, below to anions. For this reason, the membrane in question is assumed to consist of a protein located in the tighter exterior layer and having an isoelectric point around 4.5. And indeed, this pH coincides fairly well with the reversal point for electroosmosis across the skin. Returning now to Bernstein's membrane theory, P315, its main fundament was considered the great surplus of K inside the muscle fibery, the inside concentration in frog muscle, being about 20 to 40 times greater than the outside concentration. How much is bound is so far unknown. Jot had been mentioned before that muscle activity always is accompanied by loss of K to the blood, P57, and that K can move from the blood into the muscle against the concentration gradient and agreement with the expectations of a membrane, equilibrium, see Sec 4, P254 and Sec 8 introduction. Thus, a membrane potential dependent upon a selective K permeability can be anticipated. According to chemical analyses of muscle, the fibers appear to be impermeable, able to annate and to anions. See Sec 4, pages 253 with 23 milligrams. Percent K in the outside ringer and about 400 milligrams. Percent inside the fibers. After destruction of one end of the muscle sartorius, the height of the injury potential could be expected to be, but in reality, it has not been found higher than an average of 41.6 increase in co-causes, E to fall off it, as it should. Towards zero, it should even be reversed if I's postulate of the tulu ori was not complied with in earlier experiments, evidently because the muscle had been soaked or perfused for some time by isotonic ECL and was killed by this treatment. However, the postulate has been fulfilled in recent experiments on the giant squid nerve fibtears, where with half or full isotonic ECL, a reversal of the difference between 73 and 41.6 MV is more or less understood, taking into consideration the following situation. 1. The interfibral fluid and the tissue space between that fiber bundles form short circuits. 2. The various anions do not fail entomly to influence the height of the potential peak 313 and can be assumed to penetrate the muscle membrane to an individually different degree. 3. The K concentration between the fibers inside the whole muscle may easily be somewhat higher than in the bathing solution, due to slight damage or to twitching. 4. On the other hand, a certain and probably variable fraction of KT is not free, but bound. 5. Slight up and down effects of anions upon the potential were interpreted earlier as indicating changes in the colloidal state of the membrane components. This was especially obvious from the fact that the most influential anions, CN and I, provoke at the beginning of their application a positive pole which afterward reverses to a negative pole, CP 313. See further, reference number 74, brain serving for selective ion permeability is not adequate to portray the physiological conditions, and that a more flexible colloidal structure is more satisfactory as a model of the muscle membrane. Moreover, the rigid collodion membrane cannot be expected to show the balancing effect of mono and divalent cations. Regarding nerves, it has already been mentioned, P 314, that the character of their resting potential resembles that of muscles. It should be added here as significant for the colloidal properties of their membrane, that the depressant influence of K and RB upon the potential can be antag. Moreover, nerves have furnished specimens which appear to be exceedingly favorable objects for more crucial tests of Bernstein's membrane theory. CP 315, the excised giant axons of the squid. These cylindrical fibers are large enough to yield at their cut ends enough axoplasm for determining the inorganic and organic constituents about the results. 16b. 257. In studies on the excitation weight of these objects by Webb and Young, TICO was found to be 29, assuming that during rest, the membrane separating the axoplasm and seawater be permeable only to K, and that during the passage of an excitation wave the membrane be rendered permeable to anions. The calculate value is about 85 MV. The observed value, average of 7 experiments, was 77 MV. In corresponding experiments, Hodgkin and Huxley have obtained values up to 95 MV, with one lead from the intact surface and the second from the injured end. A resting potential of 50 MV has been measured, this lower value probably being due in part to the liquid junction potential between the axoplasm and the fluid surrounding the injured end. Finally, about the same average value, 51 MV, has been observed with an impaled giant fiber. That is, with one capillary fluid electrode piercing the axon membrane and pushing inside along the axis, until its tip is opposite to the tip of the second electrode outside the fiber. Under such conditions, the impaled axons often remain excitable for many hours. It is obvious that the various and partially contradictory results of this group of experiments need further study. However, in any case, it must be realized that there is a considerable number of observations indicating that, at variance to the symptoms of impermeability to ions, or also to signs of a slight passive penetration, there is evidence of an ample, though slow and probably active, passage being superimposed and detectable mainly by chemical instead of physical means of analysis. A striking example concerning muscle, which has been mentioned before, p. 257, is the result of FCI, ding rats a K-poor diet for several weeks. The K-deprived rats show a loss of about 50% of K in their muscles and a concomitant rise in oddbot. The K remaining in the depicted animal is located practically entirely inside the fibers, and A has passed into the fibers to such an extent that there is more intracellular than extracellular Na, in some cases even twice as much. This probably must be accounted for as the effect not of a passive penetration, but of an active transport, compensating for the loss of K during the long period of deprivation. Such a process, which really is a shift of Na against a diffusion gradient, is not unlikely on the basis of many similar observations with various plant cells, with kidney and liver, where the shift of cations and of anions is bound up with metabolic reactions which supply the necessary osmotic energy, see been demonstrated with human erythrocytes, which for a long time were assumed to be impermeable to cations under normal conditions, but which are now known to show an exchange of K and Na when kept for many hours at body temperature in the presence of glucose, accompanied by utilization of the sugar. T increases, not decreases. Moreover, K, which previously was lost, re-enters the cell after addition of glucose. In contrast, at low temperature, which stops the metabolic reactions, both K and Na diffuse with the concentration gradient. It often has been conceived a universal phenomenon that cells have to expend energy, even at rest, in order to preserve the dynamic state, which is characteristic of life. Also, the normal polarized state of frog and crab nerve, as indicated by the constancy of their resting potential, cannot be kept on its high level without expenditure of energy. It falls off during anoxia and rises anew after return to oxygen. These effects upon the resting potential seem to be due to the appearance of lactate and pyruvate in the normal glycolytic cycle of muscle and nerve tissue, and to the oxidation of these intermediary products only. With this cycle intact and with oxygen being present, can the membranes keep their normal molecular organization, upon which depends the normal polarized state of rest, attending failure in this regard, K, along with some agents, escape, as it may escape also during excitation. This is quite to be expected, since it has been shown definitely, mainly due to the aforementioned observations of Cole and Curtis, p. 316, that the electrical resistance of the surface membrane falls off considerably when the action potential wave travels along a Nitella cell, or along the giant axon of the squid. Oxygen may then serve not only to rebuild the membrane, but also to cause back transfer of lost contents. However, notwithstanding the high concentration gradient of K from inside to outside, it has often been denied that the decrease of the polarization is associated with the release of K. It is true, muscle activity is always accompanied by the loss of K, but this can be an immediate consequence of the contraction process, CP 257. In nerves, there is no conclusive evidence for a release of K either on stimulation or on oxygen lack. However, it must be realized that the chances of obtaining such evidence are rather poor. In myelinated frog, nerves a loss probably is restricted to the small area of the Riviere knots, and recent measurements of the changes of the impedance and of the capacity during excitation have yielded the surprising result that in the giant axon of the squid, only 2% of the area of the membrane, in Nitella 15%, are involved in the increase of permeability. If then, during the short duration of an excitation wave, electrolyte should escape, the small amount could evade demonstration, provided the loss was repaired immediately by a back transport similar to a secretory active transfer. The best chance for a demonstration of such escape might be offered by an object, the excitation reaction of which travels with low speed, and the local depolarization of which is correspondingly of a relatively long duration, as it is in Nitella, where, in an experiment referred to before, P316, there has been found indeed some indication of leakage. In conclusion, some earlier observations of J. Loeb, Osterley, and others correlating colloidal and electrical properties of certain animal and plant membranes may be discussed. According to Loeb, The eggs of the Recavator Teleos fungulus can be kept living in seawater as well as in pure water. When an egg is put into KTL solution, the heart of the embryo, which is covered by a thick egg membrane, the chorion, stops beating after the salt diffusing across the chorion, and the ectoderm of the embryo has reached a certain concentration. Loeb and Catalou have found that the cessation of the heartbeat persists over many hours in distilled water, as well as in isotonic saccharose solution, or more generally in absence of ions. While the pulsation reappears readily after adding a non-poisonous salt to the surrounding fluid, the rate of recovery increasing with the salt concentration. Also, low concentrations of the surprising influence of the lack of ions seem to have found a satisfactory explanation by some weight, who ascribes this effect to the resemblance of the chorion to a dried collagen membrane. According to Michaelis, KCl cannot go across this membrane into distilled water because it is impermeable to Cl. But after NaCl has been added to the water, movement of K occurs by exchange of NaCl. This has been proved to be true mainly by measuring the membrane potentials. The same procedure has been applied by Sumwalt to the chorion of the fungulus egg. One microelectrode was fixed in the subchorionic space filled with M2 KCl. Another electrode was placed in solutions of varied composition outside the egg, and the potentials, during less than a half hour, were measured across tlychord. It was found that, with K outside at various concentrations, the more dilute solution is positive. That with different anions, KL1, NOACN, acetate, the potentials are equal, and that, varying the alkali cations in M200 solutions, lye solution is the most positive, CS solution the most negative. By these results, it was made evident that the chorion membrane is selectively permeable to cations, or more correct, is more permeable to cations than to anions. However, these measurements do not account for some especially significant observations of Loeb in cattle. It was mentioned before that the heart of the embryo, which had been stopped by KCl, starts beating again in salt solutions of a certain strength. Varying the anions of anise salts, it appeared that the recovering power is greatest with citrate and falls off in the order citrate for two reasons. First, it is at variance with the aforementioned indifference of the anions toward the chorion potentials. Second, the order of anions reminds one of the Hofmeister series, but disregarding some Urcularitics, the succession of ions runs inversely to the usual order P300 to 2. The following points will contribute to a clearer understanding. First, the indifference of the anions, as stated in the potential measurements, may be due to the brief period of the time of observation, during which the penetration of the cations was predominant. Second, in order to influence the heartbeat, not only the chorion, but also the ectoderm and underlying tissue of the embryo must be penetrated. Third, especially citrate, but tartrate and phosphate also will display their recovering power for a short interval only, which is followed by poisoning. These experiments of Loeb and Catal can be compared to some extent with experiments of Rauber on the thallus of the marine alga laminarianae, studied by the method of Osterhout Chied 1. To 200 discs of the foliage were piled up to form a cylinder and soaked in salt solutions, replacing the normal milieu, seawater. Conductivity measurements served as indicating the ion permeability of the cells composing the plant tissue. With different nene salts, the conductivity rose fairly rapidly with CN, No3, faster with acetate, and fastest with C4 tartrate phosphate citrate. This behavior reminds one of the inverted Hofmeister series as much or as little as in the experiments of Loeb and Catel. However, the significance of the experiments is considerably reduced by the fact that the half-molar pure NaS salt solutions can be assumed to impair the life of the cells. P. 245. As is evident regarding citrate, phosphate, and tartrate. Furthermore, the subject of this study is a tissue composed of several structures, the cells, the cell walls, and the intercellular substance, each of which offers a specific resistance to the current, contributes to the conduction according to its relative volume, and possesses unknown specific colloidal proxurities. Finally, attention also must be turned to the fact that even colloidal material which is simpler than the double membrane of the fungulus egg or the composite architecture of laminaria. Displays the phenomenon of inversion of the Hofmeister series of anions, for instance, under the influence of higher salt concentrations or after shifting the pH in one or the other direction. See pages hence. It is obvious enough that a more thorough investigation of these different processes is needed before they may be quoted as counter-evidence against an interpretation of physiological states or processes, as analyzed in this chapter. On the basis of colloid chemistry, the basic principle of the bioelectric membrane potentials can be illustrated by referring to the electromotive force of a Helmholtz concentration chain of the following type: C agnoa, Yag agnoa, where the EK tetric current is let off from Ci and C2 by identical fluid electrodes suitably conomostructed to avoid the establishment of liquid junction potentials at the contact with Ci and C2, Sec4 P64. Then, if CiC2 T equals EA2A equals A log, CK log corresponding to the two potentials at either surface of the silver diaphragm, which separates Ci and C2. These potentials differ according to the different tendencies of Ag ions to enter into the metallic silver from Cl and to pass out of it into C2. It appears that the diaphragm is like a membrane selectively permeable to cations, for instance, like the surface membrane of a giant nerve fiber in contact on one side, with high K on their 21. It is then conceivable that Nernst's thermodynamic theory of concentration chains, see pages 63 and 317, likewise applies to the behavior of the physiological structures. But conditions here are generally more or less complicated by the thermodynamic epotent tiles being superimposed upon electrokinetic ECHG potentials, which similarly are located at the interface between the membrane material and the adjacent solutions, and are of special interest because of the characteristic sieve-like structure of the physiological membranes and their great porosity providing a large internal surface for adsorption. But as outlined in Sec 1, pages 83, the F potential differs from the E potential in that its site is not exactly at the phase boundary, but somewhat distant between a thin layer of the solution, which is fixed by adhesive forces to the membrane material, and thus quasi-rigid, and the free solution. This interface has been called the rigidity boundary or the surface of shear. Here, a diffuse double layer of ions is formed, the orientation of which is determined by the electrical charge of the membrane material, which, for one or another reason, is either positive or negative. If an electric field is applied tangential to the internal pore walls of a membrane, which is interposed as a diaphragm into an electrolyte solution, that is, if an external electromotive force is applied to the ends of the capillary pores, then, due to the presence of an excess of ions of one kind in the movable boundary layer, the latter is moved, taking with it the free solution within the core of the pores. Electroosmosis is zero, according to the formula P85, from the rate of the fluid movement U, as brought about by the unit electromotive force, C ampere sec, per Voltsenia. The electrokinetic, or F potential, can be calculated. This potential is somewhat similar to a liquid junction potential, P86, but in contrast to this, is not indicative of a dynamic process like a diffusion, but is a certain small part of the static 6 potential of membranes. It is clear that the concentration potential differences measured across a porous membrane must differ parallel with the F potential of the membrane, the mobility of the free cations, while passing a membrane with positive charge being decreased. As well as in a negative membrane, the free ions likewise are decreased. This is particularly evident in experiments with membranes, whose electrical charge is readily changed by the attachment of ions, as observed, for instance, with colloid amphiolites, proteins, gelatin, by changing the pH, as well as with more indifferent substances like collodion or agar, when they have been coated with a film of these colloids. Examples of this influence have been quoted previously, p. 318. Referring to the numerous aforementioned membrane potentials of cellular origin, it follows that all the alterations of their height affected by inorganic salts, or rather, by the ions, are indicative of adsorption. which modifies the relative mobility of the ions in passing through the charged membranes. 1. Electrophoresis of cells. Further information concerning the physiological significance of the electrokinetic potentials is obtained by studying electrophoresis of single cells. According to the formula, 85, their F-potentials can be calculated from measurements of the velocities of suspended cells moving in an external field. Erythrocytes have been the principal material used. Their surface can be assumed to be the site of an E-potential, since the surface is selectively anion permeable, and since the distribution of ions has been shown to be a Donan distribution. The E potential should be determined directly by inserting a microelectrode into a red cell, as previously described for Nutella and for giant nerve fibers, P321, and taking the second lead from the outside. But such a transverse measurement has not been carried out as yet. As a matter of fact, the tangential measurement of the F potential shows that this F yields to be altered by disintegrating the surface membrane of the erythrocytes. In other words, by hemolysis. The intact red cells and their stromata, ghosts, suspended in the same medium, and under otherwise identical conditions, have the same mobility. The F potentials of various suspended particles, either inorganic or organic, like pieces of glass, quartz, collagen, starch, cotton, and droplets of mineral oil, are characteristically different. Most of the particles move toward the cathode, some, for example, ferric oxide, toward the anode. Size, shape, and orientation of the particles have no influence upon their potential. The differences of direction and velocity of migration disappear in the presence of colloid amphibs, like gelatine, when coating at the same pH the various surfaces. The particles then behave like particles of gelatin. Accordingly, for example, when glass is suspended in acetate buffer at pH 5.6 and coated by globin is at pH 6.6, or serum globulin is P5.6, or serum albumin is P4.9, the F potential is found to be positive, 0, and negative, respectively. The behavior of the erythrocytes is somewhat different. Again, shape and size are irrelevant. Single corpuscles masses held together by agglutination or by rouleau formation, and ghosts migrate with the same speed. On the other hand, rinsing the cells, which could be supposed to be normally surrounded by an adsorption layer of plasma proteins with phosphate buffer PTL74, has practically no effect upon the R nobility. Addition of gelatin to this suspension likewise fails to bring about a distinct change of the potential, whereas it does with suspensions of quartz or cholesterol. This seems rather incompatible with the idea of the F potential of the red cells being due to adsorbed protein. Moreover, pH changes have been found to have little effect upon the potential, and this may explain also the rather unexpectedly low value of the isoelectric point, which with sheep and with human cells is found at about pH 3.6. Provided the cells remain unmolars and warm, apparently then the E potential of the erythrocytes should be referred to their surface proper, and the rather great differences of the electrokinetic potentials found with various animals may be associated with the chemically and physicochemically species-specific surface structures, but this interpretation has not been proved as yet. The following table shows the mean velocity of mammalian erythrocytes, v equals ephemerer 2 phi electroosmophis, across membranes. Another kinetic method for determining the G potential of physiological objects is based upon observation of electroosmosis, CP 328. Serous membranes, mesentery, peritoneum, pericardium, and skin, best placed as diaphragms in buffered solutions, are subjected to a direct electric current. The rate of fluid shift, in contrast to the rate of electrophoresis of red cells, is easily changed by changes in one or the other direction, and is 0 at a definite pH indicating the isoelectric point of the membrane material. This has been found with mammalian membranes to be about 4.9, probably referable. So far, the bioelectric potentials of muscle and nerve have been conceived as originating from the presence of sieve-like surface membranes, which are interposed between the intracellular and extracellular electrolyte solutions, However, more than 20 years ago, another concept was proposed, which in many respects is rivaling the former. This is the concept of solvent-like membranes as envelopes for the protoplasm. Both theories have been proposed and outlined in close contact with the contemporary development of our knowledge of cell permeability, emphasizing for some time the importance of lipoid solubility, and later the factor of the molecular volume of the permeating substances. It seems desirable to refer, at least briefly, to the main points of the solvent theory, which was built up chiefly by Buettner-Chu, who claimed that oil-like organic solvents are essential structural elements of the surface of the cells and are the seat of their potentials. In general, organic liquids which are immiscible with water are poor solvents for strong inorganic electrolytes, since their low dielectric constant prevents an appreciable dissociation into free ions. This holds true mainly for the solvents of a chemically neutral character, like the neutral hydrocarbon oils. The situation is different in the cat of organic solvents having some acid or basic properties, either by their chemical nature or by impurities or by additions such as phenol, guaiacol, salicylic aldehyde, and toluidine, aniline or solvents like Nierenstein's mixtures, chaps 10 and 13. They behave as weak electrolytes and can dissolve small amounts of strong electrolytes, chiefly by ionic exchange. Through conductivity measurements, it can be shown that the inorganic alkali salts, upon shaking their aqueous solutions with solvents like guaiacol or benzaldehyde, pass into these liquids in characteristically different amounts. Now, when two aqueous solutions of the same electrolyte at different concentrations are placed on either side of an intermediate layer of an organic fluid, an electromotive force can be demonstrated, which, for example, with salicylic aldehyde or with guaiacol, in addition to certain amounts of oleic acid as membrane, and with a concentration ratio of KLL 100, is equal to about 100 MV, the positive pole being on the side of the lower concentration. If the acidic oil is replaced by a basic one, toluidine or aniline, the sign of the poles is reversed. This is like reversing the charge of an ordinary colloidal membrane by impregnating it with a basic diastrophin or an alkaloid. The following explanation of the functioning of the oil chains has been proposed. Nernstu and Kramer-Jong have outlined the theory that at the boundary of two phases, one an electrolyte solution and the other a water immiscible liquid solvent, a phase boundary potential is set up, owing to the individual distribution tendencies of each kind of ion. It follows that in any liquid chain consisting of a solvent membrane interposed between two different electrolyte solutions, two different phase boundary potentials must be present, the difference between which is a measure of the different tendencies of cations and anions to enter through the phase-bound area into the oil phase. Taking as an example the aforementioned concentration chain with the two aqueous solutions containing the same electrolyte at different concentrations, the following explanation can be proposed, according to Buettner. At the interface between a KCl solution and salicylic aldehyde, K enters the organic solvent at a higher rate than Cl, because it can be exchanged for H present in the aldehyde in a definite concentration. In the concentration chain, CiClTl salicylic aldehyde, C2KCl, with ClLC2, in spite of the concentration difference between Ci and C2 at either of the phase boundaries, an equal amount of K, dependent on the limited small content of acid within the organic solvent, enters from both sides. Therefore, the chain functions according to the following scheme. GLK in water, CK in salicylic aldehyde, C2K in water, the potential evident that, in this case, the solvent membrane behaves like a selective cation permeable membrane. Correspondingly, basic solvents form anion permeable membranes. But a quantitative agreement between theory and experiment, as suggested by the formula, cannot be expected because of the just mentioned interreaction between the electrolyte solutions and the organic solvent. This is evident from careful measurements by Osterhagen on a model with guaiacol representing the protoplast of one of the giant plant cells, chapter 21, interposed between K guaiacolite solutions of different concentration, which are in equilibrium with the interface. Physicochemical and chemical analyses of the interface show that concentration differences inside this interface, undeniably involving diffusion potentials within it, cannot be neglected. When different sodium salts are compared, the values of these potentials follow each other in the well-known order of the lyotropic series. P 293. Of anions, SO4 UL NO3, INCN with CN giving the strongest, so far the weakest positive pole, irrespective of the oil phase having an acidic or basic character. This possibly is due to the fact that in the series of anions, hydration decreases from SO4 to CN, and that therefore CN has not only the highest adsorption affinity, but also the highest solubility in the hydrophobic organic solvents. See pages 299 and 318 in addition. If the influence of the series of anions and cations upon the potential differences of the oil chains is compared, it appears that in general the cations have the stronger effect in chains with acidic solvents, like salicylic aldehyde or GiCl. The anions in chains with basic solvents, like toluidine. Therefore, with a basic oil, CN produces a stronger positive pole than SO4, as in the case of acid gelatin, while with an acidic oil, CS calls forth a stronger negative pole than Li does, as with a membrane of alkaline gelatin, CP 319. Further, the influence of a neutral solvent like olive oil is small, but can be enhanced, for instance, by the addition of oleic acid or of lecithin salts, with one organic ion often reveal an especially strong effect, either on the positive or on the negative direction of the current, depending upon whether the anion or the cation is organic. In conclusion, it might be considered desirable to refer briefly to the surfactant films as another kind of models, in addition to the porous and the solvent membranes, for illustration of the origin of the various bioelectrical potentials. However, this seems premature since the difficulties of spread engine these fragile films between two aqueous solutions, instead of between water and air or water and oil. In order to study the influence of organic or inorganic ions under conditions similar to those in living material in general, the inorganic ions have been found to influence vital reactions according to their adsorption at the interface between water and the physiological object. In other words, the hydrotropic series of anions, CNJ NO3, TRCl4 and of cations, caenolia, which refers not only to organic colloids but also to cells and tissues, is a series of decreasing adsorption, increasing hydration, or, as one may also say in terms of the theory of Buechner, a series of decreasing distribution between an aqueous and an oil-like phase. In investigating the properties of the cation peak permeable collodion membrane, this interrelation first became evident when, in the experiments of Michaelis strychnine salt, and even more pilocarpine salt, separated by the membrane from the salt, gave a positive potential at variance with the rule that the selective permeation of cations decreases with increasing diameter, P299. Correspondingly, the permeability of the selective anion permeable collodion membrane, Ps 317, was found to be greatest with CN, which by no means is the smallest ion in the anion series, P318. Later, other alkaloids and dyestuff bases were shown to behave like strychnine. Similarly, various organic anions with relatively large diameters, benzoctane, salicylate, aromatic sulfonates, appeared to produce a greater negative potential than CLFCs, finally testing the series of dialkylamine ion. The positive potential was found to decrease from the methyl to the propyl, corresponding to the rising diameter, but to increase considerably from the butyl to the amyl compound. According to the rising surface affinity, all these and other facts can be explained as indicating adsorption to be a factor favoring penetration. This possibly can be accounted for by assuming that the special adsorption affinities alter the homogeneous distribution of the penetrating ions inside the pores in such a way as to increase the rate of passage near the wall. On the other HG&D, in the absence of special affinities between the solutes and the pore wall, apart from the electric charge of the membrane substance, which in an ordinary collodion membrane repels the anions and attracts the cations, the solutes would pass the channels in the sieve membrane as in normal diffusion. Turning now from the rigid collodion membrane to the more flexible physiological membranes, which are composed of various colloids, adsorption may again be found effective because in general surface active substances, whether electrolytes of greater or smaller ionization strength or non-electrolytes, are lipoid soluble and as such can migrate through the lipoid components of these membranes. See chap 11 to 13. In addition to the influence of the molecular size and of the ordinary adsorption, there can be involved in the penetration of the membranes the dual adsorption affinities of a special class of organic ions, representing, as such, new tools for studying the nature of cell permeability. Newberg has called attention to the fact that many water-insoluble or nearly insoluble substances of diverse nature, higher alcohols, aniline, nitrobenzene, oils, cassops, proteins, starch, become soluble in the aqueous solution of the alkali salts of many organic acids, if the latter are present in high concentration solubilization. Such hydrotropic proiorities are displayed mainly by aromatic carboxylic and sulfuric acids, as well as by higher fatty acids and higher alkyl sulfuric acids. Most of the salts are fairly strong electrolytes. All these hydrotropic compounds are related to each other by a certain configuration of their molecules, one molecular half being polar hydrophilic, the other nonpolar organophilic hydrophobic. Their hydrotropic properties are based upon the adsorption affinity of the organic half, which attaches the molecule to the water-insoluble hydrophobic particles, and upon the affinity of the polar inorganic half for the water dipoles, Langmuir iarkins. If the pull toward the water is strong enough, the insoluble particles can be torn to pieces and thus be dispersed. But, depending upon the greater or lesser firmness of their structure, following the attachment of the organophilic half, the hydrophilic half, with its cloud of water dipoles, can be pulled inside the particles, with the result that an imbibition of water or even swelling of the particles occurs. Corresponding to this theory, coarse suspensions of proteins of starch of lipoids are emulsified and dispersed by adding nonpolar duopolar ions, while with ions prevailingly hydrophilic, owing to a multitude of polar groups, like citrate, tartrate, succinate, and other plurihydroxy pluricarboxylic anions, dehydration and decrease of dispersity is the result. According to this concept, one can expect and really observe that, depending upon the balance between the opposite molecular forces of the added ions, their organophilic half may adhere to a non-polar structure, for example, a plasma membrane, with such strength as to produce an irreversible alteration, CP 338, or that the hydrophilic radicals may be so dominant that the weaker organophilic fail to exhibit their influence, 68 chap. 5 and 37, 5, or that the antagonistic forces of the two halves may be great enough to produce opposing strains capable of mechanically disorganizing or denaturing larger molecules, for example, hemoglobin. See pages 297 and 338 for experimental studies. Substances of such a variety of non-polar-polar behavior can be selected. First from the list given in Newberg's paper, and second, among technical products utilized for wetting, dispersing, and emulsifying purposes, the so-called detergents in contrast to these substances. Many of the natural products of the metabolism of animals or plants, which come into contact with the plasma membrane, either from within or from without, the cells, lactate, pyruvate, succinate, malate, citrate, various phosphorylation products, and many others lack the qualities in question. They are prevailingly hydrophilic and therefore physicochemically rather indifferent to the physiological membranes. And although our knowledge in this regard is very poor, they probably fail to pass by simple diffusion in one or the other direction, except the specifically anion permeable membrane of the erythrocytes, which compared to TL, is penetrated by these ions with remarkable slowness, possibly due to dehydration, to tighter packing, and thus to strengthening of the lateral adhesive forces between the structural units. However, in a survey of the products of cell metabolism, some compounds are encountered which, by their distinct non-polar-polar configuration, can be expected to be more or less membrane active in the sense of the aforementioned reactions. Such substances are the salts of the higher homologs of the saturated or unsaturated fatty acids, the salts of the bile acids, and other similar compounds. In order to exemplify the significance of the non-polar-polar configuration of organic ions, we turn now to the study of excitation and of production of resting potentials in muscle and nerve. The influence of non-polar-polar ions on the resting potentiles and the excitability of muscle and nerve. The synthetic nonpolar electrolytes, so far, 3 groups of nonpolar-polar anions have been investigated: alkyl sulfonax rososna, dialkyl succinyl sulfates of iron, and alkylbenzene sulfonates. They are strong electrolytes in water, but due to the nonpolar organophilic groups, they are more or less soluble in organic solvents, dialkyl succinyl sulfates, for instance, in kerosene, xylol, ether, glycerol, and olive oil. Their organophilic char actor increases with the number and the chain length of the alkyl groups present. Correspondingly, the interfacial tension water-air and water-organic solvent decreases. Aber unpublished experiments, dissolved in Ringer, these substances effectuate with frog muscles sartorius a resting potential like K. That is a depolarization. The threshold concentration of dialkyl succinyl sulfates, for instance, has been found to 0.7x10. In general, the reversibility of these bioelectric effects is low and diminishes with rising surface activity. On the basis of the theory, these influences can be accounted for as a dispersing effect, brought about by the pull of the interfacial array of the hydrophilic group of the anion toward the water and by the penetration of its organophilic groups into the surface film, with a subsequent widening of the characteristic spacing in the latter and therefore, eventually, an increase in the content of free water. This disintegration is probably followed by some entrance into the film structure of the anions, for example, Kiel. No more repelled entirely of the active nonpolar-polar ions can result in a complete rearrangement. Returning to the dispersing effects of K or SCN, pages 300, 333, there seems not to be a fundamental, but rather a quantitative difference between these and the effects here dealt with, which are due to the definite opposing forces of the nonpolar-polar compounds. Also, there is a certain relationship between surface-active non-electrolytes and the nonpolar-polar electrolytes. For instance, narcotics, after being brought locally in contact with a sartorius muscle, have a reversible depolarizing CFET-AFDCT like that of K. This may be referred to the particular affinities of the narcotics toward the lipoid C-chap 23, which are structural components in the surface membrane, and which as such can be dislocated by the intruding molecules of narcotics, so that, again, spatial changes in the normal molecular orientation will result in muscle. A K-like reversible depolarizing effect has been produced. Hydrid Q and can like that of K. Be balanced by alkaline earth, ionic ions. Only in one respect the surface structure of cells and fibers seems to be acted upon specifically by the nonpolar-polar ions, that is, the denaturing influence upon proteins. Denaturation of proteins can be brought about many nonpolar-polar agents, as for instance by those mentioned here, P336, but not by KCN and narcotics. The denaturation of proteins is easily indicated, for example, by the liberation of HNS groups plus groups. This liberation has been found concomitant with the loss of the specific effects of enzymes and viruses, with the separation of the protein part from visual purple, from chloroplastin, and others. These reactions occur at low concentration of the nonpolar-polar agents and at neutrality. Certainly they contribute to the often observed irreversibility of the electrical phenomena. With many synthetic nonpolar-polar electrolytes, in contrast to those mentioned so far, the typical activity is located not in the anion, but in the cation. Most frequently, the neutral salts of quaternary ammonium bases, particularly salts of the type alkyl dimethyl benzyl ammonium chloride, have been utilized in solving biological problems. Their influence differs mainly from that of the anionic agents by the fact that the dispersing or the splitting influence can be accompanied or succeeded by a precipitation, which is due to a discharge of colloidal protein anions present or formed in the cell structures which are susceptible to the non-polar polar agents. For instance, at pH 5-6 precipitation by a cationic agent occurs with pepsin, but not with trypsin, because the isoelectric point of pepsin is pH 2.9, that of trypsin 6.5. The threshold concentration needed for producing a resting potential with a frog muscle by one of the quaternary ammonium bases has not been found to vary markedly between CS and ES2X in conclusion. One group of simple synthetic organic cations may be dealt with as exemplifying the typical bearing of the organophilic affinity. The dialkylamine ions comprising the series from CH3 to CF511 have been compared to the inorganic cations Li, NRB, and K with regard to the resting potential produced with crab nerves. The results show that the C4 and CS compounds develop a negative pole like K, or RB, while Ci and Cs resemble Li and A2, the anions of some natural nonpolar polar carboxylic acids. The greatest physiological significance among the natural nonpolar polar electrolytes can be assigned to the commonly occurring alkali salts of the fatty acids. However, the dual character of their ions in general is not so marked as that of the aforementioned compounds, because we are dealing with the carboxylic acids, which are weaker electrolytes than the sulfonic acids, and because the products of an intricate hydrolytic reaction of the soaps interfere. The fatty acids with longer C-atom chains are chiefly effective through their surface activity and their lipoid solubility. The classical proof of the nonpolar-polar character of their anions is the ability to form, on a water-organic solvent interface, a film held together by lateral adhesion forces and with the polar Q groups anchored in the water, the nonpolar C chains oriented more or less perpendicularly toward the organic solvent. The nonpolar-polar character of the lower members of the series, C2 to C4, or C5, is too weak to show a behavior in the hydrotropic series of anions different R. Höber unpublished experiments. From other hydrophilic anions, that is, they are located in the series between Cl and SO4, P2 and 93, like lactate, pyruvate, tartrate, and in others, C6T. 8. Muscle or nerve relative to CLL and enter erythrocytes somewhat slower than TL8, P337. However, beginning with or beyond valerate, the higher members of this series induce a negative pole, indicating organophilic affinity in correspondence with which the threshold concentration falls. The reversibility of these effects decreases with the increasing length of other natural nonpolar polar carboxylic acids are the conjugated bile acids, for example, glycocholic and taurocholic acids. For many reactions, they are known to be hydrotropic, they are surface active, and their local application at 2x10 molar to a frog muscle leads to a K-like potential effect. In contrast to this, even a saturated solution, about 10 molar of dihydrocolate, has no influence, in accordance with its surface inactivity, which probably can be accredited to the presence in the ring system of 3 keto groups. Finally, benzoate and salicylate belong to the same physicochemical and physiological category of organic anions. Each and accordingly have been shown to induce a depolarization of the muscle membrane at very low concentrations, see also SEC. 248. 3. The influence of nonpolar-polar ions on muscle excitability. 3 groups of synthetic nonpolar-polar organic anions, P337, have been investigated as to their power to alter the resting potential. 2 of them have presented a uniform reaction, namely the alkyl sulfates, GSCEOOC12, and the dialkyl succinyl sulfates, 2C4, 2C5, TOCA6, and TOS8. As aforementioned, these 2 groups induce negativity, which may be referred to the strong attachment of the nonpolar half molecules to the fiber surface. On the one hand, and the strong pull of the polar half molecules toward the water on the other P336. The effect can be assumed to be a distortion of the membrane structure. Parallel to these studies, the effect upon excitability has been investigated by stimulating pretreated sartorius muscles with condenser discharges, maximal stimuli, 1 in 10 to 20 seconds. Again, the reaction was uniform. A decline of the height of contractation over a period of time, dependent in its duration upon the concentration and the strength of the contrasting nonpolar and polar forces. It seems as though even with small concentrations, the membranes cannot resist the powerful dual stresses. However, the effect of the third group of R.H. unpublished experiments, these agents, the aryl sulfonates, is diphophate. So far, on the one hand, benzene, toluene, xylene, and isopropyl benzene sulfonates, and on the other, octyl and dicyl benzene sulfonates have been studied. The last two substances, resembling the alkyl sulfonates and the dialkyl succinyl sulfates in the longer C chains of the alkyl rests, are similar to these in their properties, namely in producing, even in the smallest concentrations, decreasing contractility, however. With the others, at variance with all the last mentioned compounds, the decrease of contractility is preceded affinity, which is indicated by their lower surface activity. Only after exposing the muscles to high concentrations of these weaker agents, for example, xylene and isopropyl tolusulfonate, do they respond from the beginning with decreasing contractions. Evidently, these substances can be compared to those earlier described inorganic ions, pages 289-290, which also in low concentration have a stimulating and in high concentrations a depressing effect. Their influence, again, can be interpreted as a loosening process in the surface membranes, which, if allowed to progress only to a somewhat diminished coherence of the structural units, is correlated with activation. But beyond that, results in real disorganization, which is incompatible with function. However, with regard to certain discrepancies, it should be kept in mind that the mechanisms of altering the surface membranes by inorganic ions and by nonpolar-polar ions are different. See pages 290-9F. Concerning their influence on the potential CP340, the conjugated bile salts also may be mentioned. These, with their highly organophilic ring system and their less hydrophilic carboxyl group sticking to the surface membrane, lower the muscle excitability slowly and often irreversibly 10 more. Further, the aforementioned quaternary ammonium salts, P339, likewise, are inhibitory at and above threshold concentration. Bioelectric potentials have been discussed in this section chiefly as providing evidence concerning the surface structure of cells, their sensitivity toward various agents, and the physiological implications of the concomitant alterations. In these respects, it seems particularly promising to extend the studies to the giant plant cells E. allonia, Halocyst, Nicua, Cari, which offer excellent possibilities for measuring membrane potentials across the cell surface. Similar to the Potsdam geometric measurements across the membrane of the giant axon of the squid, which have been mentioned earlier, P31. However, the characteristics of the latter object permit drawing more clear-cut conclusions than the experiment with the plant cells. For, in order to carry out transverse measurements in plant cells, one electrode is pierced through the cell wall into the large sap vacuole and is thus separated from the outside electrode by the vacuolar membrane, the protoplasmic mantle, about 10 microns in thickness, and the exterior plasma membrane, whereas in experiments on nerves, the electrode is placed in the axoplasm, the exterior one lying outside the single external axoplasm membrane. The more complex physiological conditions in plant cells become particularly obvious if the electrolyte concentrations in the two solutions adjacent to the protoplasmic membranes are changed. The normal resting potential of Halocystis ovalesi, for instance, is equal to 80 MV with the positive pole outside, as with muscle or nerve. After the cell sap has been substituted by seawater and the outside and inside seawater, both brought from the normal pH of 8 to pH 5, the reaction of the sap. The potential difference is still 80 MV, although there is no concentration gradient whatsoever between the two sides of the protoplasm. This is all the more unexpected, as the natural positive potential could be referred to a preferential membrane permeability to K, which in the sap of Halocystis ovalis exceeds the K of seawater 30 times. K equals 0.3 molar equals 0. Thus, it appears that the two membranes have different properties, and in combination they resemble the artificial asymmetrical membranes which have been built up by Buechner, Wilbrant, and others. Wilbrant, for instance, has pasted an ordinary cation-permeable collagen membrane to one impregnated either with a basic dyestuff or with an alkaloid salt, and hence anion-permeable P318, and thus obtained a double membrane which with 0.001 NKCl on either side yields a potential of several hundred MV. The next problem is to explain the direction and the height of the potentials of the giant plant cells, each of which has its significant value. The inside K apparently plays here no role. For intense, the potential of Valonia is 8 MV, with K co equal 42, and as already mentioned, substituting in Halocystis ovalis the normal cell sap, 0.3 molar KTL. By seawater, 0.0 fails to diminish the positive potential. However, outside K affects the potential in a way similar to that observed earlier with muscle and nerve. In other words, raising Co decreases the positive potential more and more, eventually to the value expected with a selectively K-permeable membrane. For example, in experiments with Nutella, where raising CoO as TL from 0.001 molar to 0.01 molar is followed by a decrease of electromotive force equal to 56 MV, that is. Nearly the theoretically expected value, however, according to more recent observations with holcysticity, this decrease often fails to appear unless the duration of the experiment is short. Whereas, in the corresponding experiment on muscle and nerve, the depolarization by CoO is fairly stable and is correlated in its degree with the concentration gradient, J 320. Here, the immediate and rapid decline of the potential is followed by a slower or faster recovery during the continued presence of the initial Ki concentration. This Rietzstein is likely to be due, at least partially, to a change of permeability, probably based upon an active reaction of the external plasma membrane, as may be concluded from chemical studies about the ion permeability of the giant plant cells. But before discussing this viewpoint, we may turn at first to the influence of other ions upon the potential of these cells, taking into account that Na and Cl are predominant in the natural surroundings of these cells, and that Chi is fairly indifferent. The normal positive potential of giant cells has been interpreted as due chiefly to the greater membrane affinity to Cl. Compared to that of NO, this hypothesis has been tested, is that but all the other anions tried, no three so far, formate, acetate, propionate, butyrate, lactate, pyruvate, glutamate, diminish the positive potential or even reverse it according to their concentration. When the above anions are introduced into the cell sap of Halocystis, the normal positive potential increases, but to a relatively small degree. Indicat in gene that the tendency of these anions to enter the protoplasm across the vacuolar membrane is less than that in the opposite direction across the external membrane. These findings are remarkable from two angles. First, the influence of the organic anions is such as could be expected from their place in the Hofmann-Sister series, P247, and sec. Ausert 222. They are physico-chemically rather indifferent, but possibly exhibit a dehydration effect upon the membranes. Second, these organic anions are supposed by Blinks to be accumulated in the protoplast as anaerobic metabolites, and their presence and their amount may be reflected by changes in the cell potential caused by effects upon the metabolic reactions. The plant cells in question differ greatly from muscle and nerve by irregular fluctuations of their potential difference. These are associated with lack and surplus of oxygen, with increase and decrease of CO2, with rise and fall of pH. And all these changes are correlated with the presence and absence of photosynthetic activity. Lack of oxygen depresses the electromotive force, especially in the dark, but light restores it through the liberation of oxygen. In CO2 also, the potential falls off, but recovers in light insofar as the C2 is used up in the synthetic reactions, and there is a simultaneous rise of pH. Lack of oxygen in nerves likewise has a reversible depressant effect, P 322, but it is small compared to that in teleesthesia, where low oxygen suffices to set down the potential after a few minutes. On the other hand, the potential and the excitability of the fresh water alga Nutella were found to resist pure hydrogen and nitrogen for many hours. We now return to the aforementioned observation that exposing holcystis to a surplus of K at first releases a rapid potential fall, which is followed preded as due to the release of an active change of permeability. We further refer to the aforementioned various fluctuations of the potential, which can be thought of as being indicative of the susceptibility of these structures to a variety of influences. Then these facts may be correlated with studies of SC. Brooks about the accumulation of Rb and of radioactive K by Valonia and Nutella, demonstrating the following intricate behavior. Either of these cations appears in the cell in a concentration surpassing that of the outside fluid, C-sec. 8, chap 3, subchap 2. But the rate of accumulation is rather independent of variations of the outside concentra, concerning other indications of special electrical reactivity of the giant plant cells to organic substances. See the papers of Osterhout and co-workers regarding the so-called potassium effect and the concentration effect. For the potassium effect, see J. General Physiol 13, 715, 19.0, 17, 105, 1933. Proxia Suki Expert. Bile. And Med, 82, 715, 1935. J. General Physiol Centration Effect. C. J. General Physiol 2013, 1936. 21, 707, 1938. T. On. This becomes more or less understandable if independent analyses of the sap gained by puncturing the vacuole and of the cell residue, that is, the cell wall and the protoplasmic mantle, are carried out. It appears that Rb and K are stored at first inside the protoplasm to a very high level, for instance with Rb 40 times. That only secondarily they are transferred into the sap, eventually long after their entry into the protoplasm, and reaching there a concentration level lower than that of the protoplasm. Later, the distribution of these ions varies as due to their escape from the protoplasm into the outside solution or toward the sap. According to the more recent studies of Brooks, these movements can display irregular fluctuations, possibly picturing the double influence of the two barriers, the external plasma membrane and the vacuolar membrane. The natural barriers delimiting the protoplasm from its environment have been found, by virtue of their more or less selective permeability to ions, to be the seat of the bioelectric potentials both in animals and in plants. JN the following section, the physiological counterpart of this interrelationship WDLB studied. A membrane, when interposed in a potential originating from an exterior source and falling off through the bordering electrolyte solutions, induces certain changes in the ion concentrations on either interface of the membrane, which are signaled by various effects. The discussion concerning this membrane polarization as a physiological phenomenon will be preceded by the description of some basic observations on models of the two main types. The oil-like and the sieve, if a direct current is conducted across a system built up by an interface of, say, nitrobenzene interposed in aciduous hydrochloric acid, and at the beginning distribution equilibrium. At each phase boundary, changes of the ion concentrations take place, which are opposite to each other, and which increase with the length of time, until a stationary state is reached. This is due to the fact that the ratios of the cation and antimobility, that is, the ionic transfer numbers, are different in water and in the organic solvent. The result is that a system which at the beginning shows no potential difference builds up a counter-electromotive force of polarization, inducing a certain steady state due to back diffusion and redistribution at and near to the phase boundaries. After interrupting the polarizing current, the system returns slowly to the initial equilibrium state. A second type of polarization is bound up with changes of the transfer numbers of anions and cations, which are due to differential adsorption on the walls of porous membranes, see pages 307 and 3, like gelatin, albumin, agar, collodion, or due to the presence of a dissociable pore wall material. If a current is passed, for instance, through a gelatin membrane lying in nasal solution at a pH above the isoelectric point, so that the membrane acquires a negative charge, the anions are relatively retarded, CP 319. One result of the arising inequality of ion concentrations at the phase boundaries is an accumulation of base at the anode at the point of entrance of the current into the membrane, readily demonstrable by indicator. Again, a counter-electromotive force of polarization appears while the current is flowing. It is of particular interest to study with a sieve membrane the interfacial changes of ion concentrations with regard to the pore size by applying different electrolytes on either side of the membrane. Some results are shown in the following experiment. A collodion membrane is prepared with pores wide enough to allow both cations and anions to pass. It is bathed on one side by a neutral solution of potassium phosphate, on the other by NaCl. Current passed from the K2HP ore side to the NaCl side is found to be distinctly stronger than one passing in the opposite direction, the membrane being more permeable to IV and TL4, rectifier effect. When the experiment is performed in such a WA, a long collodion tubing is filled with K2HOPO4 and wrapped in gauze wet with NaCl, thus roughly imitating an axon, and when one small electrode as anode, another as cathode, arc attached to the gauze some distance from each other, so that the current flows from pole to pole partly inside the tubuloid, partly outside. The polarization is stronger at the anode, and spreads more around the anode than around the cathode. Proceeding now from these model membranes to physiological membranes, one new feature of great significance is evident. Due to the constituent hydrophilic colloid amphilites, the membranes, through polar changes in the concentration and in the composition of their ionic atmosphere, are subject to remarkable alterations of their colloidal structure, to condensing or shrinking effects, and to softening or swelling effects, and even to disintegration combined with gross changes of the resistance of the membrane as such, because of increased or decreased ion permeability. Particularly, changes in the concentration of HK, K, and of OH and H4 may have a predominant influence upon the structural alterations. Such an effect is proved by a great number of observations. The various alterations as brought about by sending a direct current along a nerve or muscle, the so-called electrotom, appears in the following manner. First, one end of an excised nerve is killed by crushing. An electrode is placed on this end, the other electrode on the intact surface, and the resistance measured in both directions. With the cathode at the living, uncrushed, and catalectrotonus, the resistance of the preparation is lowered, with the anode and electrotonus raised, both changes increasing with time and current strength. The nerve in this experiment displays a rectifier effect. This effect further appears when alternating current is applied to an intact nerve with electrodes attached to two places of its surface, because one half of the current cycle meets a greater resistance than the other. Second, with a weak current applied to the nerve of a nerve muscle preparation, catalectrotonus indicates a small resistance and raised excitability, with a strong current and even more lowered resistance. But a decreased or abolished excitability, a cathodic depression at some distance from a depressing cathode, one can observe also a somewhat lowered resist third. The catalectrotonus probably is due, at least partially, to a surplus of inside K of outside Cl being shifted to the membrane by the outgoing current and thus producing a loosening effect. K salts applied directly to a nerve or muscle are known to raise excitability at small concentrations, to depress it at higher concentrations, and to produce, therefore, rectification is decreased or abolished by KCl, mainly because the difference of permeability between K and Na disappears, our Gutman IC. Fourth, inexcitability by KK can be canceled by an anelectrotus, although anelectrotonus by itself is a depressant agent due to its condensing influence. Also, Ca produces inexcitability, but not by loosening the membrane structure like K, rather by condensing it, P3O3. For this reason, the depression by Ca can be canceled by a catalectrotonus. This interpretation is in good agreement with experiments on the fiber-like long giant plant cells and Atelidon, which respond to an electrical stimulus by releasing an excitation wave, Q316. The height of this wave is modified as to whether the wave is superimposed upon the regular permanent plasma membrane potential, or upon a potential which is shifted to another height by an outgoing or by an ingoing polarizing current, or by application of, for instance, the normal membrane potential of Nutella can be depressed by KCLL even to 0 with 0.05 m when separate ingoing electrical stimuli of increasing strength. Starting with 2.5 GS are superimposed, no potential wave indicating polarizability can be demonstrated until 15 CXAA centimeter or more are applied. Then counter-electromotive forces of polarization reappear. This is analogous to the behavior of the nerve muscle preparation in Woronzo's experiment, where the paralyzing effect of K is antagonized by establishing an anelectrotus. In another experiment with Nutella, the normal membrane potential is brought to 0 by a powerful stimulus, and is then rebuilt spontaneously within 10 to 15 seconds to its original height. During this time of recovery, test stimuli show the counter-electromotive force of polarization to grow progressively. Sixth, finally, on sending an interrupted direct current of moderate strength in one direction along a muscle, the heights of contraction decay gradually until after some time, the muscle stops twitching entirely and seems to be fatigued. However, after reversing the direction of the interrupted current, the muscle immediately starts contracting to the initial height. But again, it is fatigued after the same interval. This can be repeated many times when Dungsfekt of F. Skiminski and thus is proved to be not a real fatigue, but rather a cathedral depression. In this state, the muscle loses its excitability due to the repeated loosening influence of the outgoing current and the concomitant increase of ion permeability, which can be rapidly counterbalanced by reversing the current, and thus subjecting the loosened area to the condensing influence of the anode. Correspondingly, in experiments upon the giant axon of the squid, which is passed by a direct current of adequate strength, it has been found that the electrical transverse conductivity can be raised at the cathode to the same value as obtained during normal activity, and at the anode diminished to a minimum not far from 0, which could be supplemented by many others. Fairly conclusive evidence has been afforded in favor of the concept proposed 40 years ago, chiefly on the basis of experiments concerning the influence of inorganic ions on resting potentials of muscles, that excitation is a reversible increase of permeability to ions, brought about by a limited and transitory dispersion of the colloidal structure of the plasma membrane. This result leads to the question whether they are not involved in this loosening effect additional factors beyond the influence of the inorganic ions which necessarily appear in the vicinity of the membrane during excitation in concentrations other than when at rest. The chemical complexities of the plasma membrane should be considered, and perhaps still more the very characteristic specialized composition of the nerve membranes, including in their constituents split products possibly capable of acting as precursors to elicit reversibly, and in small amount, strong bioelectric effects. SAC pages 337, 339. These considerations tempt one to add to the conception of the excitation wave as being, according to the old Stromkin theory of LL, Hermann, a mere electrical phenomenon, that is, a propagated depolarization-repolarization, another feature, namely, a chemical wave which consists of the liberation of the aforementioned precursors and of their reinsertion. The following points may be inferred to be favorable to such a concept. The lipoids of the nervous system consist of 1. the monophosphatides, lecithin and cephalin, p-pound 94, pound, as diaminophosphatase, the sphingomyelines containing, besides phosphoric acid and choline or cholamine, an unsaturated bivalent amino alcohol, sphingosine, and a fatty acid with pound 4C atoms, either saturated as lignoceric acid or unsaturated as nervonic acid, and 3 cerebrosides, which are lacking to the phosphoric acid and the base, but include again lignoceric or nervonic acid, respectively, as their hydroxy derivatives cerebronic and oxynervonic eats. With regard to the monophosphatides, it should be added that the fatty acids included in their molecule, oleic, linolenic, and arachidonic acid, likewise, are unsaturated components. Now, it is known from the work of Langmuir and of Adamon degrees that the presence of double bonds in fatty acids causes the surface films to expand at the interface, air-water much more easily than films with saturated chains of similar length, due to the fact that the double bonds attract the water more than a saturated linkage. Further, all the aforementioned acids are significant as electrolytes with a polar-nonpolar configuration of their molecules, and are likely, therefore, to display loosening and finally disintegrating cytolizing properties, as has been proved by their depolarizing effect on muscles, CPS 37F. It is true that so far it has not been proved that, while an excitation wave is running along a nerve, some such substances are liberated. But on the one hand, a widely distributed enzyme, a lecithinase, has been found in nerve substance. This is able to split the unsaturated fatty acids from the lecithin molecule, leaving about unsaturated fatty acids as essential growth factors. See Höber federation of residue lysolecithera, which has a strong cytolytic power. On the other hand, von Meralt has succeeded in showing that when excitation waves are running along a frog nerve, minute amounts of a very labile substance are set free, which is surface active and therefore is accumulated in the foam of an extract of the nerve, and which, according to various reactions, is similar to acetylcholine. These observations might suggest that during excitation, phosphatides are subjected to a breakdown associated with the liberation of choline, see further P359. But probably it will be difficult to reach a definite conclusion, since, according to von Meralt, not more than about 0.07 nerve of the active substance are produced by a frog nerve of 6 centimeter length and 50 milligrams. Weight stimulated rhythmically in such a way that 6 excitation waves are caught on its length by freezing with liquid air. Other indications as to the significance of acetylcholine during excitation of the peripheral nerve have been contributed mainly by Nachmanson. According to these studies, choline esterase, the catalyzer of acetylcholine, is localized in the nerve sheath rather than in the axoplasm. In chopped or ground peripheral nerve, acetylcholine is resynthesized from choline and acetate in presence of adenosine or prosophate and phosphocreatine. There is also another substance, likewise, prevailing in the sheath of the nerve fiber, diphosphothioamine, cocarboxylase. One may assume that, during depolarization and repolarization, breakdown and recovery of the nerve membrane, a complex of chemical reactions comes into action, the individual members of which appear and disappear in a cycle of phosphorylation and dephosphorylation. It seems not unlikely that in such a cycle there are involved also lipophosphatides, which in contact with enzymes like lecithinase would become the source of highly depolarizing chemical agents, the unsaturated fatty acids, V351. But up to now, this is nothing else but a hypothesis. Concerning the view that the excitation wave is more than an electrical phenomenon, but includes a series of chemical reactions, the following interesting observations of Osterhout and Hill upon Nutella cells may be mentioned. Their ability to respond to an electrical stimulation by producing an excitation wave, P316, is lost by exposure to distilled water, but is restored by water in which the cells have been standing for some time. Further, Liechti, I. von Meralt, and M. Reinhard Helv, Physial Acta 179, 1943. This seems to be due to the fact that an organic substance continuously leaves the protoplasm and must be newly produced if irritability shall persist. In searching for the nature of the substance, it was found that the restorative effect is produced by very heterogeneous compounds: guanidine, adrenaline, ephedrine, tetrathylammonium chloride, NH4CI, also addition of blood plasma, saliva, urine. White of egg and milk reestablishes the peace of the amylayers, and the conception that chemical events and the electrical processes are linked appears to be favored further by the illuminating experiments of Lillies regarding the numerous analogies between the nerve process and his well-known iron nerve model, displaying an electrochemical wave of alternating polarization and depolarization and alternating oxidation and reduction, which on stimulation travels along a wire of passive iron coated with an iron oxide film. Narcotics can be classified as organic non-cholecystériles. Acting essentially of this kind is a narcosis. Typically, narcotics do not enter into a chemical reaction with cell components. They are chemically indifferent. It is not customary to call IICN or cheo a narcotic, nor are the depressant barbiturates and many alkaloids among them, for instance morphine, which interact with protoplasm as weak bases or weak bases. Classified as such, because of their chemical indifference in general, narcotics pass the cell unchanged, and this makes it more conceivable that substances of a very diverse chemical nature are found to have narcotizing properties. The reactions they enter into with the cells are of a physicochemical rather than of a chemical nature. They make contact with the cells by secondary valences or Van Waals forces, changing the surface properties of exterior or interior cellular structures and microstructures, which become apparent as changes of dispersity, of hydration, colloidal aggregation, of dissolving power, of adsorption affinity. Which of these physicochemical reactions is in general the essential one or is dominant is still undecided. It seems useful to analyze this situation from a historical viewpoint. Around 1950 years after its great revival by Claude Bernard, general cellular physiology received exceedingly effective stimuli from physical chemistry. Following the fundamental studies about the osmotic pressure and the ionization of salts, numerous objects of animal and plant life were subjected to a study of their osmotic properties, with the result of increasing knowledge concerning cell permeability, particularly by applying osmotic methods to plant cells and to muscles. Overton had demonstrated that numerous inorganic and organic substances dissolved in water can be classified in one of two main groups: the penetrating and the non-penetrating substances. The first, in general, comprising unphysiological compounds, which often are poisonous, especially if the penetration rate is high. The second, including among many others, normal conjoinants of the cell interior, like sugars, amino acids, and inorganic ions. From these facts, two conclusions have been drawn by Overton. First, that, considering the highly diverse chemical nature of the first group, there should be detectable a common physical property furnishing the penetrating power by a suitable interaction with the cell surface. Second, that the paradoxical impermeability of the cells toward many common cell constituents, as demonstrated by osmotic experiments, suggests the capacity for an active uptake known already at that time in gland cells, and therefore named by Overton adenoid activity. Through this reasoning, general physiology has been confronted with one of its chief problems, the problem of active transfer, its mechanism, and its thermodynamics. Furthermore, it has acquired the concept of lipoid solubility, which was inspiring in the evolution of the lipoid theory, of permeability as well as in that of the lipoid theory of narcosis. The latter made its appearance as a byproduct, so to speak, of Overton's studies on cell permeability, insofar as it became obvious that the rate of penetration and the narcotic strength, expressed as the reciprocal value of the minimal narcotizing concentration, increase with the increase of the relative lipoid solubility measured by the distribution coefficient lipoid, water, see Sec 1, Chap 5. On the basis of pharmacological considerations, the lipoid theory of narcosis was presented simultaneously and independently by H.H. Meyer. Both these theories necessarily focused attention upon the plasma membrane, its chemical composition, and its structure as basis for its properties. Lipoids, namely organic substances resembling fat in their dissolving power, exist in great variety. Overton, H.H. Meyer, and many others used olive oil as a lipoid model, although from the beginning they were aware that this neutral fat could hardly exist as a normal component in the plasma membrane. Ether, benzene. Mixtures of benzene A with cholesterol or lecithin neutral oil with the addition of oleic acid and or diamine and others also have been used as model substances, especially in studies of problems of permeability. But none of them was found quite satisfactory, although in numerous experiments there appeared a rough correlation between distribution coefficient on the one side, penetration rate, and narcotizing capacity on the other. According to the Meyer-Overton theory, one would expect that, as the outside concentration was raised, the narcotizing effect would become evident with all narcotics at the same molarity in the lipoid inside the cell, irrespective of the chemical formulas of the narcotics applied. For instance, in experiments with tadpoles, the critical concentration in water is reached when the tadpoles just become immobilized by the paralysis of their central nervous system. In the case of propyl alcohol, the critical concentration is found to be 0.11 molar, and the distribution coefficient olive oil, water, is 0.0. The critical molar concentration in the lipoids of the central nervous system should then be 0.11x 0.13 equals 0.0141. However, the following E. Overton study in Ilber die narcose, Fischer Jenna, 1901, Leigh-Bill Roth, degrees shows in the last column not more than a rough approximation to the postulated constancy of the calculated distribution ratio for the lipoid to the central nervous system, although the values in the first and second columns illustrate fairly well the principle of the lipoid theory of narcosis distrib coefto. J. Olive oil, water narcotic cone for mice volume percent in air. It appears that the values of the last column are scattered around an average value of 0.45, but vary between the wide limits of 0.09. However, in another series of experiments by K. H. Meyer and H. Hopkimmel 21, where 17 narcotics in vapor form were administered to mice, a much better agreement with the postulates of the theory was attained. As is obvious from the fact that, for the critical molarity in the lipoid of the central nervous system, an average value of 0.07 molar was calculated, with deviations between only 0.04 and 0.1. This result has been referred by K2 Meyer to the fact that, at variance with the group of narcotics applied in the first mentioned series, alcohols, esters, ether, and acetonides, the narcotics of the second series, chiefly hydrocarbons and their chlorine derivatives, have a molecular configuration free, or nearly free, from polar groups. There are several reasons which might be discussed in this regard. But one major point seems to be that with apolar polar solutes, for example, alcohols, the distribution coefficient lipoid oil, water is more variable and more dependent upon the molar concentration applied in the experiment than it is with apolar solutes. For example, chloroform, see Sec 1, Chap 5, Sec 2. It has been mentioned previously, P. It has been mentioned previously. 356. The olive oil was never assumed to be an ideal model of a cell lipoid, or even to resemble chemically a natural lipoid. And furthermore, that in all organs, and particularly in the central nervous system, there certainly is a rather large group of substances which physicochemically could be regarded as functioning as lipoids, for instance, lecithin, or more generally phosphatides, higher fatty acids, and sterols. Indeed, all these have served as lipoid models in various experiments, mainly concerning permeability, Overton, Rulin, Callander, H. Meyer, and others. Considering investigations like these, K.H. Moyer, in continuing his studies on narcosis of tadpoles, has attempted to discover a solvent which does not display the irregularities evident in the experiments with the apolar polar solutes, Table 16. As a matter of fact, oline alcohol cis-GZO has been found to have suitable properties, as appears from the following table 18. In this experiment with 13 different apolar-polar narcotics, the critical narcotizing concentration in the lipoid appears to be reached with 0.03 mol. I, on the average, the values scattering only between 0.048. Although the critical narcotic concentration in water changes from 0.33 to in the choice of this model lipoid, K.H. Meyer was guided by the following, or at least by a similar consideration. As mentioned earlier, the lipoids of the nervous system are monophosphatides, diaminophosphates, and cerebrosidols. All of them contain the unsaturated fatty acids with 2 to 4 double bonds, CP351. Tentatively adopting the idea that the combination of alcoholic O groups and of long C chains may mediate the theoretical behavior, K.H. Meyer decided to try, instead of olive oil, oline alcohol. Furthermore, for somewhat different reasons, the monophosphatides should be dealt with as containing the long-chain acids oleic, linoleic, linolenic, and arachidonic acids. The unsaturated character of which possibly contributes to the lipoid property in question. Thus, it becomes suggestive to postulate that special lipoids are assigned to special functions. Narcosis appears in the nervous system of the tadpole when the concentration of the narcotic in the lipoid in question has been raised to 0.03 molar. Evidently, this lipoid is somehow essential for the normal functioning. So far, it is unknown why the dissolution of a narcotic in a lipoid to a certain concentration is accompanied by the characteristic functional alteration, viz., an inhibition of excitation, of contraction, of ciliary movement, or of protoplasmic streaming. Perhaps one can assume that the breakdown of the membrane following stimulation, pages 315 FF, 351, is the effect of the liberation of non-polar-polar unsaturated fatty acids from the fairly unstable molecules of the lecithins, the sphingomyelins, or the cerebrosids, and that this breakdown of a structural lipoid can be inhibited by charging it with narcotic. If the results of K.H. Meyer and Hemme compel one to believe that in the natural mixture of many fat-like solvents called lipoid present in the nervous system or in other organs, there is one of special functional importance, then, of course, there is no sense in determining the distribution coefficients with the mixture extracted from the organ, as frequently has been done in order to test whether the distribution coefficients of the various narcotics are really a measure of the narcotic strength, as postulated by the lipoid theory. However, on the other hand, it will be well worthwhile continuing the study, as in the experiment of K.H. Meyer and Hemme, the narcosis of various functions with reference to various individual lipoids, the distribution coefficients of which are well known. There is no other way to decide finally whether Overton and Ativitu Meyer were justified in drawing the conclusion that practically all chemically indifferent narcotics are able to produce a certain degree of inhibition if they have entered into their functionally essential lipoid to the same molar concentration. And so far, there is only this one remarkable experimental series of K.H. Meyer and Hemme in confirmation of this original deduction. 2. The adsorption theory of narcosis, a second physicochemical theory of narcosis, a second physicochemical theory of narcosis. Competing for a long time with the lipoid theory appeared after Trube in 1904, turned attention to the fact that a large group of surface active substances, that is, substances which lower the interfacial tension between water and a second phase, include many narcotics. He also pointed out that there is an interrelationship between the narcotic strength and the surface activity, adsorbability, like that existing between narcotic strength and lipoid solubility, and further that there is a quantitative connection, known as Trube's rule of the homologous series, which at first was assumed not to have an analog in the lipoid theory. The two theories even seem to exclude each other in that a solute taken up from an aqueous phase into a solvent like a lipoid is homogeneously distributed throughout the lipoid phase, whereas adsorption is the accumulation of a substance at the surface between the two phases. Thus, comparing the two theories, one may formulate the adsorption theory in conformance with the original general formulation of the lipoid theory of HH. Meyer and Overton, by saying that narcosis occurs when, by raising the concentration of a narcotic irrespective of its chemical nature, the interfacial tension is lowered to a definite point, due to its adsorption affinity toward the substrate of its inhibitory effect. In the following, it will be examined whether such a conception is justified. There are two main objections to such a general formulation of the adsorption theory. First, there are substances which produce narcosis without being adsorbable, as far as is known, upon any cell structure or constituent, and there are also substances which are adsorbable without being able to narcotize. Second, the rule of the homologous series has been interesting results can be expected all the more, since, according to Overton, the critical narcotic concentrations in plant cells and protozoa are about 6 times greater than in vertebrates. Overton Studien über die Narkose, G. Fischer, Jena, 1901. This striking difference may well be related to differences in the functional lipoids, found to be significant not only for adsorption, but also for other distribution phenomena. With regard to the first point, as has been mentioned before, P358, a series of hydrocarbons and their Cl derivatives, like methane, ethylene, acetylene, ethyl chloride, chloroform, carbon tetrachloride. Our surface inactive is, they do not lower the interfacial tension at the phase-bound airy water benzene or water oil or water air, although they are narcotics with their narcotic strength increasing in a homologous scries Evith-Klein length of the C-atom chain, as shown. For example, by the following table. Isenarcotic, one centrations for mice further, in a series of 11 narcotic substances examined by Lazarou and co-workers, there are only 2 which slightly lower the interfacial tension, cyclohexane-CO, which has 2 double bonds and ether containing oxygen. Evidently, only the apolar or nearly apolar narcotics are surface inactive. It follows that adsorption cannot be an unequivocal condition any factor in narcosis. On the other hand, substances which lower the tension of a phase boundary are not of necessity narcotics. Adsorbability is evidently bound up with a nonpolar-polar molecular configuration. It is this configuration which brings about a surplus concentration of the molecules in the interface by anchoring the polar hydrophilic part in the aqueous phase, while the apolar part has a tendency to enter the second phase. How much this concentration can be effectuated seems to be a matter of the mutual affinities of the molecular groups. For instance, naphthalene sulfonates, cytyl sulfate, and the salts of fatty acids with C-atom chains longer than about Ce are surface active, but ineffective as narcotics. However, they are cytolytics, for example, hemolytics, and display this property according to their surface activity. Isohemolytic solutions are isocapillary I2-tene, but with narcotics like the monovalent aliphatic alcohols and numerous others. Isocapillary solutions have been shown to be also isonarcotic. Possibly this is due to the fact that narcotics are non-electrolytes, the van der Waals forces of which operating in these non-polar-polar molecules in opposite directions are not strong enough and not enough remote from each other to prevent the non-polar lipophilic from entering the lipoid phase. As to the second point, Traub, investigating the capillary activity of organic compounds by the stalagmometric method, was struck by the fact that in a homologous series of surface, active non-electrolytes, for example, alcohols, the concentrations of isocapillary and of isonarcotic solutions differ from each other by a factor Q approximately equal to 3, as is illustrated in the following table 20. Showing the decrease of 2 consumption of goose erythrocytes and the inhibition of the frog heart, Warkiel capillary depressed Dachshund inhibition frog heart. This can be interpreted as significant to adsorption. In a homologous series, the addition of one CE2 group increases the interfacial activity. In other words, increases the distribution coefficient surface. Water by a certain factor, Q, in such a way that as the number of TH2 groups progresses arithmetically, the value of the distribution coefficients progresses geometrically, for example, 1, 3, 32. The numerical value of this factor, Q, is determined by the differences of the secondary valences of the nonpolar and the polar parts of the molecules involved. These account for the energy required to bring the substances in question into equilibrium with the two phases. 217 for this reason. With other distribution phenomena, other factors Q have been encountered too, and as appears in the following table 25. It is obvious that Trobe's rule does not refer specifically to adsorption, but that it is significant for various distribution phenomena, including the distribution organic solvent, water. In general, it is observed that the amount of the substance adsorbed from a solution to the surface of the second phase is not proportional to its concentration, but that an exponential curve, the adsorption isotherm Freundlich, accounts for the distribution in such a way that the distributive on coefficient surface. Water decreases with increasing concentration in the solution. So far as the adsorption of a substance to a cell or to a cell component is indicated by a functional alteration, it appears that the concentration action curve is not a straight line, as it would be if, for instance, a narcotic were taken up by a cell instead of remaining attached to the interface and entered a lipoid phase with a distribution coefficient lipoid. Distral coef benzene, water distri coef, cottonseed oil, water distro coef cottonseed oil, water distri gaseous va dasa, water 100 degrees distrib coef enzyme, water 50% inhibition of citrucino dehydrogenase. However, in adsorption processes, neither a straight line nor an exponential correlation can be expected definitely. There are numerous deviations from the usual isotherm relationship, just as the distribution coefficient solvent. Water, likewise, is by no means with regularity a constant. See, for example, Table 27. See further Sec 1 chap. It has been pointed out, K.H. Meyer and H.H. Hemme, that theoretically Tlie straight line relationship is more likely to be observed in adsorption experiments dealing with relatively low concentrations of the adsorbendum, whereas at higher concentration ranges, the concentration action curve should resemble more the common isotherm. These theoretical conclusions seem to be justified by observations concerning the narcosis of enzymes. Here frequently, the poisoning of an enzyme with various concentrations of the inhibitory agent has given evidence of the typical exponential curve of the adsorption isotherm. But, on the other hand, for example, the concentration action curve of invertase in water inactivated progressively by raising the concentration of ethyl alcohol approximates at lower concentrations a linear relation. And only at high concentrations does the exponential relation. Thus, we reach the conclusion that between the two main rival theories of narcosis brought about by the typically indifferent narcotics, p. 355, there is no definite choice except in the case of the nonpolar polar narcotics, which seem to be effective exclusively on the basis of a solubility in certain lipoids. But so far, this has been shown only by experiments upon the central nervous system. On the other hand, in reconsidering that the polar groups confer to narcotic molecules surface activity or interfacial activity, a special role is assigned to these, which was indicated already by applying the often rejected term of narcosis of enzymes. The narcotics in this case, as in many others, are believed to inactivate the enzymes. As microheterogeneous structures or any other cellular structures, irrespective of any lipoids being present or absent, by their attachment to the surface so that the substrate present in the fluid parts cannot reach the site of reaction. From the viewpoint of adsorption, this is quite a clarifying conception which, by various modifications, allows an approximation to the usual manifestations of narcosis. For instance, the enzyme invertase can be inactivated by appropriate amounts of narcotics, but this is due to the fact that the ordinary invertase preparations are contaminated by proteins which present a colloidal phase for the adsorption of the narcotics. For AFD, our careful purification of the enzyme, the sensitivity to narcotics disappears but the addition of albumin or globulin. Further, it has been demonstrated by many experiments that the inhibitory effect of narcotics upon cell reactions decreases progressively upon destroying the structural constituents of the cells by cutting, grinding, macerating, and expressing the protoplasm as it has been shown particularly with regard to cell respiration, or rather to the oxygen uptake. This has been interpreted as caused by the diminution of adsorbing areas. Also, it has been observed that in such a mass, narcotics of low solubility like thymol, toluene, phenylethylamine, which are known to stop growth and development of intact cells, appear to leave some enzymatic activity intact, instead of, however. In experiments of this kind, it must be borne in mind that the refractory behavior toward narcotics could be referred also to the striking specificity of the reactions of enzymes. For instance, the uptake of O2 present as substrate, but not with succinate, and the oxygen uptake of brain is inhibited by ethyl erythane, but not that of yeast, quastyl propylmethylcarbonyl ethyl. Compounds at 3 different pH values which immobilize the larvae within 5 minutes. It is obvious that the threshold concentrations of the alcohols are independent of the 7-TTF, that the paralyzing effect of the alkaloids increases with rising, and that of the barbituric acids decreases with rising PW. This is in agreement with both the lipoid theory and the adsorption theory of narcosis. As the undissociated acid is increased at the lower, the urate-associated base at the higher pH, and as the undissociated compounds are more adsorbable and more lipoid-soluble than the ionized salts. But by mentioning these experiments, we already crossed the borderline confining this discussion, CP 355, to studies concerning the indifferent narcotics, which react with the cells only by secondary balances. 3. Permeability and narcosis. The most common aspect of narcosis is that of abolishing excitability and of blocking the excitatory process, since on the broadening basis of an increasing amount of experimental data, it has been rather generally accepted that excitation is accompanied by a rise of permeability, particularly of permeability to ions. It is suggestive to believe that narcosis is dependent upon the adsorption of the interfacially active narcotics to those structures in which the excitatory process takes place, and the resultant exclusion of these from the complex of chemical and physicochemical reactions constituting the process of excitation. In this respect, narcosis is a decrease of permeability which, considering the electrical manifestations of muscle and nerve, may be thought of as being a decrease of permeability to ions, but also may include permeability to other solutes and to water. The adequacy of such a concept can be tested in model experiments on sieve-like membranes, the pore diameter of which, due to the fairly great rigidity of the building material, is stable and approaches molecular dimension. As in the dried collodion membrane of Michaelis, the following observations are especially pertinent. With membranes of a certain porosity, the rate of diffusion of glucose during the first hours or days decreased progressively to a low constant value. This can be considered as due to the fact that these artificial membranes, instead of having pores with a uniform diameter, contain an assortment of pores of different widths, so that only a small percentage of the pores is available to the relatively large glucose molecules, and that gradually more and more of these channels become clogged by the sugar molecules. Correspondingly, one may assume that interfacially active substances, such as many narcotics, form an adsorption layer upon the pore walls, and the passage through the channels to a degree dependent upon the molecular volume of the penetrating substances. Even small molecules, such as water, could be prevented from entering. Some experiments of anthocyanine seem in favor of this assumption. In one set of experiments with toast tuba-shaped collodion vessels filled with 0.5 molar sucrose solution, the rate of osmosis of water was studied with and without urethanes, C3 to C, or alcohols, C4 to C, in isocapillary concentrations on both sides of the sachet. The osmosis was markedly and reversibly retarded by the narcotic. In another set of experiments on the rate of diffusion of CN and Cl ion, similar results were obtained. One membrane was found completely impermeable to CN after the addition of 0.1 mol butyrethane, Furthermore, there are quite a number of experiments on physiological objects, the results of which resemble the key experiments on the artificial membranes. Some of these are concerned with the entrance or the escape of substances across the surface of red blood corpuscles. The slight spontaneous loss of electrolytes, which follows rinsing the corpuscles with sucrose solutions, and which is indicated by conductivity measurements, is diminished by a proper amount of narcotic. Likewise, the exchange of SO4 and Cl across the selectively anion permeable surface is retarded by various narcotics, ureas, substituted ureas, and alcohols. E2 volume increase following the suspension of human erythrocytes in solutions of slowly penetrating non-electrolytes, glycerol, erythritol, xylose, arabinose, CC4P. 240 also is slowed down in isocapillary concentrations, alcohols, and urethanes. To the interpretation of all these observations, one principal objection may be made. The observed changes refer to rates, whereas narcosis is, in essence, an equilibrium state in which time does not play a role. With a particular drug concentration, one can establish a certain state of either complete or partial narcosis for an indefinite length of time. However, under certain conditions, rate measurements also are doubtless indicative of narcosis in the ordinary sense of being an equilibrium state. For instance, in the experiments of Anselmino and Hoenig, swelling of erythrocytes in glycerol solution is due to 230. These experiments have been repeated by E. Ponder and J. C. Abels, Proc. and Med. 86, 551, 1937, with results conflating with the conclusion which may be drawn from the experiments of Anselmino. Urethanes again were found to retard the diffusion rate of CN, but the diffusion of other anions, including Kl and cations, remained quite unaffected. In nearly all the model experiments with collagen membranes, the membrane material has been considered to present a rigid sieve structure, Sec 5, pages 317. However, recently, evidence has been provided, C. Kate Solner and P.W. Beck, J. Channel. Physiol 27, 451, 1944, that certain organic non-electrolytes and electrolytes can enter the membrane substance as an organic solvent. They can be accumulated in the substance and can cause the membrane to swell, or they can be accumulated by adsorption at the internal interfaces. But hydrophilic organic compounds, like sugars, polyhydric alcohols, and anions of hydroxyl carboxylic acids, P. 247, are inert. Organophilic, carbophilic compounds, for example, valeric acid, valermide, amyl alcohol, are active. These processes, at least beyond a certain concentration range, must reflect upon the permeability. The entrance of glycerol, accompanied by the entrance of water until hemolysis occurs. This swelling is retarded more and more by increasing concentrations of alcohols according to the length of their C chains, C3 to 7. Thus, very small concentrations of heptyl alcohol suffice to prevent the hemolysis for a long time. It is probable that nearly the entire assortment of pores of the sieve-like membrane is occluded by the adsorption to their walls of these large molecules. And a nearly complete narcosis of the ordinary exchange is similar to that mentioned before concerning the diffusion of CN across a collagen membrane, and its blocking by butyrethane. Thus, the decrement of rates in these series of experiments can be interpreted to mean approach to narcosis as an equilibrium. Additional evidence about the increase of the resistance to the passage of solutes caused by the absorption of narcotics in the pathways which extend from the exterior to the internal surface structure of cells and tissues is furnished by studies concerning the bioelectrical properties, old observations on the resting potential of sartorius muscles, which arises during the local treatment with inorganic neutral salts, P. 313, and which to some extent can be compared to the normal reversible electric reaction after excitation, have shown that the curve of its gradual development takes a more flattened course in the presence of a narcotic. This again can be accounted for by assuming that adsorption of the narcotics decreases the porosity of the structures in question. The same problem has been studied in muscles with and without the presence of narcotics. It was found that low concentrations of a narcotic bring about a reversible increase of the electric resistance, higher concentrations, and often irreversible decrease. These alterations can be referred to effects upon the membrane of the fibers because the increase or decrease of the resistance is accompanied by only a slight change of the electrical capacity. This corresponds to the changes during excitation, Nutella, squid giant nerve fiber, where the breakdown of the membrane resistance is, again accompanied by not more than a slight decrease of the capacity. In this connection, it should be mentioned that permeability is affected thus. Certain substances fail to penetrate the surfaces of cells or organs. Concerning a more thorough discussion of the distinction between rate and equilibrium, see MH. Jacobs and A.K. Parpart Bio Bull 62, 313, 1932. Compare further in regard to decrease of permeability the experiments on fish eggs by J.F. McClendon, M.J. Kiziel, 38, 173, 1915, and on plant cells by Lepeshkin, Berdistras 29, 349, 1911, A.T.R. Fithel, Biochem. Hist. Chapter 112, 259, 1920, and H. Lully's, Pflugerns, Arch, F. Digis Physio 207, 8, 1925. Unless energy is made available for the transfer by metabolic reactions, these sometimes, but not regularly, are initiated by nervous excitation. It was mentioned earlier, p. 366, that in nerves it is the self-propagating complex of chemical and physicochemical reactions, the excitation wave released upon a stimulus, which is possibly stopped by the layer of the adsorbed narcotic molecules covering the nerve membranes. In a corresponding manner, the energy-providing aggregates of molecular structures in actively absorbing and secreting organs may be thrown out of action by narcotics. In contrast to the so far referred ample and various evidence that permeability is decreased by narcotics, there are several observations displaying increase under conditions where there is no reason to assume that the results are due to impairment by excessive concentration. Thus, according to Jacobs and Parpart, GD, the permeability to glycerol of the erythrocytes of certain animals, man, rat, rabbit, guinea pig, is decreased by N-butyl alcohol in a certain range of concentrations, whilst that of ox, sheep, pig, dog, cat is increased. See also regarding cat's corpuscles, H. Davson, J. Fell, and Comp Physio, 16I 317, 1940. And according to Barlin, the permeability of cells of Cara to ethylene glycogurea, trimethylcitrate, and hexamethylentamine, substances which can enter the cells by simple diffusion, is increased by ether applied in such a concentration that the protoplasmic streaming in the cells is reversibly slowed, though not more than 10 to 50%, whereas the permeability to lye-on under the same conditions is unchanged, or possibly slightly decreased. These conflicting effects need further investigation. In conclusion, although in this chapter the adsorption theory of narcosis has been emphasized, the lipoid theory must be given due consideration, inasmuch as certain lyl-oroids appear to lose their ability to take part in excitatory process through preferential dissolution of the narcotics, P359. Furthermore, it should be mentioned that the concept of narcosis is sometimes applied in a much broader sense. For instance, in speaking of MG narcosis or Vagicire narcosis of the central nervous system, one is confronted with the question whether the associated phenomena of reversible paralysis can be paralleled to those treated in this chapter. From the view joint of traditional classification, these effects certainly cannot be recognized as narcoses. The classical types of narcotics are organic non-electrolytes, which, according to the studies of the last 40 years, have been found to act in the polyphasic system of cells and tissues, either by adsorption at the phase boundaries or by distribution to the lipoid phases. But should an extension of the conventional concept of narcosis appear useful, it would be adequate to supplement the adsorption and the lipoid theories of narcosis in organic ions, which for the purpose of electronarcosis are pushed by an electric current through a living tissue, are very likely to act upon the colloidal constituents of the cellular interphase, where adsorption or solution in lipoids likewise takes place. However, the result would be an excess generalization of the term narcosis, since many other reversible inactivations by ions, the paralysis by K or by Nunge, are polar organic ions, or the cathodic depression of nerve, and others also would have to be called narcosis Buechner R. Die Entstehung elektrischer Störungen in Lebenden, KCC Stuttgart 1020 of Overton, E. Studien Tiber die Narkose, G. Fischer Jena, 1901.